Ses deneme. Ses deneme. Ses deneme. Ses deneme. Ses deneme. Buna da bakalım mı?
Hi, good morning. I would like to invite Engin Oral to do his welcome speech.
Good morning. Can I have my slide presentation? Does it too much? Good morning, everybody, again, dear colleagues and friends. Okay, this is the one. It's working, this one, right? It is our great pleasure and excitement that I welcome you on behalf of the chairs hosted here in Vibrant City in Istanbul. This conference stands as testament to the power of collaboration, which is very, very important, and synergy among leading institutions in the world and experts in this field. I am delighted to announce that this event is being held in collaboration with two estimate groups. And the first one, Endoke group from the University of Oxford, and the second one, Expect team from the University of Edinburgh. Their contributions and expertise have been invaluable in shaping the content and vision of this meeting. I thank them for their effort and their motivation and their time. Over the course of these 2 days, we will touch into latest findings, innovations, and best practice in the field of endometriosis and adenomyosis. Through scientific and keynote presentations and panel discussions, we will explore the multi-phased aspect of these conditions to discuss the underlying pathophysiology, the impact of the patient's life, and the latest therapeutic advancement. These are the speakers that we invited from the England, and we have the online speakers from the France and the United States. And yes, night we will have the meet expert at breakfast the opportunity to discuss these authors, and the 6 authors will be discussed with you in our historical place, Professor Levy, called Professor Levy, just near there, here. And every table must be, and 10 attendant with the 2 authors to discuss the mentorship and this disease. In closing, I would like to express my deepest gratitude to all our speakers from Turkey and abroad, our sponsors and organization team, the figure team. They work hard to organize this event and attendance for their dedication and support and time. But these 3, our fellow is very, very important. They did everything for this meeting. The first one is the Fitnat Topbaş Selçuklu, and these are 3 of them from our Endo Young Specialists in Turkey group, and Fitnat did great job. She did everything from the travel agency, from the academic science, and from the prepare of this speech. And now she is doing the PhD thesis in Oxford group. The other one is Emre Papucu, another young group, but he has become just professor. He is working in Ankara, Turkey. The third one, and the Fırat again, are in our young group, in our society. He did a postdoctoral fellow in Edinburgh with the Edinburgh Expert Group. I would like to apply them for their hard working. Thank you. Thank you so much. Thank you so much. Now, I would like to hand the microphone to the Professor Ümit İnceboz, who is the president of our society and one of the chairs of this meeting. It is the professor.
Good morning, dear colleagues, dear guests. Welcome to this meeting as President of Endometriosis and Adenomyosis Society, Turkey. I'd like to thank you all for being here. What I'm going to do, I'm going to introduce my society, our society to you a little bit with the slides, with a couple of slides. And seen here, our society has been established 2009, and our founding president, Professor Engin Oral. And I'm in charge at the moment, and Professor Taner Usta was the past president. What is our mission? We have 2 missions, actually. The first one is scientific and the other one social. So our scientific mission is to work on endometriosis and adenomyosis, and also to exchange the current knowledge with national and international colleagues. Socially, we would like to increase the awareness of the society in terms of endometriosis and adenomyosis. This is our board members. These are board members. Obviously, we are not only 7. We have a huge backup, very proactive young colleagues. We call them endo specialists. They work very hard. They encourage us. They do lots. And proudly, I'd like to show you our international endo ambassadors. Professor Engin Oral is West Secretary at the moment. Professor Kutay Biberoğlu, I think he's not here at the moment, but he'll be soon. He is one of the board members of Asia Society of Endometriosis and Adenomyosis. Professor Taner Usta is in the advisory board of ESKE. Dr. Fitnat Selçuklu is West Early Career Board member. Dr. Göknur Topçu and Dr. Ezgi Darıcı are EL Young Core team members. And Dr. Fırat Büyüktaşkın also our ambassador. He worked in Expect Group as postdoc research fellow, proudly. How about our scientific activities? We do international and national meetings. We have books and bulletins. Let's have a look quickly. This is one of the international meeting of ours. It was last year in March in Istanbul again. It was ESCA regional workshop. We have also joined and own webinars, international ones. And we have also local national meetings. We go different places in Turkey and we gather with our colleagues and we discuss endometriosis and adenomyosis. And we call these meetings Endo Academy. And we have also webinars, national webinars, and also online meetings during which we discuss the recent literatures with the colleagues. We have 2 books. We have 2 books, one for the patients and the other one for the colleagues, or healthcare professionals, I would say. And we have bulletins, 3 monthly online. We include recent articles as well as some interviews. We have also social activities. Obviously, we have social responsibility to our public, to our nation, or to human being. We go to schools, we go to high schools, universities, we give them lectures on endometriosis and adenomyosis. Also, we do some public seminars. We do Endomarch activities. March is important because of endometriosis, obviously, Endometriosis Awareness Month. So we do live broadcasts on endometriosis from our social media, and we do some awareness activities like theaters, sports activities, and marching, etc. These are some of the photos of our activities, social activities from the school gathering, I would say. And these are the awareness, as I said, sport activities and marching and so on. We do lots in March especially. Obviously, that's not all. We work very hard. We have to work very hard. And at the end of my talk, I'd like to thank, to express my appreciation first. I'd like to express my sincere thanks to experts who are going to share their knowledge, their expertise with us today and tomorrow. And also to you, obviously, being here, because without you being here, there wouldn't be a meeting anyway. And also, I'd like to thank to our sponsors. Without them, we couldn't afford or we couldn't realize this meeting. So I would like to thank all of them. Obviously, to Figure organization, they did a lot. They work very hard during nights and day. And lastly, I'd like to our team, our Endometriosis Adenomyosis Society Turkey team. We work very hard, and we have to work very hard. Thank you very much.
So thank you very much for that wonderful introduction, particularly to your wonderful active society. I'm Professor Philippa Saunders, and I'm here representing the Edinburgh Expect team and my colleague.
Welcome, and thank you for joining.
So it gives me great pleasure to introduce our first speaker, who's Professor Karina Zondervan, if I can get my technology working. Airdrops and things. So Professor Zondervan is Professor of Reproductive and Genomic Epidemiology and Head of the Nuffield Department of Women's and Reproductive Health at the University of Oxford in the UK. She's co-director of Oxford Endometriosis Care Centre, and she's had a very distinguished and excellent research career at looking at understanding the pathogenesis of women's health disorders, particularly through genomic, molecular and environmental epidemiology. And we're delighted that Professor Sonderhan is going to share her expertise and knowledge with us and kick off the meeting. Welcome, Karina.
Thank you so much, Philippa, for that kind introduction. And first of all, thank you, in particular to the organisers of this meeting. I'm absolutely delighted to be here. So, in particular, Professor Oral, for really taking this fantastic initiative. And I'm delighted that we're here on the 15th anniversary of the Endometriosis and Adenomyosis Society of Turkey. So, a special 2 days, I think. Yes, I have about half an hour to give you a bit of a whistle-stop tour of the etiology, what we understand about the etiology of endometriosis. The first few slides I'll go through very quickly because I'm sure it won't need any introduction for most of you, but just in case some of you have registered for this conference wanting to know a little bit more about endometriosis itself. As you may know, it's an often chronic inflammatory condition defined by the presence of tissue resembling endometrium outside of the uterus that is usually predominantly within the pelvis, but there's also increased recognition of extrapelvic endometriosis. It's an estrogen-dependent condition, and it's a major cause for pelvic pain and infertility, although obviously we recognize more and more there's a whole array of symptoms that is associated with endometriosis, including, for example, fatigue. In terms of diagnosis, gold standard remains surgical diagnosis, visual diagnosis through laparoscopy, although there's some role now and actually increasing role for imaging, ultrasound and MRI. As many of you know, endometriosis can look very different within the pelvis. The characterization is sort of mentioned here. And for a long time, I think physicians, surgeons, clinicians, researchers have wondered whether actually these presentations are all one disease or whether they represent different entities. And I'll come on to that a little bit later. As I'm sure you know as well, there is a classification that's now several decades old by the American Society of Reproductive Medicine. It's a score of 1 to 4 based on localization of lesions and extent of adhesions. I'm sure you're all familiar with the original paper with the sort of various doodles of what this would look like. And I'm sure you've had personal experience of looking at these pictures as well. Very broad brushstroke. There are exceptions, but stage 1 and 2 typically is combined to sort of smaller peritoneal lesions that could be superficial, whereas stage 3-4 disease typically has a varying involvement with endometrioma and more extensive adhesions. And this sort of dichotomy is important because I'll come on to that in terms of our research findings, what we know about endometriosis a little bit later. Now, what we also know is that there's only a very modest correlation with fertility based on this classification. There's really no correlation with pain severity. Increasingly, a different classification is used, which is comparing ovarian endometriosis or endometrioma with deep lesions and superficial endometriosis. And again, that's something that I'll mention, but in particular, Nilüfer Amioğlu, looking at genetics, will come on to that classification later. Endometriosis has long been seen as a disease that only occurs in high-income Western countries. It's very clear that that impression is false and was really fueled until recently by difference, and still is, by differences in access to healthcare and also in differences in access to education of women seeking healthcare. We published as a very large collaboration over a decade ago now 2 studies, a global study of women's health the Women's Health Symptoms Survey, which focused on understanding global impact and symptom-based predictability of endometriosis across the world. Now, just very briefly, the study included a prospective recruitment of all women scheduled to undergo their very first laparoscopy for symptoms or for tubal sterilization. They had no previous surgical diagnosis of endometriosis. Data collection happened at each centre and followed an extensive questionnaire, and data were entered into a secure online database that was analysed centrally. What did that study find? And that was a study that was actually led by Kelechi Noaham, who, for whom it was his DPhil thesis. The patterns of diagnostic incidence will be very familiar to you, I think, if you look across the literature as well. So a wide range of diagnostic incidence of endometriosis, clearly very dependent also on whether a particular centre specialised in endometriosis and whether there was prior imaging before a decision was made on surgery. So that varied hugely across the different centres, with an average of around 50% of women diagnosed with endometriosis within who were symptomatic. And again, the classification of stage 1, 2 versus stage 3, 4, again varied per centre, again depending on what the focus and emphasis usually of that centre was. On average, women reported that their symptoms started in their mid-20s. On average, they consulted their doctor about a year afterwards, and on average, they were diagnosed 6 years after their initial consultation. So on average, a delay, if you want to call it that way, of about 7 years between first symptoms and surgical confirmation. That diagnostic delay also varied, but perhaps not so well. I mean, there's quite a variation between the centres. Typically, those women who had access to government-funded healthcare had to wait the longest compared to private insurance or individual insurance, individual-funded healthcare. As you can see here, the dark bars are the government-funded healthcare. But again, there were big differences, and I think one observation that was notable was that the two centres in China actually had the shortest delays. Global societal burden, we know for endometriosis is huge. So population prevalence, again, has sat estimated around 5 to 10% for many years now, translates to about 190 million women worldwide. We know that there are very significant symptom-related consequences for quality of life of women and a very high economic burden, and which has been estimated as similar to, say, type 2 diabetes, which surprises still many people. Now, on to etiology. I think one important statement to make or consideration to have is that we use different terms for trying to understand the biology of a disease. So etiology is not the same as pathophysiology or pathogenesis. Etiology is all about causes of disease, whereas pathophysiology looks at what physiological processes along the way are disordered in a disease, which may be causative or may just be part and parcel of the disease process. And pathogenesis is again looking at that biological mechanism that leads to disease and its maintenance. But it doesn't necessarily, those processes don't necessarily have to be causative. So that's sort of pictured here in this diagram. So the postulated origins of endometriosis are largely, as I'm sure you're aware, most peritoneal disease is thought to arise from retrograde menstruation, where menstrual debris with viable endometrial cells enters the pelvis, and these cells adhere to peritoneal surfaces. But there's certainly also a place for potential for coelomic metaplasia, which is the in situ changes of cells that arise into sort of endometrial-like structures. and vascular and lymphatic metastasis, so the transport of these cells through other systems. So, and we know, for example, extrapelvic lesions cannot be explained by retrograde menstruation alone. Now, in terms of pathophysiology and pathogenesis, we know endometriosis is very complex. We understand quite a bit about what the pathophysiology looks like, what the disease looks like in terms of its underlying biology. So we know there's a localized inflammatory response. There's some evidence there's even a systemic inflammatory response, but certainly localized, that inflammatory response is very pronounced with chemokines, prostaglandins, and various interleukins being produced, and the recruitment of various different immune type cells, particularly macrophages, being recruited to the area. And there's also evidence there's localized immune dysregulation. So these macrophages have a reduced ability to essentially clear up these endometriotic cells, so reduced phagocytosis, and has reduced activity of natural killer cells as well. All these processes result in an increased propensity for angiogenesis, of vascularization and innervation, and we know that these macrophages co-localize with various sort of growth factors that lead to also nerval growth and increased sensitivity of these nerves to pain. And all of these inflammatory processes also promote the cellular adhesion and proliferation locally, and there's various molecules that are up or down regulated to promote that. Of course, really important is the hormonal influence on this, and we'll hear from Doug Gibson later on this. So there's a localized hormone response and progesterone resistance that also promote the growth of these lesions. Of course, there's systemic hormones that impact on this, and also there is increased evidence there's somatic genetic mutations that perhaps promote the growth of these lesions further and promote clonal expansion, although again, we don't really know whether these are sort of byproducts of this process. The key question, though, for etiology, which part of this is pathogenesis, part of the disease process, and which of these processes are actually causative in disease, which are altered. So a very nice figure in Philippa Saunders and Andrew Horn's review paper in Cell really hits this home as well, that the endometriotic lesion microenvironment is a very complex and altered microenvironment where all of these processes that I just described are involved and are altered in some way and are all promoting the growth of these lesions. Again, the question is, are these processes intrinsically altered in women who develop endometriosis and therefore play a role in causing the disease and its etiology, or to what extent, are they essentially a result of tissue being in the wrong place? Just checking my time. So hypotheses that have been investigated in the context of etiology, fairly concise. As mentioned, the origin of the endometrial tissue in the pelvis mainly thought to be from retrograde menstruation, although there clearly is a place for other mechanisms that explain extrapelvic endometriosis. And then investigators have mainly focused on these categorizations or these variety of potential factors, innate or acquired differences in growth of endometriosis, that cells and adhesion to peritoneal surfaces, innate or acquired altered hormone regulation, and innate or acquired altered immune system, and to what extent environmental exposures might play a role. And importantly, also, we know there are genetic risk factors which could underpin this innate propensity for these mechanisms to be altered in women with endometriosis. So how do we study etiology of disease? One of the big tools we have is epidemiology, because with epidemiology, we can make inferences around causation in human disease. So we have the classical epidemiology methods, which look at environmental and non-genetic exposures. So we infer causation probability from that through various means, and that depends on the study design. So, for example, a prospective cohort study where you measure exposures before diagnosis is actually established is the gold standard for this. But also looking at the results and asking the question, is there a dose response in relation to the exposure, and to what extent do we have replication data across studies are all very important. And then, of course, quite often, certain exposures that come out of these studies are then tested and validated in more experimental settings to see to what extent those findings actually hold true. And then at a genetic and molecular level, we can also do this through genetic epidemiology methods. And again, Nilofer will discuss that a bit later. So that gives us an opportunity for entirely hypothesis-free interrogation of the underlying biological etiology of endometriosis. And we can also ask questions about etiology of subtypes of disease. And causation through genetics is often inferred, but it also, again, requires replication evidence and then validation evidence through other experimental processes. So how do we apply this to endometriosis? Well, endometriosis is a notoriously difficult disease to study for a number of reasons. And they mainly relate to this difficult diagnostic process and the fact that we often only diagnose women with endometriosis after many years of symptoms and after many years of exposures. So endometriosis is really seen these days as a life course disease, which can start very early on in adolescence with various exposures and changes of lifestyle, etc., along the way, and symptoms and sequelae of endometriosis really even persisting beyond menopause. Now, this is a slide from Amy Schriffrier's paper published in 2018, a nice review of epidemiology. And it just, it only serves to remind us all how complex the diagnostic pathway is. And when we sample women for our epidemiological and clinical studies, they can be sampled in all sorts of different ways that result in bias in what we see. So comparison of risk factor study results are often difficult because of these selection biases. There's differences in diagnostic pathways between centres, etc. There's often little or no information on the time point for risk factor assessment. And case definition is often poorly specified, and control selection is difficult. Do we select controls from the same clinic, and they are essentially screened for endometriosis? Will they be fertile or unfertile, etc.? And then sample sizes are tricky as well. So if we take a very simple example, if we want to look at the impact of physical activity habits on the risk of endometriosis, what we want to study is that first bit of habits leading to endometriosis incidence. But what we typically study is a change in physical habits that occur after diagnosis. Now, again, this is just, these studies are all quite old now. I mean, there's various studies that have been published since, but just to show how different various non-genetic risk factor studies are in terms of their inclusion criteria, and there have been very limited cohort study data. They mainly come from the Nurses' Health Study 2, led by Stacey Misma in the US. So just to briefly take you through what we know about risk factors. So typically we say endometriosis risk increases with age, but actually from the cohort study, we know in terms of incidence, there seems to be a sort of U-shaped with a peak around the women in their 30s. Again, there seems to be an increased risk typically with education, higher education. We know now that's mainly related to access to healthcare. And also, we have in the past thought that there might be differences in endometriosis risk by ancestry, but actually we now know that's likely related to access to healthcare as well. And we really have very little evidence that there's an ancestry-based differential prevalence for endometriosis. And this is in contrast to for example, uterine fibroids, which are much more common in Black women. So we know menstrual cycle factors have really hit home that retrograde menstruation is an important factor in endometriosis. So shorter cycle length and longer duration of flow all have consistently been reported to be associated with endometriosis risk. The relationship with parity is tricky because, of course, infertility is part and parcel of the endometriosis process quite often. But again, there seems to be some evidence of reduced risk with increased number of children. There's a consistent association with low BMI and also with increased number of nevi and skin sensitivity, which we understand very little about. And then the other factor that's been consistently reported in the past has been a reduced risk with smoking, which has baffled quite often many people, has been interpreted as related to estrogen exposure. Now, it's important again to highlight that biases and also the complexity of the characteristics of women with endometriosis play a role here. So this is shown very nicely by Stacey Mismer in a Nurses' Health study. So if you look at cigarette smoking and you stratify by women whether they have been diagnosed through essentially the infertility workup through the pain route, then you really only see that reduced risk related to endometriosis in the infertility patients. Again, if you look at diet and healthy eating, that link only seems to exist for women who present with pain rather than with infertility. So again, it's really important to look at these risk factors very carefully and look at the underlying diagnostic process by which women have been diagnosed. So to conclude from the environmental or rather non-genetic exposures, cycle-related exposure risk profile suggests retrograde menstruation and or hormone exposure as etiological factors. And there's also this link with low BMI and increased number of nevi, which really suggest common etiological mechanisms, but they need to be studied further. And really, we have not much else to go on in terms of non-genetic factors. So then we come to genetics, and I'll just have just a very few slides on this because Nilofer is really going to delve into this. So we know that endometriosis is heritable, and we've known this for quite a long time, looking at family studies. So heritability is the proportion of disease in the population that's attributable to genetic factors. And for endometriosis, that's around 50%, with about 26% due to common variants. We know this from twin studies and other types of studies. And that's a reasonable heritability, actually, compared to other conditions. So within a very large consortium, recently published in Nature Genetics, the latest findings on genetics of endometriosis, and Nilofer is going to cover that, and that involved more than 60,000 women with endometriosis. Really an unprecedented number, and it's come up with some very exciting new results that Nilofer will cover. One thing to highlight, though, and this comes back again to our classification of endometriosis, which is that consistently in these genetic studies, stage 3/4 endometriosis has a much stronger genetic signal, is much more genetically driven than stage 1-2. And all the effect sizes of the loci that we found are all greater for stage 3/4 disease. And that's important for us trying to understand endometriosis per se and its subtypes. We can try and unpick that through phenomics and phenotypic dissection. So the question we have now is, are those entities very distinct? Do they arise separately and distinctly, or does stage 3/4 evolve under certain genetic conditions from stage 1 and 2. And what does that really mean? And Nilofer will really delve into this. You know, is this really, I mean, stage 1, 2 versus stage 3, 4 is a very crude separation. What does that mean? I mean, are we looking at peritoneal lesions versus ovarian, endometrioma? Are we looking at distinct inflammatory subtypes? You know, are we looking at propensity for extensive adhesions, so fibrogenic potential, for example? And that's something that really is an important focus. And really, is this distinction just a mask for a lot of different subtypes that actually underlie endometriosis? And that comes back to an analogy we often make, which is with cancer. So if we look at where the breast cancer field was a couple of decades ago, where if a woman was diagnosed with breast cancer, there was one type of therapy, and that was it. Whereas now, on the basis of molecular profiling, we know there are subtypes of breast cancer that are treated very differently, that have very different risk factor profiles, and very different prognoses. So we think that endometriosis is essentially, we need to look at endometriosis in a similar way. And of course, we need to look at endometriosis comorbidities. We know there's increased risk of autoimmune and other immune diseases. There's comorbidity with other hormonal traits. There's a strong comorbidity with uterine fibroids, for example. There's comorbidities with cancers, particularly ovarian cancer, etc. So untangling this in a phenotypic way, but using genetics and using the biological samples that we have is really important. So my last few slides is just to introduce you, if you don't know already, an initiative that really helps us moving this field forward, which is Worth Effect, the Endometriosis Phenome and Biobanking Harmonization Project. So we really now have the tools to do this type of work at much larger scale. So back in 2013, we got a working group together of 34 academic centers and 3 big industry partners. At the time, that was Bayer and Roche and AbbVie, to really work on unified approaches to data collection and to sample collection. And that resulted in 4 papers with freely available tools, questionnaires, and sample collection protocols to really ensure that in different centers, data and sample were collected in the same way so that it could be combined for large-scale collaborative studies. As of today, we have 58 research sites in 24 countries, and really that gives us an unprecedented opportunity to conduct the large-scale collaborative studies that we need to do. And you know, really, really pleased that here in Istanbul, that work is going on as well. So, just to conclude my overview, I think we know that the pathophysiology of endometriosis is really getting increasingly well characterized. We understand these sort of biological processes probably a lot better than we did a decade ago. And we have several talks on this later in the program. But we have few robust epidemiological, etiological factors. So we know an increased risk with an exposure to menstruation, pointing at retrograde menstruation and hormonal regulation as etiological factors. We have an increased risk with a lower body mass index and with propensity to have nevi and sun sensitivity of the skin. And we really don't understand that mechanism very well at all yet. We have genetic risk factors that underpin endometriosis that are being unraveled and can really tell us in a hopefully unbiased way what biological processes are important in terms of etiology. And then we know that there will be risk factor profiles that are likely to vary by endometriosis subtypes. And for that, we need to do, again, larger studies, also at genetic level, and those are ongoing in our follow-up work in the consortium, and those profiles have yet to be identified. And lastly, just a concept that we need to dissect endometriosis as a phenotypic syndrome to identify subtypes with distinct profiles through standardized collection of surgical, clinical, and personal data, combined with that really rich molecular and genetic data that we can collect all together on women with and without endometriosis. So that's where I'll stop, and to thank, importantly, all the women who participate in all the studies that we do together, and a huge number of collaborators that have contributed to this work. Thank you very much.
Thank you for that excellent kick-off to the meeting and for sticking so perfectly to your 30-minute slot. Thank you very much, Karina. And I'd like to personally thank you for your leadership and the effect tools, which have made such a massive difference. I don't see any roving microphones. This is a small and friendly meeting, and I'd encourage you to talk to Professor Zondervan during the breaks. Thank you.
Now we are proceeding with our second speaker. I'll introduce her to you, Jacqueline Maybe. She's the principal investigator at the Maybe Lab at the University of Edinburgh, and she's a senior clinical lecturer in the MRC Center for Reproductive Health at the University of Edinburgh, and she is honorary consultant gynecologist. And her research mainly focuses on the role of hypoxia and inflammation in the breakdown of repair of the womb lining during menstruation, and how these pathways can be targeted with medical treatments to limit.
Here in Istanbul, what a wonderful city. I have been given the title Etiology of Adenomyosis, and I think Karina's done a lovely introduction of what etiology actually means, and I hope she will approve of the tack that I've taken with this. I think adenomyosis is often the poor cousin of endometriosis, and I think it's a wonderful time to be working in this area because there's so much that we have still to do. But what I'm going to try and run through in this talk is some of the work that's been done to date, and then hopefully place things in context. I always like to start with the why. So very quickly, I'm going to go through why we should care about the etiology of adenomyosis, then what it is very quickly, but really focus most of the talk about how it may occur and what may cause it. And I've split this into, like Karina, epidemiological studies that have been done. And then I want to talk through some of the tissue studies that have been done using tissue from women with and without adenomyosis. And I also want to talk a little bit about some of the animal models that we've got, thinking this might be a good way to give us some hints about what may actually be causing it if we're trying to develop adenomyosis in a species that otherwise doesn't have it. And then lastly, at the end, I want to just have a brief slide about where I think we should be focusing our efforts on adenomyosis research. And I'm sure everyone in this room will have their own ideas about that, and it'd be lovely to have some discussions about where we move from here. So start with the why. Why should we care? Well, I think we're all aware that the symptoms of adenomyosis are really life-altering, perhaps not life-threatening, but certainly life-changing for the patients we see in clinic. They present with pain, which is usually around the time of menstruation, so dysmenorrhea, that can be really very significant. They often have heavy menstrual bleeding, which means they can't leave the house for a week out of a month, which has huge impacts for work, school, all of the activities that women are doing, and also can impact on fertility. And we know that in studies of IVF patients, there was a 40% reduction in live birth rate for those with adenomyosis. So this is really quite a significant impact for women. But what we can't forget is that 30% of women who have adenomyosis are functioning like superheroes with no symptoms at all. And I think this is a really important group that we kind of neglect in our research. I think it's important to remember these women so we don't overdiagnose adenomyosis, and also to study these women to see what's different histologically or genetically or any other factors that means that they can function properly where some of their counterparts with adenomyosis are experiencing terrible symptoms. So I think this is a really important group to remember. So what is adenomyosis? Well, I think we're all aware of the BIRD definition where you get migration of the endometrium into the myometrial layer of the uterus that causes a diffuse enlargement of the uterus. And you can see microscopically endometrial stromal cells and epithelial cells embedded within a sort of hypertrophic myometrial layer. And I've got a little slide there just to remind you all of the histological features that our pathology colleagues see not infrequently after hysterectomy. I think the difficulty with adenomyosis is that even histological diagnosis following hysterectomy is really variable. And I'm not sure as clinicians we're sort of as aware of this as we should be. So looking at previous studies where they've given the same sample hysterectomy specimens to different pathologists, there's a variation between 10 and 80% of a diagnosis of adenomyosis. And that's one histological gold standard classification and definition of diagnosis of endometriosis. And I think this comes because there isn't a standardization for histological diagnosis. This junctional zone between the endometrium and the myometrium, actually physiologically, there's a lot of migration and movement in that layer. And there's a bit of disparity between different pathologists about what depth of endometriosis, of adenomyosis into the myometrium actually constitutes a diagnosis, whether it's 2.5 millimeters, 4 millimeters, 8 millimeters. People have different definitions depending on their own personal preference. So even our gold standard diagnosis isn't brilliant. And I think that depth of penetration is variable. But there's been some work to sort of try and classify things and look at the depth of penetration, the number of foci, adenomyosis deposits, the number of those, trying to work out what is normal, what's abnormal, and then the classification of this more diffuse migration versus the focal lesion. There's also been a lot of work with ultrasound and MRI diagnosis, and the MUSA criteria have been really helpful with this. I'm not going to go through this in great detail, but just to say that there are particular features on ultrasound scan that I think now allows us to diagnose this without having to do a hysterectomy. Though I think there's a lot of work to be done to validate these between each other and really standardize things across the globe. So how does it occur? It's the million-dollar question. I think there's been lots of theories proposed in the past. I've split these into four. So the first one being invagination of the endometrium into the myometrium via this altered or absent junctional zone. And I think the junctional zone histologically is quite hard to define. It's probably easier to see using some of our ultrasound and MRI techniques. But it's really that disruption or damage to that zone that allows the endometrium to then migrate into the myometrium. And that theory has been around for a very long time. A slightly different theory is the tissue injury and repair theory, where there's disruption of this junctional zone due to repeated inflammation, damage surgically, or the theory that there's increased contractions in some women that damages that junctional zone somehow and causes that migration through. There's also the stem cell theory, where perhaps some endometrial stem cells are located within the myometrial layer, and it's the development of the deposits within the myometrial layer without any invasion has been proposed in the past. And also the retrograde menstruation theory. Is this actually endometrium that's shed into the pelvis and is migrating from the outside in? So what I want to do is go through those theories, and I'm going to use the abbreviations that you see here: INV for invagination, TIAR for tissue injury and repair, SC for stem cell, and RM for retrograde menstruation throughout the rest of my talk as I sort of work through the studies and which ones may support or refute those theories. So firstly, epidemiological studies. As Karina said, these kind of cross-sectional case control studies can be really helpful, very difficult to carry out in the perfect manner, but people have looked at pathological diagnosis following hysterectomy for benign conditions or chronic conditions, non-cancerous conditions. And I found that women who have subsequently been diagnosed with adenomyosis on those hysterectomy samples tended to have higher parity, and tended to have termination of pregnancy and surgical interventions in their uterus in the past. And these really support the invagination and tissue injury and repair theory. But I think subsequently there have been some imaging studies, and they have looked at a cohort of nulliparous women with regular menstrual cycles, not using hormonal contraception, and about 150 women coming through were scanned, and 30% of them had ultrasound features of adenomyosis. And this kind of flies in the face of this sort of high parity theory, and makes us realize that it's very careful what data you put in the top as to what conclusions you can draw. And perhaps those people going for hysterectomy are a very select population. So that maybe suggests that the stem cell theory might have more credibility in this younger population, as this is developing very early on before they've had lots of children or lots of interventions. There's also been some evidence recently that on MRI scan, these focal deposits on the outer layer of the myometrium of adenomyosis are correlated very closely with deep infiltrating endometriosis. And that lends some support that there may be this outside-in migration, particularly for those cohort of people with the outer layer adenomyosis. And that supports the retrograde menstruation theory. So in conclusion, these epidemiological studies are supporting probably all of the above theories. But one thing that was very consistent in these studies was the very high incidence of comorbidities in the people who are diagnosed with adenomyosis. And I'll come back to that a bit later on when I speak about some of the genetic work that's been done. So it seems that there's lots of comorbidities in the patients experiencing symptoms. So just a little bit about the inside out or outside in, just looking at the different layers of the endometrium. We know that, and some of this criteria has been outlined in the Musa criteria, but the type 1 adenomyosis is where it looks like it's coming from the endometrial layer and migrating into the inner layer of the myometrium through the junctional zone, whereas type 3 is where these adenomyotic lesions are more on the outside layer of the uterus. And there's been some histological work done showing that the type 1 adenomyosis lesions are very similar to endometrium, and the type 3 have sort of more thin epithelial cells and very scanty stroma stained by CD10, suggesting that they're a bit more like endometrial deposits than endometrium per se. So I think there's some evidence that there's a bit of a split here with the invagination theory being more appropriate for the inner lesions and perhaps retrograde menstruation for the outer lesions. So what about tissue studies? There's been a whole host of studies done looking at candidates, comparing the eutopic endometrium, ectopic endometrium, and this work has been brilliant in our understanding, but probably slightly more of the pathogenesis than the cause of adenomyosis. But it seems that the endometrium of women with adenomyosis has a sort of higher estrogenic response, slightly progesterone resistant, probably slightly more inflammatory endometrium, and sort of the apoptosis seems to be different, perhaps predisposing them to have an ability to migrate through any damage to the junctional zone. Within the adenomyosis deposits, there seems to be an increased vasculature, very hypoxic environment with increased factors such as VEGF, probably contributing to the symptom of pain and discomfort within these very inflamed lesions. But really, I think that to try and tease out what the actual cause of that, we probably do need to look more at genetic studies. And there's some work came out from the States in 2019, which I think was really helpful in looking at some of the etiology of adenomyosis. And what they did was they looked firstly at whole tissue from adenomyosis deposits and the eutopic endometrium, and they found that women with adenomyosis had CRAS mutations in both the normal endometrium and in the ectopic and adenomyotic deposits. And these seem to be activating mutations. These mutations are present in normal endometrium, but seem to be increased in women with adenomyosis. And when they looked with laser capture dissection, it was very much within the epithelial cells of the endometrium, had quite a high rate of these mutations. Subsequent work has shown that they may be driving some of the phenotypic differences that we see within the adenomyotic tissue. And in particular, there was hypermethylation of the progesterone receptor gene, and that decreased expression of progesterone receptors, both A and B, within the endometrium, may be driving that progesterone resistance. So that really gives some credibility to this invagination theory, looking that those deposits were very similar to the endometrium in the women with adenomyosis. I think really importantly, within this study, they also saw lots of comorbidity, and within the same women, they sampled the fibroids and saw that CRAS mutations weren't really important there, and the smooth muscle cells was really driven by a different somatic mutation in MED12, which really does say that it's not coming from the myometrium itself, it's really from the endometrium. So that sort of went against that stem cell theory. It seems that fibroids are developing from the myometrial layer, whereas the adenomyosis is coming from the endometrium itself. Interestingly, there was also comorbidity with endometriosis, as we all know, and it also had these CRAS mutations in the epithelial cells, I guess giving more credence to the fact that there may be two variations of adenomyosis, and that the retrograde menstruation theory and that outside-in theory still stands. So I think this was really useful work in trying to tease out some of these differences, and I think gives us probably the best evidence so far that this is coming from the eutopic endometrium migrating into the muscle layer. I think there's been some really lovely work about understanding the sort of physiology of the endometrium in recent years, and the group in Liverpool in the UK have shown us a lot about the glandular architecture within the endometrium, which really gives us this 3D model of the endometrium, which is really useful. So I think previously we thought our glands were these circular structures that we'd seen on H&E over the years. More recently, we had this theory that it was like the bowel, and there were these crypts, and within the base of the crypts there were endometrial stem cells that were regenerating the endometrium. But the group in Liverpool, using sort of a serial sectioning of endometrium and reconstructing it using computer techniques, have really shown that the glands aren't this blind-ending tube, but are really mycelium-like, lots of networks within the basal endometrium. And this complex network probably gives the endometrium that resilience for endometrial repair. And epithelial stem cells are resident within that basal layer. And you can see if it's got all of these structures running along the base of the endometrium, if there's a slight breach to the junctional zone, then perhaps those stem cells can migrate through into the myometrium. And the group in Japan indeed have shown this using similar techniques and computerized it. And you can see in the top right there that one of the epithelial glands is actually communicating from the endometrium down into the myometrial layer. And that really is quite good evidence that adenomyosis is coming from the endometrium and migrating in. So I think that gives a bit more support to the invagination hypothesis. So what about animal models then? I think these are a really interesting component and give us lots of information. I'm not sure they're the perfect model. I think it's always best to study women if you're looking at a disease that affects women. But animal models can be a really useful adjuvant to that. And I'm just going to describe two models that have been developed. More recently, it's been the mechanical thermal induction of injury by the Chinese group. And they take a probe through the vagina and injure one of the uterine horns, either mechanically or they can do it with a sort of an ablation technique. And then they rest the mice for 10 to 12 weeks and then look and they see adenomyotic deposits within the myometrial layer after this tissue injury technique. So they have given some credibility to tissue injury and repair hypothesis using this model, and certainly they're seeing a nice deposit. The authors themselves have said that they're not sure about long-term with this, and that's difficult in the mouse model because they don't cyclically menstruate, but whether the inflammatory system can clear these deposits is still to be determined. They've also done some really nice work looking at pre-treatment prior to the injury and showing that this reduces the amount of adenomyosis using beta blockers, for example. So there's some interesting work there that would be interesting to take further to see if we can apply it in some of our clinical practice. The other model is the postnatal tamoxifen administration, and if mice are given oral tamoxifen, which is a selective estrogen receptor agonist, then they, or modulator, they give the oral tamoxifen day 2 to 5 postnatally. 42 days later, you get 100% adenomyosis, which is a really interesting finding. And really interestingly, in this paper, they looked at the mice at day 6 postnatal and saw real disruption of the development of the myometrium and endometrium and that junctional zone. So I wonder if that sort of gives some support to the importance of the development anatomically of that junctional zone, and whether perhaps there's some stem cells within the myometrium if that's disrupted at that very early stage. So I think it's interesting to run these models alongside our work in humans to really inform us of what the potential etiology could be. So just to sort of tie this all together, I mean, I think we've got these 4 theories, and I don't think we can discount any of them. I think there's probably a bit of a merging of all of them. I think this invagination theory it does stand true, that there's the inside out spread. That's probably related to tissue injury and repair of the junctional zone. And I think there's probably another cohort of people who are having the retrograde menstruation, having their outside in theory stand true. And stem cells, they may reside in the myometrial layer on their own. I think it's probably more likely that the basal layer is where the stem cells are. We've got some good evidence that the epithelial cells, the epithelial stem cells are present. And if there's a disruption of that junctional zone, then they could potentially spread into the myometrium and cause isolated deposits. So I think, although it seems that we haven't perhaps moved on much from within the uterus, outside the uterus, or growth within the uterine wall from the 1921 theory, I think the work that's been done more recently has teased out some of the finer detail of this. And although it probably stands true, we've got a bit of an increased understanding of why that is. So where should we focus our efforts? As I say, I think we're all in the right job. We're not going to be out of a job anytime soon. There's much to do in this space. I'm very keen that prevention is better than cure, and I think looking at the etiology will give us some clues how to prevent adenomyosis happening in our patients. I think we've probably enough evidence now to say that we shouldn't be doing uterine surgery on people unless there's a very good indication for it. I think that's just a sensible practice in general, but I think we've now got support that if we're doing multiple surgical procedures, then there is an increased risk of adenomyosis forming after that. Invariably, we have to do surgery sometimes, so I think we have to look at some of these etiological studies and do some more work in mouse models and some clinical trials perhaps to look at, can we predict who is particularly at risk, and then perhaps amend some of our protocols, perhaps giving some preoperative medication to those who are at high risk of adenomyosis to prevent it developing after we have had to go in and do surgical procedures within the uterus. We really need to standardize and improve the diagnosis of adenomyosis. I'm sure everyone's in agreement with that, and there's some great work being done through the International Federation of Obstetrics and Gynecology, FIGO, looking at some of the standardization. And I think we need to validate some of the imaging protocols with the pathology protocols and work with our colleagues in pathology to really standardize and validate and create global consensus in this area. I think we need to understand the cause of the symptoms in adenomyosis, probably separately to the etiology or merged with it, because I don't think we yet know why some people get abnormal uterine bleeding. What's the actual endometrial defect that causes the heavy menstrual bleeding and adenomyosis? what particularly is causing the pain and the infertility. And I think when we really tease out these mechanisms, then I think that's the only time we can really create more specific, precise treatments for the people sitting in front of us in clinic. And I think this quote from Maya Angelou is really pertinent for adenomyosis. Do the best you can until you know better. And when you know better, do better. And I think we really need to do better for the patients coming to our clinic. So I'd just like to acknowledge everybody who's taken part in our menstrual studies to date, the fantastic team we've got in Edinburgh and wonderful collaborators across the globe, and the people that funded us. And just a short plug for the wonderful city of Edinburgh. If you're ever over, please do give us a shout. And it's a wonderful city to visit and a great department to work in. Thank you very much.
Thanks very much, Jackie. I think in the interest of time, we're going to encourage people to talk during the lunch break, which is obviously oncoming. Next, I'd like to welcome Dr. Douglas Gibson to the stage. Dr. Gibson is a group leader at the Institute of Regeneration and Repair at the University of Edinburgh, and he's the lead for discovery science within our EXPECT group. He's a holder of prestigious Wellcome Trust Henry Dale Fellowship, and within his team, they're focusing on understanding how hormones particularly control tissue function, macrophage function, and how this might impact on women's health. And today, Doug is charged with getting us up to speed on steroid biology and endometriosis, a not inconsiderable task.
Thank you very much for the introduction, and I'd like to echo my thanks to the organisers for the opportunity to speak with you today. It's fantastic to be here in Istanbul, a wonderful and beautiful city. So steroid biology and endometriosis is a rather large topic, as Philip has intimated. But hopefully what I'm going to talk to you today will just kind of go over what we understand and what is informing the influence of steroid biology on our understanding of the etiology and the pathophysiology of endometriosis. So as we know, endometriosis is a hormone-dependent inflammatory disorder, as has been articulated very clearly this morning by Krina. And one of the key drivers within the disease is the presence of estradiol from the ovaries. So we know that estrogens as a class of hormones can stimulate endometrial cells and tissues, and it's thought that the presence of estradiol in endometriosis is able to both drive the growth of lesions and encourage their persistence. It's also important to note that within lesions themselves, there's capacity for what's called intracrine biosynthesis of steroids. So that means that the lesions are making their own hormones. And this localized production of hormones is, of course, associated with stimulation of the lesion, but also in modulating the lesion microenvironment. So that can encourage the development of different inflammatory processes and angiogenic processes which might contribute to disease. So I thought I'd start with what is known in this area, and if you take nothing else from this presentation, this slide should cover it. And that's largely because a lot of what our knowledge has developed around has been reinforced by studies that have continued in the last 5-10 years. And so there's a couple of dogmas that we can actually reinforce by revisiting some of this literature. So as has been mentioned by myself and others this morning, endometriosis is considered an estrogen-dependent disease. We know that progesterone signaling is impaired. This is often called progesterone resistance. And this is important for fertility, but also because progesterone is an important molecular counterbalance to the action of estrogens. As has been mentioned, and I think will be touched upon later today, genetic studies link defects in steroid signaling, both through the metabolism and action of steroids, to the etiology of endometriosis. But what we know from profiling tissues is that it's really related to estrogen receptor signaling and also metabolism in relation to production of estrogens. And this is very clearly characterized by changes in the receptor ratios that result in a predominance of one of the isoforms of the estrogen receptor beta, being predominant over ER alpha within lesions. As mentioned, local production of hormones within the lesions is important. This is characterized by altered aromatase, as well as a subclass of enzymes called 17-beta HSDs, hydroxysteroid dehydrogenases. And as a consequence of this altered receptor expression and metabolism, estrogen-dependent genes are upregulated in both eutopic and ectopic endometrial tissues. And this is associated with activating pathways that promote growth of lesions and impairment of fertility. So because of all these different integrating factors, many of the new therapeutic concepts are focusing on understanding how the action of these local hormones within lesions could be limited or blocked to help develop and improve medical therapy for endometriosis. So it's important to recognize when we talk about estrogen signaling, estrogen receptor signaling is not one linear event, it's an integration of different signaling inputs. And this is just to summarize some of those different elements, not to be overcomplicated, but essentially what it amounts to is that we can have different isoforms of estrogen receptors. These receptors are proteins which are activated when the hormone binds to them, usually estradiol, but also other weaker estrogens such as estrone or estrogenic therapeutics such as tamoxifen. When the receptor protein is bound by or activated by a ligand, it moves to the nucleus to change transcription, which in turn changes protein synthesis and cell function. And this can include variations in receptor isoform expression, as I've mentioned, so variations in alpha and beta isoforms, as well as over on the right here, this relatively recently identified G protein coupled estrogen receptor called GPER, which can have distinct actions in response to estrogens. So in order to understand how estrogens are functioning, we need to go basically back to the most simplest understanding of how estrogens work, and that is in the native tissue itself, so in the endometrium. And we know that estrogen receptors are fundamental to the cyclical function of the endometrium throughout the menstrual cycle. This is characterized by expression of these two key isoforms I've mentioned. Firstly, ER alpha, which is expressed robustly here, you can see in brown by this immunohistochemistry stain, is expressed in epithelial cells and in stromal cells of the endometrium. And during the proliferative phase of the cycle, activation of this receptor promotes cell proliferation and angiogenesis. Similarly, ER beta is expressed within stromal and epithelial cells, but also has been detected in endothelial cells that line blood vessels and inflammatory cells. And as a consequence, ER beta signaling is associated with inflammatory pathways as well as vascular remodeling. And you can already see that these key functional processes that these receptors can mediate have very important roles potentially in the pathophysiology of endometriosis. So when we consider these receptors in the context of lesions, what we find is that a number of studies have shown this. This is going all the way back to 1994. Some of you may remember that. So endometriosis lesions do express ER alpha. So it's not that alpha is missing, and it's linked to the same cell types that are present in lesions. But what we see is that the expression is reduced relative to the utopic endometrium. Similarly, ER beta is expressed in lesions. This time we have the utopic endometrium in the top left, and ovarian lesions in the two right panels, and a superficial lesion in the bottom left. So what hopefully you can see from these immunohistochemistry images is that although you can find ER beta in stromal and epithelial cells, the intensity of the ER beta is different. And that's linked to increased intensity within the epithelial cells within the lesions. So in vitro studies can look at how this can have an impact on cell function. One of the key ways that this can be investigated is by isolating stromal cells from endometrial tissues and from ovarian endometriomas, because there's an abundance of these cells that can be isolated. And what this study by Zhu et al. Has shown is that if they look at the expression of these two isoforms, either by qPCR to look at the mRNA or Western blot to look at the protein, is that the ratio, as I've described already, is altered. So this seems to suggest that there's less ER alpha in endometriotic stromal cells and more ER beta. And this is true for both mRNA and protein. So it's not just that there's a change in the transcriptional availability of these receptors, this actually translates through to the protein that's present within the cells. As I've mentioned, this is a very tractable approach for understanding cell signaling. So more recent work by Gu et al. Has shown that by looking at stromal cells, they can understand how alterations in the estrogen receptor ratios, predominantly by looking at ER beta, can lead to activation of pathways that might be important for the development of endometriosis. So in this instance, estrogens can activate ER beta, which has a downstream impact on NF-kappa beta signaling, leading to the production of CCL2. So CCL2 is an important chemokine for the recruitment of monocytes and macrophages. And what they were able to show in their in vitro assays is that recruitment of monocytes and differentiation of macrophages could happen in response to activation of stromal cells. And reciprocally, the presence of these differentiated macrophages could actually promote proliferation of the endometrial stromal cells. So in this sense, signaling through estrogens where there's a predominance of ER beta creates an inflammatory environment which perpetuates proliferation of stromal cells and hence potentially lesions. So looking at whole tissues, we see the same pattern again. So looking at, in this study by Fujimoto, they looked at the expression of both ER isoforms, this time just with mRNA. And hopefully what you can see on the far right is that the ratio of ER alpha and ER beta is altered as we've already described. So ER beta is greater in the tissues. And this holds true across a number of different studies, so we can be very confident this is a robust observation. I'm not expecting you to read everything in this table, because I certainly can't. But this is just to highlight the fact that particularly this observation is associated with data derived from ovarian endometriosis. So in studies that have looked at other types of lesions, the observation is not as consistently made. In some cases, if for example, peritoneal endometriosis is looked at, ER beta appears to be downregulated. And this speaks to some of the comments that we've had this morning already, where we have to not overly generalize our observations and understand that there's distinct entities within different lesion subtypes. And this is true also of some of the observation and dogmas we might hold to be true in relation to hormones. So as a consequence of this altered estrogen receptor ratio, which is in part driven by alterations in methylation of the ER beta promoter, we see a consequence that means that the presence of the ligand, estradiol, is able to activate ER beta dependent pathways within endometriotic stromal cells. This is associated with very important pathways related to lesion development and progression, such as inhibiting apoptotic pathways, promoting proliferation, and enhancing invasion of endometriotic cells. And the same is true on the right, where we have an understanding that within the whole lesion tissue, rather than just a stromal compartment, there's a difference in the ER beta to alpha ratio, which is associated with upregulation of downstream mediators such as c-Myc and IL-6, which are associated with promotion of inflammation and proliferation of the lesions. So just changing that receptor ratio can have these downstream consequences, which are believed to contribute to the pathophysiology of the disease. As I mentioned, the G protein coupled estrogen receptor is potentially also important in understanding estrogen signaling. And relatively recent work within the last 5 to 10 years has identified that GPER is expressed in the endometrium and in the utopic endometrium in endometriosis. Importantly, the expression of GPER seems to be enhanced. So it's greater in epithelial cells and stromal cells from utopic endometrium compared to healthy endometrium. And notably, when stromal cells are isolated from these tissues and stimulated with a ligand that specifically targets GPER, this is called G1, they could see dose-dependent increases in factors which are very important for, again, lesion formation. So this is VEGF, an angiogenic factor, and MMP, an inflammatory tissue remodeling factor. And importantly, estradiol could mediate these exact same effects in the studies, but they were able to identify that it was specifically through the GPER by using this selective agonist. Importantly, as well as in the utopic endometrium, studies suggest that GPER expression is increased in endometriotic lesions. In this instance, a range of different tissue types were profiled. So this was associated with an increase in ovarian, peritoneal, and deep lesion tissues. And in the same study, they were able to isolate endometriotic epithelial cells and see whether the GPER activity could affect proliferation of those cells. So what they were able to show, looking at the bottom left, is that estradiol could promote proliferation of these cells. However, if they blocked GPER using a selective antagonist called G15, they could get a dose-dependent decrease in cell proliferation. So this is consistent with this relatively new receptor being present in endometriosis and potentially being able to modulate pathways that might be important in disease. So given the complexity of receptor isoform variation, different signaling pathways that are downstream of those receptors, one of the key ways in which to have an effect on estrogen signaling in endometriosis is actually to prevent formation of estrogens. And this is the principal approach for GnRH modulators. So GnRH usually is promoting production of LH and FSH from the pituitary, which stimulates the gonads, in this case the ovary, to produce sex hormones. If we take, for example, the GnRH antagonist elagolix, in this instance, it can inhibit GnRH receptors in the pituitary gland to reduce both circulating gonadotropins and estradiol. And importantly, this has been associated with a reduction in dysmenorrhea and non-menstrual pelvic pain and improved quality of life. So by shutting down systemically the availability of estrogens, all those different pathways that we've just discussed appear to have possibly been suppressed, as evidenced by changes in symptoms within the patients. But it's important to note that this is a very heavy-handed approach to modulating steroid biology in the patient. And of course, shutting down the GnRH axis is associated with hypoestrogenic or menopausal-like effects. These include hot flushes, as well as reduced bone mineral density. So there's perhaps better ways or more nuanced ways to target estrogen signaling in endometriosis, and that's where the local metabolism comes in. So just to very briefly cover this simplified diagram of how this might work. So within a lesion, the parent molecule cholesterol can be converted through a series of enzymatic steps to produce estradiol. And what's important is that enzymes in this pathway have been shown to be dysregulated in endometriosis, resulting in greater estrogen production. The key enzyme in this process is called aromatase, also called CYP19A1, and this is fundamentally required for formation of all estrogens. And this enzyme converts androgen precursors, so androstenedione or testosterone, into estrone or estradiol. Once estrogens are formed, they're shuttled between active and inactive forms. So that's the inactive form estrone, which has weak activity at the receptors, and active estradiol, which has high activity at the receptors. And furthermore, there's an additional mechanism through this enzyme called STS, or steroid sulfatase, which can take sulfated steroids in the circulation, which are inactive, and bring them locally into an environment to activate them within tissues. So understanding the expression and activity of all these enzymes is really key to understanding how local hormone action and steroid biology might be affecting endometriosis. So in this work, led by Andrea Romano's group, they were able to show very clearly that within lesions, within ectopic tissues, activation of estradiol, the active form of estrogen, predominates. So that is consistent with the lesions themselves producing their own estradiol, which of course, as we know, can stimulate all these different pathways that are important for pathogenesis. And this was particularly true of ectopic tissue and not of either normal or eutopic endometrial counterparts. As you probably know, a great number of studies have looked at aromatase expression in endometriosis. This is, again, just to highlight some of the information related to that. And it's important to highlight some caveats that might be associated with these studies. So generally speaking, our knowledge about aromatase expression in lesions is derived from studies looking at immunohistochemistry, which has its own caveats in relation to specificity of antibodies. There's good mRNA data to suggest aromatase is expressed. And some of these studies suggest there's correlation with pain symptoms that might be important for understanding how different levels of estrogens could be affecting different subtypes of disease. And again, as I mentioned in the previous example with estrogen receptors, it's important to recognize that there still is a degree of incongruence between some of these studies. So it's not that every lesion that's tested is expressing aromatase. And this maybe speaks again to this kind of concept of considering more specifically the heterogeneity of different disease presentations. So as a consequence of the fundamental role of aromatase in producing estrogens and this data I've just discussed about evidence for expression and production of estrogens, aromatase inhibitors have been trialed as a potential therapeutic, either alone or in combination. So importantly, what this seems to do, generally speaking, because they've been given in combination with GnRH modulators, the aromatase inhibitors seem to have perhaps a greater risk of having hypoestrogenic effects because you're hitting estrogen production in two different ways. So these are often associated with loss of bone mineral density. It looks like there's potential for aromatase inhibitors to have an impact on pain, but what's not clear is whether they could actually have an impact on recurrence or progression of disease. So looking beyond aromatase, this other enzyme that you might not be as familiar with, steroid sulfatase, is a potentially important mediator. And that expression has been demonstrated both in eutopic tissues and in endometriotic tissues, again shown by immunohistochemistry at the top here in brown. And these studies have shown that both the expression of the enzyme, either by mRNA or protein, but more critically, the actual activity of the enzyme is active within ectopic tissues, and that because of the expression of this enzyme, more estrogen is produced as a consequence of this sulfation pathway. And what's interesting is that this is also appears to be associated with severity of disease. So STS activity and the severity of disease is correlated in a way that aromatase doesn't appear to be. So in this study by Atal Purohit, they looked first at aromatase activity, which I've mentioned is present, but they didn't see any difference between the simple binary separation of minimal, mild, or moderate to severe. But when they looked at the STS, they could see a significant elevation in STS activity associated with moderate to severe disease. So again, this suggests that inhibition of this enzyme, particularly in the lesion environment, might be pertinent as a therapeutic approach, particularly in moderate to severe disease. So a number of different STS inhibitors have been developed, one of which is E2-mate, which shows that it has been shown to decrease STS activity in uterine tissues and in lesion tissues. So that's highly promising. What was interesting in this study was that they looked at potentially how that could affect human tissues. So my colleague Yanis will explain some animal models later on today. But in this instance, they explanted human tissues into a mouse model to see how the impact of that treatment could have on the growth of those tissues. And they found that the STS inhibitor decreased the growth of those human tissues. So that the human tissues are producing their own estrogens through STS, and when that was inhibited, their growth was decreased. However, they didn't find an impact on proliferation or apoptosis rates. So this suggests that this could be a particularly useful approach for limiting estrogen production within lesions, but so far, to the best of my knowledge, no clinical trial has been conducted testing STS inhibitors for this indication. So this leads me to the 17-beta-HSD isozymes. So particularly, we're going to talk about type 1 and type 2 here. So type 1 is important for activation, and type 2 is important for inactivation of estradiol. So looking at the expression of this type 1 enzyme, again, it's been detected and reported in both the eutopic tissue and in the lesions. In a study from Andrea Romano's group, they looked at the activity of this enzyme in healthy tissue, eutopic tissue, and ectopic tissue. Interestingly, a lot of these studies that are focusing on other enzymes don't really report many changes in aromatase, and I think that's worth keeping in mind in the context of the different studies we're talking about. But what they could see was evidence for changes in the activity of 17-beta-HSD enzymes in association with lesion tissues. So this was characterized by, over on the left, bottom left, an increase in type 1 activity, so activation of E2 from E1, a decrease in type 2 activity, so decreased conversion of E2 back into the inactive E1 form. So overall, that's a net effect of having greater active estrogens within lesion tissues. And again, this observation of more type 1 over type 2 17-beta-HSDs has been replicated in a number of different studies, largely by looking at the expression of the enzymes. But I think this previous study looking at the activity is the one that gives us the most robust information. So just again to highlight that this is generally what's observed. In this instance, this seems to be probably a more robust observation because it's been recapitulated across many different lesion types, so peritoneal, ovarian, and deep disease. So as a consequence, you'd consider that inhibiting this enzyme might be important for reducing estrogen synthesis in lesions. And that's what's been done in a study recently by Poirier et al. And they were able to test two different 17-beta-HSD1 inhibitors, where they were able to reduce that activity by around 70%. They took forward this candidate molecule PBRM on the left into a baboon model of endometriosis. And what they were able to show in that system was that co-administration of this 17-beta-HSD1 inhibitor reduced the number of lesions formed in the baboon model. Interestingly, in this context, co-administration of letrozole as an alternative treatment, so that's an aromatase inhibitor, also had a similar effect. So this is a robust way of reinforcing the fact that local estrogen production, either via aromatase or 17-beta-HSD1, certainly in this baboon model, is critical for growth and persistence of lesions. One of the advantages of taking that system out, of course, is that they were able to longitudinally follow these animals with the intervention. So collectively, the evidence mounts and points towards local production of estrogens, which can have a stimulating effect on lesions. So as is shown here in the schematic, a number of different key enzymes are increased or overexpressed within lesions, such as aromatase, STS, and 17-beta-HSD1. Unfortunately, most of the therapeutics that have been trialed for targeting some of these key enzymes so far haven't yielded a consistent therapeutic option. But interestingly, this target 17-beta-HSD1 is currently under development. And so the state of play with that, originally developed by Ferendo Pharma, then taken over by Organon. So this compound is now called OG6219. So it's gone through, on the bottom two sections there, phase 1 trials, and it's currently undergoing a phase 2 dose-finding trial, which is recruiting currently and due to complete at the end of this year. So we should know more about the possibility of targeting this enzyme as a potential therapeutic. And as I mentioned, based on the expression study, this one seems to be expressed across different lesion types, so that might hold a greater capacity for targeting, in a broad sense, therapeutically the estrogen production. How are we doing for time? Are we okay? Yeah. So beyond estrogens, it's also important to recognize that steroids are a diverse range of different compounds. As I mentioned, androgens are natural precursors to estrogens. And it's important to recognize that as well as having local elevation of estrogens within lesions, there's also evidence that this is the case for other hormones. So principally, this is a study from Matti Putinen's group, and they've looked specifically at the concentration of hormones within lesion tissues, and they've measured this using a very accurate and specific technique known as LC-MS. And what they're able to show is that within the lesion tissues, there's actually an elevation of the androgen testosterone. Interestingly, when they followed up, and I encourage you to go digging in supplementary figures of this paper, because this is where I got this, they could show that androgen receptor, shown here in brown by immunohistochemistry, was expressed abundantly within the lesion tissues. So AR was found in peritoneal, ovarian, and in deep disease. And then when they did molecular profiling to look at potential downstream targets of androgen receptors, they could see a very clear evidence for enhanced or elevated gene expression of factors that are known to be regulated by androgen receptors. So this suggests that androgens could be active within lesions, and that could be contributing to some of the changes that take place within the lesion microenvironment. So it would be remiss to not mention pregnancy, progesterone, because it's very important for pregnancy and producing remodeling within the endometrium in preparation for that through a process of decidulization. As I've also mentioned, and is shown over here on the right, progesterone is an important molecular brake for estrogen-dependent proliferation of the epithelium. So progesterone is basically fundamental to the function of the endometrium, but we know that the signaling capacity in endometriotic tissues and also in utopic tissues is diminished. And this is characterized by something that's termed progesterone resistance. And this really nice study shown in the schematic on the left highlights just the extent of the problem. So if we take cells from these two environments, so healthy endometrium or endometriotic cells, and co-incubate them with the exact same treatment side by side, what we see is an absence of activation of pathways that are associated with acquiring a decidual phenotype, so promoting decidualization. So this happens quite freely in the normal cells. You see upregulation of key factors such as connexin 43, 17-beta-HSD2, and these gene changes just aren't happening in the endometriotic cells. And this is due to a number of different reasons. So the receptors themselves, as shown on the table on the right, are decreased, as well as factors associated with their signaling are decreased. And there's evidence for increased microRNAs, which actually silence progesterone signals, being elevated within endometrial tissues. So as well as receptor signaling, receptor availability being reduced, picking up on this, again, the study from Matti Putinen's group that I mentioned previously, what they've actually shown is that the abundance of progesterone within the tissues within both the eutopic tissues and in lesions is vastly reduced. So as well as not having the appropriate signaling machinery, the ligand that can activate those receptors that may still be present is vastly reduced in comparison to normal tissues. So that suggests that the progesterone availability and signaling is going to be impaired, and that's indeed what is observed in patients. So taking that all together, again, hopefully this is just reinforcing things that we already knew. So endometriosis is an estrogen-dependent disease. There's good evidence for altered receptor expression, altered expression of synthesis enzymes, and importantly, activity of those enzymes, as well as changes in their downstream targets. We know that changes in steroid biology may be distinct between lesion subtypes. And I think based on reviewing this evidence for this presentation, I think we really need to reappraise how those differences might be important in contributing to heterogeneity within disease. What we can say with confidence is that there's a greater predominance of ER beta over alpha, driven by changes in the expression of these isoforms and methylation of the ER beta promoter. We know that the pathways that are important for lesion formation are upregulated as a consequence of this, and these include cell proliferation and inflammation, as well as suppression of apoptotic pathways. As I've mentioned very briefly at the end there, there's new evidence that androgen signaling might be important in lesions, and this may be a relevant future target. And as is well characterized, progesterone signaling is impaired, which has important impacts on fertility, but will be difficult to actually target due to the multiple deficits in that signaling pathway, as I've described. So both the receptor expression, downstream signaling, and ligand availability within tissue microenvironments. So hopefully the new therapeutic concepts that I've discussed that are targeting 17-beta-HSD1 should have a degree of efficacy, but we're waiting on the outcome of those trials, which, as I've mentioned, are due at the end of this year. But taken together, I think we can be confident in saying that estrogen signaling is a key driver of endometriosis. Alterations in receptor and enzyme activity perpetuate this difference, and that potentially therapeutic targeting of that could be a future direction that could bring novel therapeutics. So I'd like to finish there, and thank you for your attention.
Thanks, Doug, for giving us a great overview of a very complicated area. Thank you very much. Now we have an opportunity for one of the very important short presentations.
Yes, now we will announce the best Bedside paper, which was elected for oral presentation on the topic identification of candidate genes involved in adenomyosis pathogenesis, whole exome sequence analysis of a pathologically confirmed adenomyosis case, and it will be presented by Sevcan Aydın. Sevcan Aydın is doing her PhD studies in İstanbul University as the Sancar Experimental Research Institute in the Department of Genetics, and she also got her master's degree on genetics at the same faculty, and her research mainly focuses on endo and adenomyosis and whole exome sequencing.
Thank you for information. Hello, everyone. I am very excited because I will talk in front of remarkable scientists about my desired topic, genetic infrastructure of adenomyosis. And I will talk about part of my master thesis. And to begin with, adenomyosis is a uterine benign condition characterized by the ectopic localization. Ectopic localization of endometrial-like tissue in myometrium. And adenomyosis and endometriosis can be considered closely related disease, and because they share common mechanisms and in fact, gene and gene family and symptoms, and bringing up the hypothesis of common pathogenesis. And therefore, we aim both to identify rare variants in genes previously associated with endometriosis and novel candidate variants which may be involved the adenomyosis pathogenesis. And then we utilized whole exome sequences on a case diagnosed with adenomyosis only via pathologically confirmed. I will talk about a step-by-step material methodology. First of all, a woman with a family history, 2 affected siblings and 1 daughter of adenomyosis underwent a hysterectomy due to treatment-resistant abnormal uterus blending, and then genomic DNA was isolated from peripheral blood sample and was subjected to whole exome sequences analysis on Illumina NextSeq platform, and utilizing bioinformatics analysis and including aligned and data analysis on Genome is a NextSeq platform. Genome is a NextSeq platform is a open access tool, and then finally, detection of the candidate gene parallel filtering approach, and is most crucial for exome analysis, genetic detection of the candidate gene. And I will talk about next slide, detailed information for detection of candidate gene novel and rare variants. And detailed information for the detection of candidate rare and novel gene. First of all enter-west data that is included in target region data and on-target region data. And first of all, off-target region data and low qualified data was excluded. And high qualified genetic variants was analyzed two parallel filtering approach. And first of all, for detection of rare candidate gene, and I'm sorry, just a second. I'm sorry, just a second. Let's continue. Yes. First filtering approach, panel gene for detection of rare candidate gene, and we complied panel gene obtained from GWAS, previously known GWAS data, and genome-wide association study, and NGS and functional studies for both adenomyosis and its closely related condition, endometriosis. For the detection of rare variants, high qualified variants was analysis according to minor allele frequency less than 1% and in-house allele frequency. And second approach for detection of the novel candidate gene, and according to minor allele frequency less than 1%, in-house allele frequency, and then pathogenicity was analyzed. Pathogenetic criteria is most important criteria for WAS analysis because pathogenicity scores is the most crucial. And we determined was determined utilizing ACMG guideline and ClinVar along with a ClinVar database associated with disease and related gene. And then finally, rare or novel candidate gene associated with adenomyosis and endometriosis. And looking at the results and clinician results, first of all, pathological evaluation confirmed adenomyosis while excluding endometriosis in this case. And then we obtained from 2,000 and 44,000 genetic variants of target and in-target region, respectively, and high qualified variants 40,000 and low qualified variant and off-target variant excluded, and obtained from GWAS data a number of the genetic variants, approximately 1,000, and selected from the detection of the novel gene data a number of the one pathogenic variants and two likely pathogenic variants and 40,000 and uncertain significant variants. And to sum up, 6 rare variants and 6 novel variants, a candidate gene associated with adenomyosis and endometriosis. And you can see the 6 rare variants we analyze in this diagram. Candidate rare gene obtained from previously known GWAS data. As you can see, this These variants, including 2 missense variants and rest of synonym variants, rest of gene and synonym variants. And you can see the 6 genetic variants we analyze in table. These three gene function play a role, microtubule function, may be considered associated with infertility, and then this gene play a role, transcription factor associated with infertility, and then gene related to ubiquitination to notch signal receptors, and final gene involved in axon guidance and cell migration, and novel gene, as you can see. And to conclude, we identify a total of 12 genetic variants that may be involved in adenomyosis pathogenesis. Our results demonstrated that adenomyosis and endometriosis are inextricably linked to each other. These conditions are intertwined disease because shared common mechanisms, and such as TR theory and metaplasia theory, and gene family as a result. And in our case, stranded hypothesis of common pathogenesis share among 2 conditions. And absolutely all results, when I replicated independent cohort, will be contribute to other underlying mechanisms of the condition. And with this, I would like to acknowledge, and our team separate in 2 groups, clinician groups and genetics groups. And first of all, always supported me regardless of any obstacle, my advisor, Dr. Feyza Nur Tuncer, and head of the clinician team, Professor Dr. Engin Oral, whatever happened, always supported our team. And members of the clinician team, Dr. Fitnat Topbaş and Pinar Yalçın Bahat, follow up the patient and always supported our team. And we thank the patient, member of the taken in part in or the study. And finally, I would like to thank you all for your information, for attention. Thank you.
So thank you for your lovely talk. So congratulations on some lovely work. So I just want to close this first session, thank the speakers for their excellent contributions, and then we'll hand over to the new chair, and we look forward to the keynote lecture about genomics. Thank you.
We will welcome Feyza Nur Tuncer Kılınç as a moderator for the keynote lecture, and Nilüfer Rahmioğlu to do her speech on genomics of endometriosis and adenomyosis.
Thank you. My name is Feyza Tuncer, and I'm pleased to welcome Dr. Nilüfer Rahmioğlu for the keynote lecture. Dr. Rahmioğlu is a senior research scientist at Wellcome Center for Human Genetics and Endometriosis Care Group at University of Oxford. She is the principal investigator of COHERE, which is Cyprus Women's Health Research Initiative. She's as well the lead analyst of the latest International Endometriosis Genomics Consortium, Genome-Wide Association Analysis. And with great pleasure, I would like to give the floor to Dr. Rahmioğlu.
Thank you very much, Feyza. It's a great pleasure to be in Istanbul here today and talk to you about genetics of endometriosis and adenomyosis. I'd like to start by giving you some definitions around what genetic variation is, because I think it's quite important to understand these concepts to be able to place, like the previous talk, we heard more about the rare genetic variants versus what GWAS is doing, so that we have common language. So basically, we are born with differences in our DNA that result in differences between individuals. These differences in the DNA sequence are called polymorphisms when they're common, and mutations when they're rare. And there are different types of genetic variation, the most common ones being single nucleotide polymorphisms, which are base pair changes along the genome that are most commonly also studied. In terms of the genetic architecture of disease, you can categorize disease into a Mendelian rare type disease, as well as a common complex disease. And what I'm illustrating to you in this figure is two spectrums where you have the frequency of the investigated genetic variants on the x-axis, and on the y-axis is the effect, the penetrance of those genetic variants. And what I'd like you to notice is the Mendelian rare disease sit on the top right corner, where we have single rare variants that cause disease. So these variants carry very high risk. And these are primarily defined in family studies. Some examples of these diseases include cystic fibrosis, sickle cell anemia, as well as Huntington disease. And usually family-based studies are conducted to identify these variants. Common complex disease, they sit on the right bottom corner there. And there are many common genetic variants, each conferring small increases in risk that cause disease. And these are common disease, so you sporadically also see them within the population. And examples include type 2 diabetes, different types of cancers, cardiovascular disease, and endometriosis and adenomyosis sit within this group. To identify the heritable common genetic basis of complex disease, we do these studies called genome-wide association studies, which I'll talk a bit more about later in the talk. Now, my talk is going to be mainly centered around endometriosis, as adenomyosis genetics is severely under-investigated. But I'd like to start with adenomyosis to give you an overview of what's out there. Usually for common complex disease, we start by estimating the genetic variability that contributes to disease risk, which we term heritability. And for adenomyosis, we don't have an estimate for the amount of common genetic variants that contribute to disease risk yet. There are some various small-sized candidate gene association studies that shows involvement of some genes, but they're not really robust evidence. And very nicely presented by Sevcan, there are family-based studies that are trying to identify these rare variants that might be associated with familial forms of adenomyosis. And there are some more recent efforts where we're trying to identify the common genetic variant basis for this condition. So why are we interested in identifying these genetic variants that are causal for common complex conditions? Well, first of all, this helps us to understand the underlying disease mechanisms for these conditions. It gives us more insights whether we're looking at one disease or whether different subtypes of conditions, which in turn will lead to identification of more targeted treatment approaches, as well as identification of more personalized diagnostic biomarkers. So as I mentioned, genome-wide association studies, in short I'll call them GWAS, are very powerful study designs that allow us to detect and identify these variants. And very basically, what we do in a GWAS is we genotype a case group and a control group, and then we test the differences in allele frequencies of single nucleotide polymorphisms between these two groups. And I'd like to share some of Fitnat's DPhil work with you here today. So she's conducted the first GWAS study for adenomyosis in the UK Biobank, which is a huge resource that includes 500,000 females on whom we have medical record data, as well as genotype data. And within that data set, there are 804 adenomyosis cases with surgical history. So she's conducted a GWAS comparing these 804 adenomyosis cases versus more than 100,000 female controls without adenomyosis. And here you can see a Manhattan plot that illustrates an overview of those results. So what you have in here is on the x-axis is the genomic location, and on the y-axis is the association significance. So what you see with the red line in the top is genomic significance. So you would expect the peaks, the dots, to pass that threshold to identify genomic significant robust signals associated with the condition of interest. And there is also a nominal threshold there denoted with a blue line. Those are nominal associations that require basically replication or validation. And as you can see here, there are multiple peaks that we are identifying for adenomyosis. However, they need to be replicated in larger data sets, and Fitnat is currently working on running GWAS in FinGen and all of us data, and hopefully meta-analyzing that and identifying some robust genetic signals for adenomyosis, which we hope will help us understand the underlying biology. And with that, I'm going to switch gears to talk about mainly endometriosis genetics. As very nicely described by Krina, endometriosis has a heritable basis, which is estimated to be around 50% from family studies. And of this genetic proportion, 27% is estimated to be due to common genetic variants. And when we looked at the different stages of disease, it was seen that stage 3 and 4 disease carries a greater genetic loading compared to stage 1 and 2 disease, hinting at genetic structure and differences between these potential subtypes of the condition. I'm not going to talk about etiology, as you heard about it in a lot of detail in the morning. But endometriosis is a very heterogeneous condition with multiple symptoms and varied representations in the pelvic cavity. And what I would like to bring to your attention again is the lack of correlation between the symptoms reported by the patients and the extent of the disease in the pelvis. And moreover, the fact that we don't understand whether it is a progressive disease or whether we're looking at potential different subtypes of this condition. So utilizing genetics, we hope to be able to identify or answer some of these questions. The data that I'm going to present to you today comes from the International Endometriosis Genomics Consortium, which is a global effort aiming to identify the genes associated with endometriosis utilizing GWAS. And this work has been published last year in 2023, so you could look up the citation if you're interested in further details. Here are the centers that contributed to the current analysis that I'm going to show you today. We have 24 studies, which includes more than 60,000 endometriosis cases. And then, sorry, my slide is not showing properly. There we are. So we conducted the first GWAS, including everyone who had endometriosis diagnosis, which is the most powerful analysis we've conducted. But also, we've run 3 additional GWAS analyses, including subset of studies that included more detailed surgical data. So stage 1 and 2 disease versus controls, stage 3 and 4 disease versus controls, and endometriosis cases who also reported infertility versus controls. Here are the results for the overall endometriosis GWAS. So as you've seen for adenomyosis, this is an overview. And what you can see with endometriosis, we are identifying 42 genome-wide significant signals that are robustly associated with endometriosis. In the interest of time, I will deep dive into one of these loci to illustrate the complexity around these signals. So this particular one is sitting on chromosome 6, And as very nicely presented by Doug, this signal is actually sitting around the estrogen receptor 1 gene. And when we run further analyses trying to find map and identify causal variants for this locus, we actually ended up identifying five independent signals. So indeed, it is a very complex region that require further analysis to identify the causal variants. Moving on to the subtype-based GWAS results. What you see here is, again, a similar presentation. It's just a circular Manhattan plot. So in blue is the overall endometriosis, but inside you have the subphenotype results. So in green is stage 3 and 4 versus controls. In orange is stage 1 and 2 disease versus controls. And in yellow is infertile endometriosis versus controls. And what you'll notice is that we identify significant associations with stage 3 and 4 disease, but nothing with stage 1 and 2 disease, and only a single locus for infertility-associated endometriosis. There is obviously an element of statistical power here, given that the sample sizes are much smaller, but we are identifying differential genetic basis, primarily for the stage of disease. Here is a closer look at the effect sizes of the 42 endometriosis loci between stage 1 and 2 disease versus stage 3 and 4 disease. This plot compares the effect sizes in the form of odds ratios for the 42 loci. So each dot on there is a SNP that is associated with endometriosis, and effect size for stage 1 and 2 is on x-axis, and effect size for stage 3 and 4 is on y-axis. And what you'll notice is that the effect sizes consistently across have larger effects in stage 3 and 4 disease compared to stage 1 and 2. Then we looked into characterizing these identified loci further using the deep phenotypic data we had from some of the studies. And the subphenotypes that we investigated were some surgical subphenotypes, so having an endometrioma, having superficial lesions, having deep lesions, and also some pain subphenotypes, primarily categorized in the top 5 pain symptoms that patients represent. First, on the surgical subtypes, we looked whether any of the surgical features drive these stronger associations we have observed with stage 3 and 4 disease. And indeed, in this plot, you can see the effect sizes for the 42 risk variants show stronger correlations with having an endometrioma. This is an assuring finding, as endometriomas are typically present in stage 3 and 4 disease. Furthermore, we observed moderate correlation with having deep lesions and very weak, almost no correlation with having superficial lesions. These results point that ovarian endometriosis seem to have a different genetic architecture compared to the other surgical representations of the disease. Moving on to the pain subtypes. Again, we looked at the correlation, so it's the similar plot, but now you have more of a heat map. And overall, we see very weak correlation between stage of disease and the pain severity across the 5 different pains and endometriosis. However, the strongest correlation we observed is between stage 1 and 2 disease and dysmenorrhea, and consistently stage 1 and 2 disease showed slightly stronger correlations with pain symptomology compared to stage 3 and 4 disease. But again, it's important to note that these are tiny sample sizes, so we only have hundreds of cases in these analyses. So what we really need is much larger data sets where we can run de novo GWAS analyses for particular pain symptomology that are reported by endometriosis patients. Now, moving on from subtypes and identification of genetic variants, what we're really interested in is, once we identify these genetic variants, is to understand actually which genes they are regulating, to understand a bit more about the biology of the condition. So one way of doing this is looking at QTL maps, which illustrates which SNPs regulate expression or methylation levels of various genes. Now, there are these QTL maps in large-scale databases, but most of these databases don't include endometrium. So within the consortium, in the Endocare team, as well as in our Australian collaborators team, we have generated the first endometrium eQTL map, and we utilize this as part of this functional annotation of the identified genetic variants. We've also used blood to capture any systemic differences, and the blood QTL maps are much larger in size, so there's more power to identify relevant genes if they have systemic effects. And then using these QTL maps and the SNPs that are associated with endometriosis, we tested whether these SNPs have a causal association with endometriosis through the eQTL route. And when we've done that, we identified within endometrium 4 target genes that are being regulated by endometriosis risk variants, and many more in blood. But as I said, this is mainly an issue of power. So as we increase the sample size for endometrium expression profiles and methylation profiles, we'll be able to detect some more tissue-specific genes. And again, in the interest of time, I would like to illustrate some of those loci for you. So the first one is sitting on chromosome 15. And we have evidence from both eQTL endometrium data as well as mQTL blood data that show us SRP14 and BMF genes as the relevant genes for this locus. And there is also evidence showing that the SRP14 SNP has chromatin interactions with the BMF locus. If you look at the biology, this variant has been found to be strongly correlated with a variant regulating a steroid hormone that makes estrogen and testosterone. And this particular steroid hormone activates neurotrophins such as nerve growth factor and brain-derived neurotrophic factor, both of which are involved in maintenance of chronic pain in various chronic disorders. Furthermore, we've seen that this particular SNP shows a stronger association in individuals that suffer from more than one type of pelvic pain. So it's the first time from GWAS data we're actually identifying some genes that are relevant for the pain mechanisms involved in endometriosis. Another locus that I'd like to highlight that's also involved in pain mechanisms is GDAP1. This locus sits on chromosome 8, and again, from both eQTL and mQTL data, from blood, we identify this gene. And this gene has been shown to be associated with dysmenorrhea severity in various previous studies, so a very interesting candidate to follow further. Now, moving from locus-specific analyses to more genetic correlations at a genome-wide level, we know from epidemiological evidence that endometriosis patients also suffer from various comorbidities. And we tried to capture these in 4 main categories. So we have immune inflammatory traits and conditions, pain symptoms and conditions, other reproductive traits, as well as obesity-related traits. And what I'm showing you here is the genetic correlation between endometriosis and these conditions that fall into these categories. What you have on the x-axis is different disease. But what I would like you to notice is those ones with a red star are the ones that show significant genetic correlation with endometriosis. And when you look at all the pain conditions and symptoms, they are significantly genetically correlated with endometriosis. And I would like to expand upon that a bit more. So the stronger, and you can see here the different pain conditions and symptoms that show this genetic correlation. What we wanted to investigate here is the strong genetic correlation for the first time shows that there could be a genetic basis to the sensitization of the nervous system that we observe in endometriosis patients. And what we wanted to do is try and understand this genetic sharing further by identifying shared genetic variants. So what we've done is this multi-trait association analysis between migraine and multi-site chronic pain and endometriosis. The reason why we chose migraine and multi-site chronic pain is the fact that they had very sizable GWAS data. And indeed, when we run these, we identified many more novel variants that are shared. So between endometriosis and multi-site chronic pain, we ended up with 6 more novel variants, 2 more with migraine, and actually 4 more that were shared across the 3 conditions. So these results definitely provide support for the hypothesis that presence of endometriosis can result in pain through interlinked activation of hormonal, immune, as well as neuronal pathways. And with that, I'd like to give you some conclusions. So first on adenomyosis. So the preliminary GWAS results seems to suggest that there is different genetic basis for adenomyosis compared to endometriosis. Larger GWAS meta-analysis are underway to have more robust data on adenomyosis that will allow us to look at the sharing between endometriosis and adenomyosis, but also differences, why we see in certain patients the comorbidity and why we don't see in others. And in terms of endometriosis, what I've shown you here, the 42 genetic variants, explain around 5% of the genetic variability. So there is actually a lot more that we are missing. And there are continued works to increase sample size within the consortium and achieving also detailed phenotypic data that will allow us to look at the subtypes and identify the remaining genetic variability underlying endometriosis. There are no high-risk variants for endometriosis, and single common genetic variants don't have any predictive value in clinic. However, polygenic risk scores are slowly making their ways into clinic for other common complex conditions. So a polygenic risk score is basically summing across the risk from all the identified common genetic variants and using that as a score to inform clinical decision-making. It's early days for endometriosis, given the limited amount of genetic variation we explain. But once we have more knowledge around it, these might be utilized in the clinic, but we're not there yet. So in terms of more what we can do is what our results are showing is that there are definitely genetic subtypes of endometriosis. In particular, this data is illustrating ovarian versus peritoneal disease having different genetic basis. And we are starting to reveal and understand the differing pain symptoms and their genetic basis within endometriosis. We've also identified many new treatment targets. So you can see a list of genes that are potential treatment targets for endometriosis and their subtypes from here that needs to be moved on to more translational and functional work. And the shared genetic basis with other pain and inflammatory conditions really opens the venue to potentially think about repurposing existing drugs for these other conditions for endometriosis. And with that, I would like to firstly thank all the participants that take part in the endometriosis and adenomyosis studies, as well as the consortium members and everyone else who we collaborate with endometriosis and adenomyosis work, as well as funding bodies. Thank you for your attention.
Thank you for this highly informative talk and great timing. I wanted to take questions actually, but we are behind the schedule, so I think we could have an interactive lunch. And thank you so much. We'll be moving with the second session on clinical pain, and I'd like to invite the moderators for this session, Drs. Koray Elten and Dr. Turgut Var, please. Thank you.
Good morning, my colleagues. I and my friend Turgut will chair this session, and which will be mainly on pain. The first presentation is how to establish a pain clinic. Professor Hibner would present it lively. However, since he has another meeting which is conflicting with this meeting, he sent his presentation. Professor Hibner is from Creighton University, USA. He is the founder of the Pelvic Pain Special Interest Group of AAGL, and also elected to be the next Secretary Treasurer of AAGL. So we would like to hear his presentation, please, the operation team.
I'm Michael Hibner from the Arizona Center for Chronic Pelvic Pain. I apologize for not being with you in Istanbul today. I was recently elected to be a Secretary Treasurer of AAGL, and at the time you're watching this video, I'm traveling to my first board meeting in this role.
Sesi baştan alabilir miyiz sesi?
After my fellowship at the Mayo Clinic, I decided to dedicate my
Uzayacaksa diğer konuşmaya mı geçsek acaba? Teknik ekipten bir fikir verebilirler mi?
I'm Michael Hibner from the Arizona Center for Chronic Pelvic Pain. I apologize for not being with you in Istanbul today. I was recently elected to be a Secretary Treasurer of AAGL, and at the time you're watching this video, I'm traveling to my first board meeting in this role. By training, I am a urogynecologist, but immediately after my fellowship at the Mayo Clinic, I decided to dedicate my professional career to patients with chronic pelvic pain. I created a very successful pelvic pain center in Scottsdale, Arizona, and today I'm going to share my experiences on how to do just that. I mostly regret not being able to be in Turkey today because I have very close ties to your beautiful country. In 2016, I organized a pelvic pain course in Arizona, attended by many friends from Turkey. And the following year, I was invited to give a talk in beautiful Istanbul at one of the medical conferences you were organizing. And I was able to reconnect with great friends. So again, I wish I could be there today. My talk today is on how to create a pelvic pain center. But of course, there is no science behind that. So I can't use any double-blind randomized study to talk about it. But I thought the best starting point is to talk about what chronic pelvic pain is. So chronic pelvic pain, as defined by the International Pelvic Pain Society, is a non-menstrual pain of 6 months or more that localizes anatomically to the pelvis, and it's severe enough to cause functional disability and require medical treatment. So as you see, what's important in this definition, that this is a non-menstrual pain. Using this definition, almost 15% of women in the United States at some point during their lifetime will experience chronic pelvic pain. It is more common than diseases that are well known, such as coronary artery disease, lower back pain, diabetes, and many others. But because of our inability or lack of knowledge on how to treat those patients, almost 61% of them have no correct diagnosis, and 90% of them are unable to have a pleasurable intercourse. This also causes a huge economical burden to the society. It is very important to realize that almost 70% of cases of chronic pelvic pain are non-gynecological. Even within gynecological causes of pain, endometriosis is just a small part of it. I, for example, see a lot of patients with pelvic congestion or venous insufficiency leading to chronic pelvic pain. Neurological diseases are also common, especially nerve entrapment. This is what my practice really specializes in. Bladder pain syndrome often coexists with other pain conditions. But by far, one of the most common reasons for chronic pelvic pain is the musculoskeletal system, specifically spasms of the pelvic floor and other surrounding muscles. And to make things more complicated, most of the patients with chronic pelvic pain have more than one reason for pain. This is often seen in patients with endometriosis, where you do the best surgical resection possible, yet those patients still continue to be in pain. That is often because additional reasons for pain were missed at the initial assessment. Fred Howard have found that 78% of women with endometriosis and chronic pelvic pain have another additional reason to have pain. This is a slide I made for medical students years ago. Later on, I used that slide in the chapter I wrote for the endometriosis book by Professor Oral. The title of that chapter is Endometriosis, the Evil Quadruplets. Endometriosis very often coexists with other conditions such as interstitial cystitis, painful bladder syndrome, irritable bowel syndrome, and spastic pelvic floor. I still remember in my fellowship training, we had patients who even after the most complete resection of endometriosis still had pain. Now I know it was due to one of those conditions. I personally believe that the single most common reason for chronic pelvic pain in women, which is often missed by physicians, is the painful spasm of the pelvic floor muscles. And one of the reasons we often miss pelvic floor muscle spasm is because we are not trained how to examine those muscles. In patients with endometriosis, we often get preoccupied with nodularity of the uterosacral ligaments or thickening of the rectovaginal septum, but we fail to examine patients with one finger to palpate the muscles. This exam needs to be done methodically by palpating with one hand and one finger the superficial, intermediate, and deep muscles of the pelvis. This happens in a mechanism which I described in Engin's book, which is called the viscerosomatic and visceral convergence. In viscerosomatic convergence, a painful impulse in the visceral organ, like endometriosis implant, travels through the afferent sensory fiber to the spinal cord, where it reconnects. It leaves the spinal cord through the motor efferent fiber to the surrounding muscle, causing this muscle to spasm. This spasm of the muscle is palpable by us, but it's also very painful. So it creates this positive feedback loop because that again travels through the afferent fiber to the spinal cord. And then it also travels through the other fiber, through the hypothalamus, all the way through the central nervous system. In Another common group of conditions often causing pelvic pain is compression of the nerves in the pelvis. This is what I really specialize in: compression of the pudendal, obturator, ilioinguinal, genitofemoral, and posterior cutaneous nerve of the thigh. So in my pelvic pain center, when I see patients with chronic pelvic pain, I ask different questions than probably most of endometriosis doctors. Of course, how did the pain start? If the pain started with some kind of an injury, it is less likely to be endometriosis. Where is the pain? What is the nature of the pain? How often does the pain occur? Is it cyclical or not cyclical? What makes the pain worse? If the pain was worse with physical activity, again, it's most likely not endometriosis. And what makes pain better? So if patients say that rest makes pain better or using a heating mat makes pain better, that's most likely the muscle pain. If patient says emptying the bladder makes the pain better, well, that's the bladder pain. And of course, what has been done so far. So is there pain with sexual intercourse or gynecological exam? Most of the patients with chronic pelvic pain will have pain with the sexual intercourse or gynecological exam. But what is very typical of the pelvic floor muscle spasm, but also sometimes seen in pelvic congestion syndrome, that there is pain after sexual intercourse or after gynecological exam. If patients have pain with sexual intercourse, it is important to ask in what position, because patients with bladder pain and sometimes pelvic muscle pain usually have pain in what is called the doggy position. And also, it is important to differentiate the pain with intercourse from pain with arousal or orgasm, because patients with pelvic floor issues or patients with pelvic nerve issues will often have pain with just arousal without any penetration, and they generally have pain with orgasm. Is the pain better in the morning or in the evening? Remember, endometriosis does not choose the time of day, but pelvic floor muscle spasm, the nerve pain, the bladder pain does. Usually patients with pelvic floor muscle spasm wake up in the morning with minimal pain, and so do patients with pudendal nerve injury. And then the pain worsens as the day goes by, and usually that pain is the worst by 5 p.m. or the time before they need to go to bed. And again, this is not endometriosis pain. Does pain wake you up at night? Usually the muscle pain or the nerve pain does not wake you up at night, but endometriosis pain can. Is the pain worse with sitting or standing? And again, endometriosis does not choose the position of the body, but the muscles do. The nerves do. So patients with pudendal nerve injury, patients with pelvic floor muscle spasm will hurt more when they're, and patients with pelvic congestion syndrome will hurt more when they are standing. And does the pain happen when the patient is turning from side to side at night when they're sleeping? Usually I see that as a sign of adhesions in the abdominal or pelvic cavity. Is there pain with full bladder? The pain with full bladder is typical of interstitial cystitis. Or is there pain at the end of urination, which is very much consistent with the spasm of the pelvic floor muscles? How many times does the patient get up to go to the bathroom at night? One time a night is normal, and everything other than that is not. And when patients go to the bathroom more than once, this also may be a sign of the pelvic floor muscle spasm, because patient is not emptying their bladder completely, therefore they don't start their night with an empty bladder. And also, probably the most important question is, can the patient start the urine flow immediately? Women should be able to start the urine flow immediately without any hesitancy, just like men sometimes do. Hesitancy in women is definitely a sign of the spasm of the pelvic floor muscles, and not the sign of endometriosis or anything else. On the exam, in addition to the gynecological exam, it's important to examine back muscles. Remember that there are 45 muscles that attach to the pelvis externally. So those patients with pelvic floor muscle spasm will usually have lower back pain, but I also have pain in the anterior and the posterior thigh. On the abdomen, you look for the incisions, hernias, check for the Cornet sign that I'll talk about in a minute, and of course do the pelvic exam where you examine pelvic floor muscles the way I described previously, and then perform your regular gynecological exam. For this very lengthy introduction, I left myself a few minutes to talk about how to create a pelvic pain center. So the most important thing is the paradigm change, meaning change how you think about pelvic pain. If you treat pelvic pain and you only treat endometriosis, you will fail to relieve pain in 70 to 85% of patients. And ask yourself a few questions. Do you want to treat endometriosis or pelvic pain? Do you want to practice gynecology or just pelvic pain? Do you still want to practice obstetrics? And are you going to see male patients? So once you decide to practice pelvic pain and to create what you're going to call a pelvic pain center and dedicate the rest of your professional career to that, the first step is to find a pelvic floor physical therapist, probably the single most important person in your practice. And if you're going to practice the nerve injury pain, then you will need an interventional radiologist. I do most of my own nerve blocks myself, but some of the nerve blocks require the use of a CT scanner, and for that you're going to need an interventional radiologist. You're going to need to find an education to learn about pelvic pain. You have not learned about pelvic pain in medical school, residency, or fellowship, and I know that very well because I taught fellowship and residency, and I still teach in medical school. You will also need to find the support staff. Your chronic pelvic pain patients may be very difficult, demanding, and overall unhappy. So you need the staff that will handle them with dignity and respect, yet be firm, and the staff that really knows what you're doing and that knows what those patients are going through. And lastly, in the later stages of your practice, you have to find the correct location. That generally comes later, but if you become very successful seeing pelvic pain patients, those patients will come from far away. So your office has to be centrally located, it has to be close to the airport, it has to be easily accessible, and it has to look right. For those patients that have been suffering for a long time, when they come to your office, it has to give them the confidence that they're in the right place. In the United States, most of the pelvic floor physical therapists are trained by Herman and Wallace Institute. This is a good starting point to find a trained pelvic physical therapist. This is also a place where I refer my patients if they are looking for a therapist in their area. I received most of my pelvic pain knowledge, at least initially, from the International Pelvic Pain Society. At the beginning, I was going to all of their meetings and all of their courses. This is a very good starting point. Another option to get a great education in pelvic pain is the Pelvic Pain Special Interest Group of AAGL. It is actually a special group that I founded. This group organizes amazing, great pelvic pain courses, and I would really encourage you to look for one. I graduated from my urogynecology fellowship in 2003, and almost immediately in 2004, I decided to dedicate my professional career to chronic pelvic pain. In 2005, I traveled to France where I got additional education in pudendal neuralgia and pudendal nerve injury. And when I returned back to the United States, I started building this pelvic pain center at St. Joseph's Hospital in Phoenix, which is also a medical school. I also started an AAGL gynecological surgery fellowship. And in a green shirt to my right on this picture, you see Dr. Nida Desai, who's my second fellow, who later on became my partner. And two of us started building this division, adding more partners, more fellows, and all the support staff. So this is an early picture from the creation of the Pelvic Pain Center here in Phoenix. So this picture is taken 10 years later in approximately the same location in my office at St. Joseph's Hospital. And as you see, the group is much bigger. This group shows my fellows and my partners. And at that time, we were a very, very well internationally recognized pelvic pain center. But COVID happened and many other things happened, and in 2020 I left, and most of my partners left, and they moved out of Arizona. I stayed because of many reasons, including the family reasons, and I decided to open my own private practice, solo practice pelvic pain center, which I call the Arizona Center for Chronic Pelvic Pain. In addition to all the other things I told you, which are important in creating a practice or pelvic pain center, I think the visual identity is very, very important. In fact, right now I'm wearing a shirt that has the same logo here. In 2003, my dad, who's an engineer, created this logo for me that I've been using while working for the university. They actually allowed me to have the additional logo in addition to the university. And when I left, a graphic artist recreated that logo, and this is what I currently use. But I think it is very important for the patients to have this uniform labeling and visual identity. So I really recommend hiring a graphic artist to have a beautiful, stunning logo that can be used on your website and all the other media. And finally, when I opened my practice in 2020, I made sure that the office looks right, that is very welcoming to patients. And this is a photograph taken at the office opening, so I had a grand opening. And you actually see from the left my father-in-law, then my dad. As you see, we're all wearing the shirts carrying that same logo. My mother-in-law that is handing me the plant, which is also the same color, matching the logo. And then on the right, in the white shirt, also with the logo, is my wife, Alex. In the back, you see the map, which at that time we just started, so there were not many spots, but right now it's covered with the locations from almost everywhere around the world. It is equally important to have a social media presence, so Instagram, Facebook, YouTube. You have to have a killer website. You have to make sure that you are up high on your Google rankings and on patient boards. So I have a professional company that does my Instagram and Facebook. I also opened a YouTube channel called Questions That Need Answers with Dr. Michael Hibner, which is quite popular with patients. I have a great website that was professionally designed by a website designer, and it's kept up, meaning every new change is made to that website. I have a company that pays attention to my Google rankings. So, for example, if you Google pelvic mesh injuries or pudendal neuralgia, that I come up high on those Google rankings. And patient boards is something that patients run themselves. You have no influence over that. But if you treat patients right, which you should do anyway, then patients will give you very good ratings on their own patient boards, and that will generate more patients. So this is my new group. I have two surgical schedulers that are standing right next to me, and on the far right side on the slide is Drew, my practice manager. The three people I really couldn't do it without. I have worked with them for about 18 years in my previous academic institutions, and when I left, they came with me. Who is not on that slide is my physical therapist, because she wasn't present on the day that we had a photographer in the office. But you see Tiffany here, my amazing pelvic floor physical therapist. We're sitting on the floor with a very grateful patient who, for several years, because of pudendal neuralgia, was not able to sit on hard surfaces. And here, after the treatment, he's able to sit on the hardwood floor. all the new patient referrals in my office, whether they come from the website, the patient discussion boards, or emails from the patients, or directly from physical therapists or physicians, come to one person, my practice manager, Drew. Drew is very, very knowledgeable. I have worked with her for about 18 years right now. So endometriosis pelvic floor, or the group that is others, which is endometriosis and all the other pain conditions. Most of the patients in my practice, especially those from far away, are seen through telehealth. If I think that they have pelvic floor issues or endometriosis, during their visit to Arizona, they're first seen by my physical therapist before they're seen by me. And those that I do not think have endometriosis pelvic floor issues, but conditions like endometriosis, are seen by me directly. These days, I see about 10 new patients a week, and half of them are men and half of them are women. Over 80% of my patients are from out of state, meaning outside of Arizona. Therefore, most of the patients that I see, I initially see through telehealth. Among female patients, I see three separate groups in equal parts: women with pelvic floor and pudendal neuralgia, women with mesh injuries, and women with endometriosis or other conditions. Among male patients, they are 100% patients with pudendal neuralgia and pelvic floor injury. So that really brings me to the events of last week. Because my practice is growing very rapidly, I needed more space. So I was able to purchase the building, and after construction, I'll be able to move there later this summer. And finally, I'll have the pelvic pain center I always dreamt of. So this is the blueprint for the building that I created myself, and I hope this can serve as a template on how to set up a pelvic pain center. So in my new building, I'm going to have a very large operating procedure room. So hopefully most of my surgical procedures will be able to be moved into this area. There's also going to be a recovery area where patients, two patients at the time, will hopefully be able to spend the night if needed. There's an area that is going to be designed for the CT scanner. So at some point, I am planning on purchasing a CT scanner and moving the CT procedure, CT guided blocks, and other procedures into the area in the office. There is going to be a large pelvic floor physical therapy area. This is the area in which the therapist will be doing the internal exams, which is a more private area. But then there is an area here. This will be for the outside exercises with some exercise machines that physical therapists are going to be using. And we are going to have 3 exam rooms. There is probably no need for more than that because the appointments are long and patients are scheduled for 1 hour each, no patient. So 3 exam rooms for 1 provider like me is more than enough. And the rest of the area here are the offices, the kitchen, the scheduling area, storage, etc. So I hope, again, that this plan can serve as a blueprint on how to build and set up a pelvic pain center. Again, thank you very much for this opportunity and allowing me to talk at your conference. And again, I very much regret not being there in person, and I hope to see you soon.
Yes, thanks to Michael Ibner. The next speaker is Christian Becker. He is the professor of reproductive science at Nuffield Department of Women's and Reproductive Health, University of Oxford, UK. co-director, Oxford Endometriosis Care Center, consultant gynecologist and subspecialist in reproductive medicine. It's a part of the children and young adult tissue preservation team, helping patients with cancer and some benign disease to preserve their fertility. Yes, the topic is endometriosis-related pain, medical management. Stage is yours.
Thank you very much. Hope you can hear me. Thank you very much for the invitation and the introduction. It feels a little bit like coming home. As you probably can hear, I'm from Germany, and I grew up in the small Turkish city of Berlin. So it feels very much known to me. So I was asked to speak about endometriosis-associated pain and the medical management. You hear more about the surgical management in a while. So I had the pleasure to be involved in the recent ESHRE guidelines on endometriosis. I'll show you the evidence-based decisions that we've made. These are my disclosures, just to let you know that none of the funds went to me. They all went to the University of Oxford. So as we've heard a few times today, endometriosis is a heterogeneous condition, and we still use this probably a bit old-fashioned surgical approach where we subdivide it into superficial disease, endometrioma, and deep disease. And as you've heard earlier, there's probably no genetic background for that properly. There are probably more biological reasons why we should probably more subdivide it. Now, because we're still quite behind compared to other diseases with this classification, we're probably also not as good as we potentially could be with our medical and probably also with the surgical management. So if we think about how to treat our patients, we should ask them, first of all, why you're here. And I actively do that often. Ask them, is your main issue the pain, or is the main issue trying to conceive? And that's obviously sometimes a mean question, because sometimes they want to do both. But if pain is the main reason, then we have a few more options available. So this is a graphic from a systematic review that Katie Vincent and I were involved in about 10 years ago now. And when I looked at this for this talk, it was quite interesting that a lot of the things we came up with back then are still true today, and probably have been, a lot of them have been confirmed over the last 10 years. But what I just want to show you is that pain is a complex situation, as you heard just now, and that we probably need much more diverse approaches, both medically and surgically, how to treat our patients. The first question is, well, do we need to treat the lesions or the symptoms, or possibly both? If I'm asked by companies, you know, what do we need to do? Well, it's often dictated by the regulators. They often want to see that the lesions are also treated, which is, I'm not sure whether that's always necessary, but that's how the regulators, at least at the moment, still often look at it. So these are, this is the algorithm from the ESHRE guideline that was published in 2022. You may have seen this. We have 3 options that we should discuss with the patient, and really should discuss with them. There's the option of analgesia, there's the option of hormone treatment, and obviously surgical treatment as well. So let's focus on the analgesia. So I think we all give our patients some form of analgesia, but if we look at the evidence that's behind it, you'll see in a minute there's not very much. However, we came up with, as you can see, a weak recommendation that women may be offered, so it's also quite weak wording, may be offered non-steroidal anti-inflammatory drugs or other analgesics, either in combination with either by itself or in combination with other drugs, to reduce endometriosis-associated pain. Now, this is based on a single study that was published in 1985, I believe, by Kalpela and Rönneberg, where they had 20 patients included. So it's a few patients only for a lot of patients that we have. It was at least a randomized controlled double-blind study where patients were put on naproxen or placebo for 2 periods and then crossed over to the alternative treatment. And what they did see that about 83% of patients who were on naproxen had a complete or substantial pain relief, but the placebo group also had some of this pain relief or improvement, which is probably the reason, if I go back to this, that it was not really statistically significant, because you see the confidence interval going through 1. Now, we do give the patients analgesia, but as I said, the data is not really there. So we have to probably expand, and that's what we did when we went through the guideline. We have to expand a little bit. So, for example, there are pretty good data for primary dysmenorrhea. Now, you would argue, obviously, primary dysmenorrhea is defined as dysmenorrhea without any known cause. I would counter-argue, well, with endometriosis, where we're struggling to identify the disease, there's probably quite a few women with primary dysmenorrhea who still have endometriosis in them, and that may cause the dysmenorrhea. So for primary dysmenorrhea, at least, there are pretty good data to suggest that non-steroidal anti-inflammatory drugs help with the pain. I've just shown here a few because the Cochrane review actually lists quite a few. Here you see for diclofenac and naproxen again, and overall there's probably some support that it's helpful. So how about hormone treatment? Again, coming from the primary dysmenorrhea side, this is Cochrane review looking at the combined oral contraceptives compared to placebo or no treatment, and they see that giving the pill seems to be of benefit for, again, primary dysmenorrhea patients. And it's irrelevant what type of pill you give, so first, second, or third, or fourth generation. So it seems to be working all right. Now, if we look at data for endometriosis patients, and I'm not going to go through all of this because of time as well, we have seen that both for oral, so oral contraceptive pills, but also vaginal rings and transdermal contraceptives, there seems to be an improvement in the endometriosis-associated dyspareunia, dysmenorrhea, and non-menstrual pelvic pain as well. Then we had a lot of discussion, should we give the pill, for example, or the others, continuously or in a cyclical fashion? Especially in those patients who have dysmenorrhea, there might be a benefit in giving it continuously, but obviously, obviously there is there's the risk of breakthrough bleeding in these patients. This is based on some data, although as you can see here, this is primarily looking from Mutinal, probably looking at recurrence of dysmenorrhea, and also on the left side, which is not significant for endometrioma. There are other studies that suggested that cyclicity is probably worse than continuous use in those patients. How about progestogens? Again, we recommend that women should be, and that's quite strongly recommended, that we should recommend progestogens to patients with endometriosis-associated pain. There was no question that we had asked before, but we felt in the guideline development group quite strongly that we need to tell the patients about the side effect profile of the pills, because we have to manage expectations. So we should tell the patients before, be aware that if you start on this medication, there is a possibility that you may have side effect X and Y, effect on mental health, breast tenderness, skin, etc. And so if they don't get on with a particular drug, tell them, satisfied, this may just be that particular progestogen, try another one. Because otherwise they come back and say, the pill is not for me, do surgery. So we really should manage the expectations from the beginning. The other thing we have to discuss with them is that when we give them contraceptives, they do have certain risks associated with this. And this is from a relatively large cohort of patients where it's shown that, as we know, endometriosis, giving the pill increases the risk of breast cancer over 10 years by about 39%. The other thing that we also should discuss with them is that the risk of developing ovarian cancer seems to be reduced in these patients as well. So it is very important that we have this interaction with the patient and not just tell them, this is what you have to do. I think those times are hopefully over. How about comparing different medications? And there are not that many comparative studies out there. Here's one that is looked at in another Cochrane review, comparing a GnRH agonist, glycerin, with a combined contraceptive pill, and you see there does not seem to be any difference in the effect on dysmenorrhea after 6 months treatment. So how about, what's the general recommendation? It is recommended to offer women hormone treatment as one of the options to reduce endometriosis-associated pain. And as I said before, it should be a shared decision-making with the patient, that she fully understands the pros and the cons, so she can make an informed decision when she decides what she wants to have. So the final few, or the final slides I would like to use on GnRH agonist and antagonist. You've seen on my conflict of interest, I've been involved in a few studies with the antagonist, and they are coming on the market now. We've all used GnRH agonists for a long time. As we all know, GnRH agonists are given usually as subcutaneous injections. I mean, nasal sprays are available as well, but injections. And what do we do when we start? We give a pulse, and therefore you have a flare-up effect, which those who do IVF know about very well. But that also can increase estrogen levels and possibly increase the pain as well. And only after maybe 2 or 3 weeks, eventually you have a downregulation of the receptors and FSH, and then estradiol goes down. Whereas with the GnRH antagonist, because you block the receptors straight away, you don't have that flare-up effect, and therefore it kicks in relatively quickly. Also, you can titrate slightly the dose, whereas that's not possible with the GnRH agonists. I think what's important to understand with the agonists and the antagonists is the estrogen threshold theory, which Bob Barbieri published in the 1990s. So with our medications, we really lower the estrogen levels significantly, and that's why patients get all the menopausal symptoms. For endometriosis, we know, or there are some data to suggest, that there's a certain amount of estrogen that is necessary to activate the disease. And with those drugs, we usually go way below that. So we need to find that sweet spot, so the therapeutic window. And that's why we normally give a GnRH agonist or antagonist with and some add-back HRT to raise the estrogen levels just enough to alleviate the menopausal symptoms, but not activate the disease. Now, with antagonists, some of the companies, they suggest GnRH antagonists by themselves, and then people can use their GnRH, they can use their HRT themselves, and there's some benefit to that. Some companies suggest GnRH antagonist already in a single pill with the HRT together, and we can discuss later on what the pros and cons of this are. These are the data from the 3 drugs that are currently out there. So Elagolix, which was the first one that was published, and that's mostly available on the US market, I think in the 200 milligram twice daily dose orally. Again, you could argue there's a certain amount of inconvenience there because you have to take it twice a day, but it was the first one out there. Then the Relugolix, which is sold as a combination therapy. As I told you before, this is the medication where HRT is already in the pill at a specific dose of the Relugolix. I think it's 40 milligrams. And you can see, similar to what you've seen before with the Elagolix, that you have a very quick reduction in dysmenorrhea after about 8 to 12 weeks. And then finally, Linzagolix as well seems to show exactly the same thing, relatively quick onset of help with dysmenorrhea, but also with non-menstrual pelvic pain, which the others also have shown. So from a ESHRE perspective, which obviously was published, 2 years ago now, so the data is a little bit older that we've used, and there have been a few more studies out there now. We were very cautious with the GnRH antagonist. I think if we had to do the update now, which we actually will have to do, there's more data now to support the GnRH antagonist. They are more practical because they can be given orally. There have not been, however, any comparative studies, so we do not know which one's better or not. From a logistics perspective, it probably is an antagonist because patients don't have to go to their gynecologist or GP every 4 weeks for the injection, which should make it also less expensive as well. Finally, aromatase inhibitors. We rarely use them in the UK, at least in Oxford, but I think in the UK. There are some data to suggest they may be of benefit. The data is by far not as strong as for the GnRH agonists or the pills, but it is still an option, especially if the other medications haven't worked very well. There's a study, an old study here, where they compared agonists, GnRH agonist with anastrozole, with the GnRH agonist alone, and there seemed to be some benefit. There are newer studies now that also suggest that aromatase inhibitors seem to be working quite well. Here, for example, letrozole with norepinephrine, 5 milligrams, seems to be reducing the size of endometriomas. Again, small studies, so you have to be a bit careful with the evidence here. We had one patient years ago who had, as you can see, a very large endometrioma connected to the aorta there, where surgically you really didn't want to jump in, and she had been on GnRH agonists before, so it was decided to put her on a aromatase inhibitor, and you can see over time how the endometrioma shrunk. So that's an N=1, so again, be very careful, but it is something in the arsenal. We recommend it as a second or third line treatment, usually because of the side effect profile and because the lack of good studies so far. So usually we would suggest the contraceptives or progestins as a first line treatment, GnRH agonist or maybe now antagonist as a second line treatment, and aromatase second and third line treatment. One thing to remember, and I'm almost done, is that no matter what we do, as I said earlier, the way we look at endometriosis is probably away from perfect. What we do medically, we do see that there's still a lot of patients who are struggling with pain during and after treatment as well. So that's not uncommon. And there are also still patients who either have no reduction of pain symptoms at all, or as I said, there's some pain still at the end of treatment. Now, whether that's because of our misdiagnosis of potential endometriosis or chronic pelvic pain and no endometriosis being involved is a possibility, obviously. But it's also well possible that the treatment options that we have are not specific enough for the disease that we're looking at. As a kind of lift over to the surgical talk that you hear in a minute, there's one study that compared surgery with medical treatment. This is from Paolo Vercellini's group from quite a few years ago now again, where they used northisterone, I think 5 milligrams once daily, which is what they usually use in this group, and compared to surgery. And as you can see on the left-hand side, these were patients who had deep disease, so rectovaginal endometriosis, and on the right-hand side, those who didn't, so more superficial disease, you could argue. And if you look at the pain scores, so the surgical group for the deep disease on the left-hand side, you can see they had high pain scores initially, 93, 91, etc., and then it dropped quite nicely after surgery, but also in the progestin groups, in the northisterone groups. It took a bit longer, but if you look at the data after 12 months, there was no significant difference anymore in the between the surgical group and the medical group. And for the more superficial disease, actually, it looked like it was slightly better the other way around after 12 months. So keep that in mind when you think about, should we do surgery on this patient, should we do medical treatment in a patient. Here are the data from that study, which you can look up. And at the very end, should we use hormone treatment as an adjunct to our surgery? There's no data to suggest that we should give hormones prior to surgery to improve our surgical treatment, but there are some data to suggest that if you are having surgery, think about, and patients not trying to conceive, obviously, think about putting them on hormones, for example, putting the coil in, if Marina coil, if she's open to that, or think about any other hormones afterwards, because there are now some data to suggest that it might improve the outcome of surgery, so reduction in pain, but also probably reduces the risk of recurrence of certain forms of endometriosis. So again, we have this algorithm, hormones, analgesia, and surgery, which you'll hear about in a minute. The data I showed you today was from the NICE guidelines, which I was allowed to share. We actually have to update this now. Andrew Horn is sitting in the audience here. He'll be delighted to hear that. Now, these are the people who were in the core group. We had, and I'll show you some data about this tomorrow. We had subgroups, so there are many more people involved in this. But I'm very grateful that the guideline came out, and I think it was quite well received. And I thank you very much for your listening. The guideline is available as a very long document with all the details, but there's also a patient version available now, I think in English, in Dutch, and I'm not sure which other language. So we're trying to make it more available for everybody. The patient version is only 28 pages or so. It's very good. I always recommend it to my patients. So if you have someone who understands English well enough, I would recommend it. Thank you very much.
Thank you, Professor Becker. We will have the questions at the end of the session if we have time. The next presentation is about surgical treatment. So the presenter is our former president, Professor Usta. He is trained in advanced laparoscopy and endometriosis surgery in selected centers in Europe, and also a scientific committee member of the AAGL. Yes, Professor Usta, the stage is yours.
Dear Chairman, dear colleague, thank you so much. So I'm very happy to talk with this topic with this very valuable and eminent faculty. When you look at the literature, they have a lot of publications, and so we are very happy to see in the Istanbul that colleague. So why we treat endometriosis after the Becker? My task is a little bit getting easier. Normally we can treat that one with a medical treatments, but in some conditions, maybe you can give some medical treatments, but there is no answer. Or is it work? I'm so sorry. Yeah, but maybe you can treat it in another way, but there's no answers, pain, or you can face some organ dysfunctions, recurrence, or cancer suspicions. In that conditions, we can choose the surgical way. So in gynecology, normally we commonly ask in the routine practice, do you have a pain or pelvic pain? But the question is very simple, but the details are very important. Michael gave us some details about the pain characteristic. When we look at the details of the pain, if the lady say that I have a dyspareunia, I have a dyschezia, in that conditions, we know that the posterior area can affect about the endometriosis. And it is not surprised that in that cases, we can see some dysuria or bladder symptoms because of the hypogastric nerve, which is effective on the posterior part. These are simple, all of us know about these symptoms, but in some conditions, we have to keep in mind that one, very rare, but very important. If you guess that that lady have an atonic bladder during the pain attack, maybe you can diagnose a sacral nerve endometriosis. That paper published 12 years ago by Chaperon Group from France. They said that if the patient have endometrioma, at the same time, we know that the pain condition, they can have a deep endometriosis. It is increase the level of the pain of that cases. This paper coming last year, this is coming from the Brazil, and Marcio Abroa said that if the lesion going to the backside of the pelvic compartments, we know that that is related with the dysmenorrhea and the dyschezia, and it is not surprise for us again because it is related with the very severe symptoms and the dyspareunia also. And they say that if you find a higher number of the lesions, then it is correlated with the pain again. This is a very old definition about the deep endometriosis. You know that everyone in the whole, I guess that know this definition, 5 millimeter criteria, but we know that 3 years ago, 4 big society come together and make a definition again. Their horn is here, and so they said that there's a definition about the deep endometriosis, but they didn't use any exact number of the diameter of definition. The main problem about the endometriosis, we say that endometriosis, but what kind of endometriosis? Nilüfer mentioned in the morning session, she said that adenomyosis is a different origin about the gene characteristic, but we call that one, this is endometrioma, chocolate cyst. This is bladder endometriosis. Some of us think that this is superficial endometriosis, but I am not agree with some of them because in some condition, yes, but in some condition, we know that this is a very deep lesion. This is adenomyosis. It makes a very complicated situation in the clinical practice, and these are the bowel endometriosis form, and that is the other point, extragenital endometriosis. When we say that this is endometriosis, yes, but that endometriotic tissue make all around the abdomen make a disease, and they can create a pain. And so we call this one, if this is endometriosis, this is not a wrong definition. Which kind of endometriosis, which level of endometriosis affect that lady's life? And so maybe you can give GnRH antagonist or you can apply the surgery, but what kind of patient waiting for you in the OR or in the office setting? This is our roadmap about surgical treatment modality. Normally, the last 30 years, we increased the knowledge about the anatomy and the surgical technique and the endoscopic treatments of endometriosis. It is not very old information when you look at the years of the treatments, only 3 decades. Last year, we lost David Redwine, so he has a very good definition about the surgical treatments of endometriosis. I guess that some of us doesn't agree with him, but in that time, he said the surgical treatments of endometriosis should aim removing all visible areas of endometriosis with margins free from disease, as the case in oncological surgeries. This can bring us some bill. That bill contain the complications. And when we talk about the complications in endometriosis, major complications related with the death conditions. So it is very important to make a good balance about recurrence, feeling, the discomfort, and then the complication rate of that cases. How can we approach that cases with a surgical way? We create the normal anatomical conditions. We want to take the all affected area, and this is the main aim of the surgical treatments of the endometriosis. We can discuss about the cure. We know that in some conditions we apply the surgery, then there's no endometriotic new lesions. In that conditions, can we talk about cure, or can we accept that endometriosis doesn't curable disease? When you talk about this is not curable disease, this is a chronic disease, it is not easy to talk about with that one with the patients. So informed consent and at the same time informed patient is very important during the decision of the treatments. Normally it can be related with the standard follow-up with the medical treatments, or it is related with the surgical way. If you apply the surgical treatments of endometriosis, but after the surgery, when the lady have a stoma and there is no good information before the surgery, you can face a very complicated situation in that conditions. When you look at the ASRM in 2020, they summarized the surgical treatments of endometriosis. Very simple. You can excise or ablate the lesions. Maybe you can apply we can talk LUNA, uterosacral neuroablations. In some conditions, presacral neurectomy, or in some selected cases, radical surgery for that cases. This Cochrane review published 2010, when we look at that paper, laparoscopic surgery results improved pain outcomes when compared to diagnostic laparoscopy alone. When we pass the 10 years, this is another Cochrane review again about surgery, pain, and endometriosis. We found that it is uncertain whether laparoscopic surgery improves overall pain. And they have some comments about uncertain whether laparoscopic excision is more effective than ablation. So the previous one, we have to apply the surgery, yes, but when we pass the 10 years, there is some doubt on the treatment choice about the surgical treatments. That study coming from the Aarhus, our friends published that one. It's a prospective study. They said that yes, endometriosis surgery have a very good working treatment model. This is a prospective one, and we can decrease the pain, and at the same time we can increase the quality of life of that cases. It was published 2019, George Condus and Matthew Leonardi, Canada and Australian article. This is a meta-analysis. They said that yes, it is very well worked about pain conditions. And there is some paper following that one. The message is the same. They are working very well when we talk about pain with the surgical treatments of the clinical way. And so at the end of that way, we know that the preoperative pain level after the surgery going down, in some cases, it is a little bit continuously, but it is not too much, and it is not very badly affect their quality of life. And some of them is following that papers. It is not easy to talk about some conditions. Dyspareunia, it is a very good paper underlying the conditions, the sound of silence, giving voice to endometriosis-related positional dyspareunia. When we talk about dyspareunia in endometriosis, we always underline that conditions. In endometriosis, we know that deep dyspareunia is the problem of that case. Non-dyspareunia may be a little bit common issue in the women, but in endometriosis, because of the deep endometriosis or obliterated cul-de-sac, deep dyspnea is a very common condition in clinical way. And Michael mentioned about that one. We have to ask that one. Is there any problem during the sex? Is there any problem in some positions, like this article's title? And we know that if we apply the surgical treatments, we can decrease the pain during the sexual intercourse, and we can increase the quality of life in that ladies. So when we look at the last ASHA guideline, and Christian Becker is here and the other writers are here, they say that it is recommended to offer surgery as one of the options to reduce endometriosis. This is not an obligation. This is not only one way. This is a method, and you can choose this one. And they say that the guidelines say that when surgery is performed, clinician may consider excision instead of ablation of endometriosis. Again, this is using the same definition about the treatment modality in endometriosis. Is there any subgroup who is more benefited than the other one? Yes. If patient have a severe disease, we know that after the surgery, they have a stronger pain relief after the surgery. And so it is not very surprise for us. One of the very difficult point is the bowel endometriosis. It is dependent on the locations. On the rectal part, we know that it is very higher risky surgery. We know that in a very good experience center, 10% of the complication rate have to talk with the patient. So we have to approach multidisciplinary way. And so on the upper part in sigmoid region, the complication risk is going down, but we have to work with the colorectal surgeon in that condition. Superficial endo, there is no good knowledge about how can we do when we see the superficial endo. If you are an endometriosis surgeon, you think that this is my enemy and I have to fight that lesions. I have to cut everything, but we need some balance. But if you want to answer that one, we need some very good publications. But when you look at the literature, there is no good information about that one. This is nerve interruption about pelvic pain during the surgery, but there is no very good new information about the nerve interruption procedures, LUNA or presacral nerve resection. When we look at the ECOG practice bulletin, they do not recommend the nerve interruption procedures, LUNA or presacral neurectomy. This was a publication 3 or 4 years ago. It's a meta-analysis about presacral neurectomy. They have some comments on it. These studies are very old, and because of that old studies, we cannot say that you have to do that one. So we have a very good new study. We need that one to say we have to do or we do not do that one. When we turn back again the ESHA guideline about endometrioma, clinicians should perform cystectomy instead of drainage and coagulations. But this is not very easy. You can face a very young woman. The ovarian reserve is very low. You cannot say that the guidelines say you can apply cystectomy. Yes, it is a very good part. This information, we have to pay attention to the rest of the patient's characteristic. The other conditions is the superficial endometriosis again. I want to ask you, do you think that this is superficial? If you think this is superficial, could you raise your hand in the hall? I guess that 3%. So I'm not agree with you. So when you open this one, you can, yes, see some color change on the peritoneal tissue, but when you follow it, now we find the hypogastric nerve, and that lesion's very thick, and it is make a contact on the nerve. When you send that one, always the histopathological examination was endometriosis. There is a very good study, and the authors are here. I guess that they collected data last month, and we will see the results. And if they have some knowledge about some preliminary reports, we will be very happy about that study because it is very important. The question is very simple. They look at the cases. This is a multi-center study. They collected, I guess, that 400 cases in a multi-center way. They only look at the peritoneal endometriosis. This is not peritoneal endometriosis. And if we apply the excision, it is working or not. It is a very simple, but it is very important question to decide that procedure during the surgical technique. This abstract was presented in American College of Obstetrics Gynecology Congress in 2022. They applied total peritoneectomy. There is no problem. They routinely applied the peritoneectomy, and then they said that the pain level is going down and the quality of life is going up. But we cannot say that we see one abstract and then all of us have to apply that one. But it is an interesting point. This is a simple peritoneal layer, and they said that we find 70% of the case have some superficial endometriosis on the peritoneal layer. I start with the anterior compartments of the deep endometriosis. This is a bladder endometriosis. This is an isolated case. These are some samples about the bladder endometriosis. It is not so difficult to diagnose that one. You need some time to diagnose it, but the main problem is these are very complex cases. If you see some simple bladder endometriosis, it is so easy. If it is close to ureter or if it's yes, you can face some problem. But the main problem is they have at the same time bowel endometriosis and another form of the endometriosis. This surgery takes a very long time. And so in some conditions, you have to spend 4 or 5 hours in the OR for benign disease. These are some examples, but I want to save our time. The other conditions, adenomyosis, in the morning, it was very good presented. The main problem is, if I say this is adenomyosis and the other colleagues say that this is adenomyosis, there is no problem. But we know that I said adenomyosis when I invite that cases to office. When we pass the 3 months, I do nothing. I diagnose adenomyosis, and then when we pass some months, we have to pay attention to say this lady have adenomyosis. In a severe form, there is no doubt that everybody can make a very good diagnosis, but in a mild form, but there is no classification about the adenomyosis. We can only say that diffuse form or focal form. And we can use some criteria, but some of them are dia criteria, some of them are india criteria, but which one is the most important of the adenomyosis diagnosis? We need much more knowledge about the diagnosis of adenomyosis. But we know that if that uterus have a higher number of the adenomyosis feature, the pain is going up and the VAS score is going up. And so if they have a lot of lesions and the age is over 40, we can think the treatments again. When we turn back again, the ESHA guideline again, hysterectomy will not necessarily cure the symptoms of the disease. This is a general information about the treatments. When hysterectomy is performed, total hysterectomy have to choose them. But they stated that this may be particularly good option in women's significant concomitant adenomyosis. Not routine recommend the hysterectomy in that cases. Let me turn back the deep endometriosis, but which is located on the posterior part. Normally, the posterior segment have to be this one, but it's a typical conditions. The feature is a little bit black. I don't know. You can easily see that this is a typical form of bowel endometriosis, rectum endometriosis. That young lady have endometrioma, but at the same time, there's a very big nodule which is attaching to the posterior uterine wall. This is some form of deep endometriosis. The main problem all around the world is the diagnosis. When we see some cases, national approach is very different. If you go to Italy, they apply the ultrasound. If you come to Turkey, we apply the ultrasound at the same time with the examinations. But I know that in UK or United States, sonographer is a other person, and they apply that one, and you have to come to all information in the same setting. It is not easy to all around the world. In some conditions, we can see endometriosis, but at the same time, that lady has some other reason about pelvic pain. This lady had a previous salpingo-oophorectomy on the left side, and they think that that lady have a pelvic congestion syndrome, and because of that, they applied some coil in the ovarian vein. We see that there's a very severe sciatalgia, but at the same time, rectal nodule. We performed the surgery. She has a vascular entrapment on the sciatic nerve, and at the same time, rectal endometriosis. If we solve the rectal endometriosis problem, yes, that lady getting a very good conditions for dyschezia and dyspareunia, but sciatalgia have to be going on after the surgery. We have to look to all kind of the segments and make a very good diagnosis, and then talk about the treatment choices of the patients. We know that if that lady have a very big nodule, at the same time they have some kidney function problem because of the external compression of the ureter. This lady coming our center for severe dysmenorrhea. Every month she have to go to emergency unit. The oral treatments doesn't work well. The problem is the very big nodule on the rectum. Because of that rectal nodule, it makes a compression on the ureter, and when you apply the ultrasound on the abdominal way, you can make a dilatations on the kidney. This is another case. Last year we performed that surgery. She is a very young woman. Yes, she has a deep endometriosis, but at the same time, when you look at the kidney, there is no parenchyma on the kidney. When we apply the scintigraphy, 0% of the clearance rate of that affected kidney. So it's a very preventable disease. We can, if we make a diagnosis before, then we can preserve that kidney function like this case. But now she lost her one kidney because of the benign disease of the gynecological condition. I have to save my time, and I jump to some of them. When we turn back the posterior part, when we turn back again the ESHA guideline, there is no recommendations about the uterosacral ligaments and the cul-de-sac. So this is one of the biggest problems for the endometriosis surgery. It is not easy. Every time you can find a very different characteristic patients like this case. I think Salih Yilmaz, he edited this video. We performed this surgery 3 days ago. She has a small endometriotic nodule. It's only about 9 millimeters, but there's a very severe adhesions posterior part of the uterus. The main problem is the sacroiliac ligament. Sacroiliac ligament is very thick. It is about 2.5 centimeters nodule on the sacroiliac ligaments. Sometimes when we talk about the nodule, always some colleagues think that that nodule belongs to bowel, but we know on the peritoneal surface or on the sacroiliac ligament, we can face a lot of very deep nodule, especially in the severe pain condition. If we choose the conservative treatments of the endometriosis, conservative surgical method, we know that the complication risk is going down, and so it is very important. It's a very benign disease, and you perform that surgery in a very young woman. If you face some complications, urinary or the bowel complications, it is very badly affect their life. And so if we solve the problem, pain problem, the other problem, and At the same time, if you decrease the level of the pain and the level of the complication, it is very good approach to treatments of the endometriosis in a surgical way. And so some of the authors support that conditions. You know that these are very well-known endometriosis specialists, and normally some of them apply the segmental resection or low anterior resection for many years. But all of us know that if you choose some conservative treatments, if it is possible, it's a very good way to treat that cases in a clinical practice. Why we face a fibrosis, why we severe pain? It's a very long story, and there's no time to discuss that one. But if normally I can say to my condition, I am an endometriosis surgeon, but if I say that I am a fibrosis surgeon, it's a very well definition about endometriosis surgery, because we always face a lot of adhesions, and it is not very easy to deal with that problem with a surgical technique. When we look at the ECOG practice bulletin, they do not recommend adhesiolizers, by the way. When we turn back about the Cochrane review, we are uncertain about the benefit of adhesiolizers, but there may be quality of life benefit to adhesiolizers. There is no clear information about that Cochrane review, but that information coming from only 3 studies. I underline that information. And my last topic is the neuroendometriosis. We published that one, I guess, 2 years ago. Main author is Ahmet Kale, main responsible author. Normally, foot drop, some colleagues think that it belongs to sciatic neuroendometriosis, but when we look at the literature, in some sacral neuroendometriosis, they can have a foot drop. At the same time, if they have some sacral nerve root endometriosis, we know that they can make some compression on the ureter, and then it is not surprise for us. We can see some kidney dysfunction and hydronephrosis and the pelviectasia. And so if you see some problem, if you suspect that one, we have to evaluate that conditions in this case. This is a sciatic nerve endometriosis. The main problem is the pain, sciatalgia, and the locomotor dysfunction. We apply some neurological investigations, not only the gynecologic one. Pelvic evaluation is important. On the sciatic nerve, we commonly use the MR neurography to see the sciatic nerve area. But the main problem is when we apply the surgery, in some cases, there is only some pulling on the peritoneal layer. There is no other lesions. If you think that there's a very big mass which is waiting for you in the intra-abdominal part, no. You can see that lesions which is belong to sciatic nerve, and this is some endometriotic tissue there. I guess 3 months later of that case, pay attention her face. The pain is gone, but it is not easy process. In some conditions, you can face a problem and you can need a very long physical therapy during that surgery. This is another neuroendometriosis, sacral nerve roots endometriosis. Normally, the pelvic peritoneal spaces have to be this one, but in that case, there is only some pulling. It is below the hypogastric nerve, below the sacroiliac ligaments. This is belong to endometriosis. This is a very good tissue. Then we follow it, find that lesions. This is another case. There is no other endometriosis on the ovary. Some colleagues think that if some ladies have a nerve endometriosis, they can have an endometrioma or adenomyosis. These are isolated cases. In some cases, yes, they have especially for sacral nerve roots endometriosis, they can have a bowel endometriosis, but in some cases like this one, they have no other endometriosis form in the pelvis, only neuroendometriosis. If they have a previous surgery, we know that it has increased the adhesions, it can increase the complications rate. And this patient's characteristic is they have a lot of surgeries because of we haven't have very good diagnosis, we haven't have a very good surgical technique to perform that surgery. This is coming from UK. they recommend that it was published 4 years ago, Ertan Sarıdoğan group. This lady is around the 40s. They say that you can apply the excisional surgery. At the same time, you can apply hysterectomy and bilateral salpingo-oophorectomy. In a selected cases, maybe we can recommend this treatment modality, not routine recommend this one. Why? When we look at the pain scores, hysterectomy group and the bilateral salpingo-oophorectomy group, the pain level is going down than the standard excisional group. I jumped this one. Always we have to remember that sometimes some bill have to waiting for us after the surgery. These are belong to some cases. After the surgery, they can face a lot of complications. So we have to discuss all treatment modality with the cases. It is not so easy to solve that problem in that case. We need to use same language with the IVF specialists, algologists, or physical therapists, whoever wants to solve that pain problem. So we have to talk to each other and discuss the all treatment modality with that case. We need a very good team. If you haven't have a very good team, like ESHA guideline recommendation, you cannot easily solve that complications, diagnosis, treatments. It is not so easy. Thanks to these team members, we are very lucky in our center. So if I conclude my presentation, it's the teamwork, definitely. Diagnosis is the most important part of endometriosis. If you diagnose a very good way, then you can discuss a lot of treatment options with that patient. But we know that we miss the diagnosis a lot of deep and infiltrating endometriosis cases. Maybe chocolate cyst endometrium is not so difficult, but the rest of them is not easy. You need some special education about the diagnosis and the imaging methods. Treatments should be individualized according to the patient characteristics, such as medical or surgical treatments. Sometimes we think that I'm a surgeon and I always have to apply the surgery. It is not a good message for the colleague. Sometimes some colleagues think that surgery is very dangerous. I have to give a medical treatment. It is not a good way for all cases. Complete removal of the lesions is needed because of the decrease of the recurrence. Rate of major complications, we know that they are related with the aggressive surgery. Maybe we can some need to balance about the very aggressive one to some conservative one. Unfortunately, due to lack of randomized prospective studies and meta-analysis, there is no agreement on what should be the gold standard for the treatments of these conditions. Yes, we look like this ship. We have some known part, but we have a lot of unknown parts, and so we are going on in that way. I really thank about your attention.
Thank you, Taner Usta, for the excellent presentation. Now, the topic is surgical management in chronic pelvic pain, and the speaker is Ahmet Kale. Ahmet Kale, Professor of Obstetrics and Gynecology at University of Health Science, Turkey, İstanbul Gazi Osman Paşa Training and Research Hospital, Turkey, Vice President of Endometriosis and Adenomyosis Society, member of European Endometriosis League and European Society of Gynecologic Endoscopy, certification in minimal invasive gynecologic surgery, neuropelviology, and deep endometriosis surgery. Yes, Ahmet Kale, stage is yours.
Thank you very much for your nice presentation. I would like to thank the organizing committee for the nice invitation. PowerPoint.
Sadece ilgili bunu ele yapabilirse tamam. O zaman şeye gerek yok.
The chronic pelvic pain topic is a little bit, if you look at the chapters, it is very complicated, but I want to simplify as much as possible this topic. I want to give a little bit brief description about the surgical management of chronic pelvic pain. If you look at the ACOG practice bulletin, it is stated that non-cyclic pain of more than 6 months duration that is localized to the anatomic pelvis, anterior abdominal wall, or at or below the umbilicus. This is the classical definition of the chronic pelvic pain. 1 in 10 women in the world suffers from chronic pelvic pain. If you look at the etiological factors, it is gynecological reasons and non-gynecological reasons. Gynecological reasons is endometriosis. Dr. Taner Usta very well mentioned about this issue. And adenomyosis, myomas, adnexial masses, pelvic inflammatory disease, and some dense adhesions can also cause chronic pelvic pain. And we also keep in mind that while evaluating chronic pelvic pain patients, there might be a irritable bowel syndrome, and there might be interstitial cystitis, or some kidney stones also can cause chronic pelvic pain. While evaluating the chronic pelvic pain, there are 3 types of pain. We should keep in mind that one of them is neuropathic pain, another one is somatic pain, and the other one is the visceral pain. How can we differentiate somatic versus visceral pain? Somatic pain arises from the damage to the body tissues. Often the musculoskeletal system is responsible for this kind of pain, and is important issue is that well-localized pain. For example, burning pain or fracture pain or muscle pain is related with the somatic pelvic pain. Another one is the visceral pain. It arises from the visceral internal organs and poorly localized. It is important. And for example, appendicitis or cholecystis is related with the visceral pain. If you look at the inferior side of the picture, the somatic pain is well localized. The patient can show easily that my pain is there, but visceral pain is poorly localized, so she cannot or she cannot very well map the area of the pain. Neuropathic pain is another pain. It is related with the injury to central or peripheral nervous system injury. It is really it's completely originating from the nerve system. It is like a shutting, burning, electric-like pain, and it's sometimes like a tingling and stopping pain can occur. This kind of pain, but this kind of pain is more severe when compared with the somatic or visceral pain because it is completely originating from the pelvic nerves. While evaluating the patient, the history and the physical examination is important. If we specific the disease associated with pelvic pain, it is easy to treat the conditions, but sometimes we cannot easily define the exact etiological factors. In these cases, multidisciplinary approach is important to define the etiological factors of the pain. We first of all begin with the examination. The Carney sign is important. If we raise the leg of the patient, the pain decreases. It means that the pain is coming from the inside of the abdominal pelvis. But if the pain increases, it means that the pain is coming from the abdominal wall. So this is called the Carney sign. Another sign is the Faber sign. Another name is the Patrick test. Flexion, abduction, external rotation of the legs means that it is called a Patrick test. If it is positive, it means that the pain is originating from the hip, lumbar, or sacroiliac region. So it is not related with our area or our pelvic area. Gynecological examination is important. As you see that when we look at with speculum examination, we can easily see the endometriotic nodule at the fornix or vagina, so we can easily find out the diagnosis. And hypertonic pelvic floor also is important. It is associated with the endometriosis, adenomyosis. Maybe sometimes it is associated with pudendal nerve pathologies or sacral nerve root pathologies. So we must touch the pelvic floor whether there was a pain or not. while evaluating the patient, our finger is important. We must also, or we should also touch the sacrouterine ligaments, whether there was a pain or not. If there is a pain in sacrouterine area, or this means that this pain is originating from the pudendal neuralgia. So detailed examination is very important. We couldn't also overlook the top red flag symptoms, because we just only not concentrate on the pain, because additionally the patient has a postcoital bleeding. It means that the patient have cervical cancer. If the patient has a postmenopausal bleeding, she might have an endometrial cancer, or she has a postmenopausal pain. Maybe we can encounter some ovarian cancer cases. So pain and additional factors are important. Imaging also important. As you see that there is a left side endometrioma, adenomyosis, and we also keep in mind that at the right top side there is a color. This color means that the pelvic congestion syndrome is related with the pain. So sometimes hydrosalpinx or fibroid also can cause pain. The simple examination is important. For example, at the upper side, as you see that there is a fibrotic band and breach can cause pain. So we can easily identify that this fibrotic tissue at the vaginal introitus can be seen, and also we can easily excise this lesion and treat the pain. And sometimes we can miss only just hydrosalpinx can cause pain. So we must also keep in mind that only the hydrosalpinx is important for pain cause, for the cause of pain. Another factor is the iatrogenic pelvic pain. Nowadays, or TVT and TOT operations is commonly performed in Turkey, also in other countries. Maybe some countries restricted this kind of mesh usage during the surgery. But this lady came to us or admitted to our clinic as a chronic pelvic pain. We find a mesh that is implanted on the vagina, and we entered the retrius places and excised this mesh. As you see that the integrated mesh fibers on the surface of the vagina, it means that this patient has really very intractable dyspareunia and dysmenorrhea during the sexual intercourse. So while advising the stress urinary incontinence surgery, and if we wish to use a TVT or TOT materials, we inform the patient in detailed manner. That's because the mesh aeration or this kind of meshes can cause really intensive pain. So we must also say this patient and this patient. If we look at the outcomes of the transvaginal mesh excision, as you see that 124 patients underwent surgical removal of mesh with pain, and after the surgery, 80% of the patients are pain-free. It means that if you excise the mesh, easily the pain will be free. This is another pathological factor, that myoma. As we know that myomas are mostly asymptomatic cases, but sometimes they are symptomatic. In this case, if it is very big and it is very huge, it can cause pain. In these cases, we can offer laparoscopic surgery, and this surgery can be done easily with laparoscopic ways. And as it is known that it is a minimally invasive surgery, and with laparoscopic way, we can easily excise the myoma, or we can make this kind of surgery in a laparoscopic way. If there is pelvic pain, infertility, and abnormal uterine bleeding, there might be adenomyosis. This patient might be an adenomyosis. So if we have lots of treatment modalities, one of them is the progestins, and another one is the GnRH analogs. But sometimes if this kind of therapies fails, the second option might be a surgery. And pelvic pain is relatively frequent symptoms, especially in adenomyosis cases. and depth of the myometrial invasion of the ectopic endometrial size does correlate with the pain, but pain doesn't correlate with the location of the adenomyosis. And immune environment leading to secretion of neurogenic substances may result in pelvic pain. Maybe pelvic pain could be a consequence of an increase in contractility of the myometrium in response to hormonal immune changes. This is the cases that failed. She admitted to our clinic with pain, but she had prescribed lots of drugs, progestin generis, but she wants to preserve her uterus. In these cases, we make a surgery. We excise the diffuse adenomyotic tissues from the uterus, and we try to protect her uterus. Just excise the diffuse adenomyotic tissues, and after excising the tissues from the uterus, we couldn't suture only and in a simple manner, because this is a different kind of techniques we used while performing this surgery, because this is a kind of double flap method. We are overlapping the tissues or serosal layer. If you don't overlap, if the patient becomes pregnant, the uterus can rupture easily. So overlapping method is very important, especially for diffuse adenomyosis cases. If we change our perspective, for example, if patient comes with pain and has a myoma uteri or adenomyosis, diffuse adenomyosis or focal adenomyoma, do we have every time make a surgery, or do we every time make a drug? And if both of them doesn't give response our therapy, what can we do? Another option is that this emerging therapy is that radiofrequency ablation. Radiofrequency ablation is another method for especially uterine myomas. And if the pain persists, for example, as you said in this literature, the pain decreased at least 74% after the radiofrequency ablation surgery or radiofrequency ablation method. How does our radiofrequency ablation works? It destroys tissue cells from coagulation and makes a vascular thrombosis in the blood vessels that supply the myoma, and inactivated hormonal receptors within the myoma that prevents tumor tissue from growing. Can we do radiofrequency ablation for adenomyosis cases? Yes, we can also do radiofrequency ablation for adenomyosis. After the ablation, 50% of the VAS scores decreased after radiofrequency ablation methods and after 1 year. It appears to offer similar pain relief to the conventional uterine sparing methods, and it is really a beneficial method, especially for chronic pelvic pain. This is our cases, and with the transvaginal ultrasound, as you see that we are inserting the needle inside the myoma uteri, and it works like a thermal ablation, and we are giving and at the tip of the needle, as you see that there is a heat. The heat of the needle is approximately 85 centigrade Celsius. We are now destroying the myoma uteri, and after whitening of the myoma uteri, as you said, the whitening is completed. It means that the myoma tissue is destroyed completely. So this method is much more easier than the surgery. So it is a reproducible method, and also it is a repeatable method. You can repeat again. For example, the patient pain persists after 6 months and admitted to you. You can also use the second and third times, fourth times. But if you wish to perform surgery, you maybe one or second time perform surgery, but at the third time, you can think that whether I perform surgery or not, this patient. So radiofrequency ablation is a different method or alternative method, especially for myomas or adenomyosis cases. I want to shift to neuropelviology. Neuropelviology is an emerging discipline for the management of the chronic pelvic pain. The godfather of the neuropelviology is the Mark Pessover. Is a new discipline focused on pathologies of the pelvic nervous system and possibilities of improved neurologic diagnosis in chronic pelvic pain. Briefly, it means that it is really deals with the somatic nerve system in the pelvis. For example, sciatic nerve passing through the pelvis, obturator nerve passing through the pelvis, pudendal nerve passing through the pelvis. Neuropelviology is mapping whether this pain originating from which nerve. It is related completely with the nerves. So while evaluating the chronic pelvic pain patients, we must also keep in mind that this pain might come from the this kind of nerves: sciatic nerve, pudendal nerve, and obturator nerve. As you see that this is a video. Our video. This is a sciatic nerve. The patient has a pain, and there are a few nerves that are coming through the pelvis. One of them is the biggest nerve is the sciatic nerve. Another one is the obturator nerve and pudendal nerve. These three nerves are related with the chronic pelvic pain. So in some cases, after neuropelviologic evaluation, we perform this kind of surgery on sciatic nerve or pudendal nerve because some nerve entrapment issues can entrap the sciatic nerve and can cause really intractable pain. And this is our evaluation before the surgery. This is a neuropelviologic evaluation. While evaluating this patient, I feel myself sometimes as a neurologist or neurosurgeon, not a gynecologist, because I really want to deal with only pelvic nerves. As you see that, for example, anal pain comes from the pudendal nerve or sacral 2 or 3, and perineal pain comes from pudendal nerve, sacral 2 through to sacral 3, and low back pain comes from the L5 to S5 nerve roots. It means that while evaluating patient, we are mapping where the pain comes from, where is the dermatome of the pain. So neuropelviologic evaluation is important. And in my routine clinic practice, I am performing gynecologic examination to find out the chronic pelvic pain etiology. And also, I additionally make a neuropelviologic examination to complete my examination. This case is a very, very interesting case. At night, her husband recorded this video and showed me that, as you see, that this is a pain attack of the patient. It is very intractable sciatic pain, but this lady has no discal hernia. The sciatic nerve is compressed by the vascular and piriformis vascular system and piriformis muscles. So as you see that you cannot do anything, and most of these kind of patients also take some drug, especially opioids. But when the time passes, the opioid, she cannot tolerate opioids, or sometimes cannot be useful. Making this kind of drugs. So we performed surgery to this lady. This is a external iliac and iliopsoas muscles, and we dissected off, and we also published these cases. As you see that there are lots of aberrant hypogastric veins, real ligated. These all veins or arteries are superior side of the sciatic nerve. My aim is to release the sciatic nerve because to stop or to end up the patient's pain. As you see that there is a white fibroid, white big nerve is the sciatic nerve. There are lots of muscles, piriformis muscles, and lots of vascular system are entrapping the sciatic nerve. So this operation is also a very dangerous operation because this kind of veins, if bleeds, this kind of veins, the catastrophic complications or bleeding can occur during this surgery. So the other teams should be alert during this surgery, especially the other teams also should be alert to this kind of surgery. The pudendal neuralgia is another kind of chronic pelvic pain or etiological factor, especially the Michael Hibner showed that the patient cannot sit on a rough surface. The pudendal neuralgia patient cannot easily sit on a rough surface, but lies down, the pain relieves. So sometimes in some cases, if the treatment modalities fails, in these cases, surgery is mandatory. This is a pudendal nerve surgery, and we release the pudendal nerve. As you see that it is very thin nerve. So while dissecting, we can also easily cut this kind of nerve. So pudendal neuralgia is another important factor for chronic pelvic pain. While evaluating chronic pelvic pain patients, we should also keep in mind that pelvic congestion syndrome is another syndrome that is related with the chronic pelvic pains. As you see that 40% of the cases of chronic pelvic pain are associated with pelvic congestion syndrome. And the diagnosis are tortuous pelvic veins with a diameter more than 4 millimeter or diameter of the ovarian vein over 8 millimeter may suggest pelvic congestion syndrome. And the gold diagnostic criteria is the CT or MR venography is important while evaluating. And for diagnosis of this kind of patients, we can prescribe some drugs, GnRH agonists or progestins to relieve this kind of symptoms. But most of the times, we perform a coil implantation or coil embolization is important. As you see that there is a tortuous veins. The left side is the left ovarian vein is dilated and due to chronic pelvic pain. The chronic pelvic pain is the cause of the, so the interventional radiologist put a coil inside of the ovarian vein. As you see that the dilated vein is reduced. As you see that the left side is dilated vein. At the end of the procedure, platinum coil is inserted in the left ovarian vein, so the vein is reduced and the pain symptoms resolves after this procedure. So this we should also keep in mind that while evaluating this patient, this pain might be originating from the pelvic congestion syndrome. Ovarian remnant syndrome is another etiology. Sometimes the women admitted to our clinic as a bilateral hysterectomy and bilateral oophorectomy, but if the pain persists, it means that some residual ovarian tissue after previous oophorectomy can be found inside the pelvic wall. For example, if you perform a very difficult surgeries, for example, endometriosis, PIDs, or if you perform a multiple previous abdominal surgeries, some ovarian remnants is found inside the pelvic cavity. So this kind of ovarian mass tissue, residual tissue, may eventually cause pelvic pain. So pelvic ultrasound is important. You can see small cysts inside the pelvic cavity. So you can prescribe some pharmacological treatments. But if this fails, the surgery is important. You should remove the ovarian remnants with laparoscopic or any other ways to decrease the chronic pelvic pain. In this case, as you see that there is an ovarian remnant and the patient underwent bilateral salpingo-oophorectomy, but small ovarian tissue is in the pelvic side wall. So there is a small cyst, but may causing a chronic pelvic pain. So we should also keep in mind that there might be an ovarian remnant. So this ovarian remnant can cause chronic pelvic pain. There are some patients some colleagues are asking that the role of hysterectomy is in the treatment of chronic pelvic pain. And in the absence of any obvious pathology, 40% of the women having a hysterectomy for chronic pelvic pain may continue to experience pain after surgery. It means that every time hysterectomy doesn't solve the problem. So we must inform the patient. Removing all of the internal organs, hysterectomy and ovary, doesn't mean that your pain will resolve completely. And adenomyosis is another topic that if you perform an adenomyosis, the symptoms of the pain will resolve or no. Adenomyosis doesn't add any benefit to the patient, so there is no value to performing adenomyosis. My last sentence is that many conditions can cause chronic pelvic pain. All can have similar symptoms, and sometimes that makes it hard to figure out source of pain. Healthcare provider will review symptoms and medical history in a detailed manner to diagnose the cause of pelvic pain. Physical, gynecological, and neurophysiological exam help in determining the cause of pelvic pain. And multidisciplinary team is sometimes needed to deal with chronic pelvic pain. Surgical treatment is the treatment method that should be applied when necessary in patients with chronic pelvic pain. Thank you very much for coming to Istanbul.
Thank you, Professor Kale. The next presentation would be one of the winning abstracts. However, due to a delay in our flight, the next presenter could not catch the schedule. So this is we are closing the session. The next session will be at 13:50 after lunch break. It will be on time. Thank you.
Soru cevap, question and answers. They are asking question and answers.
Okay, one question. I thought there would be one question, but it's okay. We are closing the session now. We are already behind the schedule. Thank you.
Ses. Var.
Okay, well, welcome back after lunch. My name's Andrew Horn. I'm from the University of Edinburgh in the UK. It's a real pleasure to be here and be part of this meeting. We've had some great sessions so far already, and I'm looking forward to this afternoon. I'm here with my co-chair, Firat, and I'll let Firat introduce himself.
Hello, everyone. I'm Fırat Büyüktaşkın. It's a pleasure to be a part of this wonderful meeting, both with magnificent Turkish Endometriosis and Adenomyosis Society, and also the team that I had the chance to work with the past year as a postdoc in Edinburgh. So it's even more special for me. And we have a great session upon us.
Thanks for that. So it's a pleasure to introduce our first speaker, who's my colleague, Professor Katie Vincent from the University of Oxford. In Oxford, she's a senior pain fellow and consultant gynecologist. And Katie's going to talk to us about mechanisms of pelvic and comorbid pain. Thank you, Katie.
Thank you very much, Andrew, and thank you very much to the organisers for inviting me here. It's a real pleasure to be in Istanbul. And so I'm going to talk about comorbid pain conditions with pelvic pain. These are my disclosures, but they're not relevant to today. So I'll start by thinking about why we should be interested in comorbid pain, then think about why comorbid pain might actually occur, and then how we can inform our clinical practice by thinking about comorbid pain. So why are we interested in comorbidities? Well, we've known for a long time that pelvic pain conditions are commonly comorbid. It's more than 20 years ago that interstitial cystitis and endometriosis were named the evil twins of chronic pelvic pain syndrome. And subsequently, IBS was added to that picture too to describe evil triplets. But actually, we know that women with pelvic pain frequently have more than one type of pain. So we see non-cyclical pain overlapping with dyspareunia, dysmenorrhea, and also bowel and bladder pain. But we know that women with pelvic pain can also have other chronic pain conditions. So more than half of young women with endometriosis already report a comorbid pain condition. And if we look in an adult population, we can see that there is a really strong overlap between all of the three pelvic pain conditions, IBS, bladder pain syndrome, and endometriosis, but also other common conditions like fibromyalgia, chronic low back pain, and migraine. And in fact, NIH, again, almost 15 years ago now, put out a document highlighting the fact that we really commonly see chronic overlapping pain conditions, and actually that lots of these are more likely to occur in women than they are in men. And there's a list here of some of the sort of more common of the chronic overlapping pain conditions, and I've just highlighted in red those that we commonly see as a gynecologist, and the fact that they are high up in this list. And we know that comorbid pain conditions are associated with a reduced quality of life. We know that individual chronic pain conditions are associated with reduced quality of life. And this was some of our data showing that women with pelvic pain were likely to have a reduced quality of life, but those who had both endometriosis and bladder pain syndrome had significantly worse quality of life than those with either of the conditions on their own. We also know that treatment of one pain might improve another. So this is work from Italy, again, more than 20 years ago now, showing that if you treat dysmenorrhea with hormonal treatments, so treatments that would be expected to only treat the dysmenorrhea, you actually see a change in symptoms of IBS. So the number of days that you have pain from your IBS, but also the intensity of pain. And similar work was done by the same group, looking at women who had renal stones and how many urinary colics they had a year. And again, showing that if you treat dysmenorrhea, you actually reduce the impact of this renal colic. So why do we think that we see comorbid pain? Well, you've already heard from Nilofer this morning about the overlap genetically between endometriosis and other pain conditions. And we know that there is good evidence that chronic pain itself is heritable. So there have been a number of twin studies, and heritability seems to be between about 30 to 60%. But we also know that twins are exposed to multiple common other factors. But actually, if we adjust for environmental and lifestyle factors, we see that about 16% of any type of chronic pain is heritable. But once we look at severe chronic pain, we're then going up to more like a third. And there's been a recent genetic study looking at the overlap between genetic risk for multiple different types of chronic pain conditions. Now, I will highlight here that they did look at pelvic pain. They looked at IBD, IBS, and cystitis, but they didn't include dysmenorrhea or endometriosis within these analyses. But what they found was that there were two subgroup or two groups of genetic overlap within this cohort. The first and strongest factor was loaded from all of the chronic pain conditions, suggesting that there was common genetics underlying them. But then there was a second factor that loaded all of the musculoskeletal conditions. So there definitely seems to be a genetic vulnerability to both chronic pain and also chronic musculoskeletal pain. But we also know that risk factors for developing chronic pain are much wider, and we know that environmental factors play a key role as well. I'm only going to focus on one of those here for the purposes of time, and that's to talk about adverse childhood experiences, because we're increasingly aware that adverse childhood experiences are risk factors for all sorts of chronic health conditions, but particularly for chronic pain. So these are defined as traumatic experiences that occur before the age of 18, and they include all types of trauma, so neglect, abuse, and general familial or household dysfunction, so parental separation or a parent being put in prison, for example. And a relatively recent WHO survey suggested that on average, people experience between 2.5 and 2.9 ACEs. We know that in the UK, 47% of the population will have had at least one, and 12.3 will have had more than 4. And as far as risk factors for chronic health conditions broadly, 4 is considered to be a sort of pretty significant risk factor. If you've had 4 adverse experiences in your childhood, you're at fairly high risk of developing long-term chronic conditions. And if we look at the specific relationship with pain, we see that adverse childhood experiences increase the risk of any kind of chronic pain in adulthood. And they're also associated with developing widespread pain and with experiencing greater pain severity. And the more ACEs that you experience, so as a dose effect like we heard about earlier, then the worse your pain will be, and the more dysfunction and disability you will have associated with your pain compared to those who have chronic pain but don't have adverse events in their past. So pain vulnerability may be to do with environmental factors, but we also know that some of the mechanisms that underlie chronic pain conditions can be common, even from very different pain conditions. So chronic inflammation, again, is something that we're becoming increasingly aware of as its risk factor for a wide variety of different chronic conditions. We know that it's very common, that it has different processes associated with it from acute inflammation, and that as with all of these things, it's usually a complex etiology, but it's we're becoming aware that it's the most significant cause of mortality, with more than 50% of deaths attributed to some form of inflammatory-related disease. And we know that chronic pain conditions commonly have chronic inflammation underlying it. And these two flowcharts are from the new ICD-11 for categorizing chronic pain. And the one you can see on the right of the screen is musculoskeletal pain, and on the left of the screen is visceral pain. And both of these have a subcategory within them for pathologies associated with chronic inflammation. So it's well established as being a predisposing factor to chronic pain. And we commonly see chronic inflammation with endometriosis, but we also see it with rheumatoid arthritis and inflammatory bowel disease. And I think it's going to be really interesting as we start to compare those conditions to understand whether similar inflammatory markers might be responsible for pain in all of these conditions. But we also know that one pain can predispose to another. You can have one first and then subsequently develop another one. And one of the key pathways that we're interested in, particularly in the pelvis, is visceral referral. And we've already heard about the complex innervation of the pelvis in the two talks preceding lunch. And so you can see that these pelvic viscera share common nerve roots, common spinal cord segments. And we see that even if we have pathology localized to one of these viscera, we can end up with hypersensitive symptoms from the other viscera. And some more detailed work has looked at which organs cross-refer to each other. And you can see that, we see a wide variety of them, colon to bladder, colon to rectum, for example. But interestingly, they seem to be divided into regional areas. So you see relationships between the thorax and the upper abdomen, or between the pelvis and the lower abdomen, but they tend not to cross across that threshold, suggesting that it's more about spinal segments than about a complete central nervous system sensitization. And what we see is that actually having one visceral condition can start to lead to the development of visceral sensitivity. So this is work from Frank Tu's group in Chicago, where he has a relatively large cohort of women with just dysmenorrhea, and they don't describe any other pelvic symptoms. But when he does a non-invasive bladder filling paradigm, so this is where the women empty their bladders and then drink a set amount of fluid, it's just over a pint, 600 mils of fluid, and they then report when they reach certain sensations. So when they would first feel that they have some sensation from the bladder, when they first feel the urge to go to the toilet, and then when they're maximally full. And what we can see is that in the women with more severe dysmenorrhea, they actually report pain from their bladder filling much earlier than those without dysmenorrhea. So even though they don't describe any bladder symptoms, they do have a more sensitive bladder. And it seems that the bladder is the most common of the pelvic organs to become sensitized. And it seems that the bladder itself is less effective at inducing sensitization in the other pelvic organs. We also know that nociplastic pain is really common, and this is part of the new categorization of chronic pain. So we know that there are 3 subtypes of chronic pain now. There is nociceptive pain, pain driven by a stimulus in the periphery. There is neuropathic pain, which is pain that is from damage to a sensory nerve fiber, either in the periphery or in the central nervous system. And then nociplastic pain, this is an umbrella term that covers pain which arises from altered nociception. And again, this flow sheet here is, again, from the ICD-11, and shows the subcategory of chronic primary pain. So that is pain conditions where we don't identify any underlying pathology. And those are all considered now to arise from nociplastic processes. And what we don't really know is whether nociplastic processes are the cause of pain, or whether they develop secondary to having chronic pain and repeated painful insults turning up the volume of the signals going through the nervous system, or whether in fact, which is what I think is most likely, that it is both, and they can either be a cause or a consequence of chronic pain. But if they were also producing a vulnerability to developing pain, it could be a very good mechanism by which one condition could then start to sensitize you to develop another. And what we're increasingly aware of is that nociplastic processes exist in a subgroup of women with chronic pelvic pain. And so we've seen that they can have widespread pain. Their pain doesn't just have to be focused in the pelvis, even if they only have a diagnosis of endometriosis. We see that other measures of involvement of the central nervous system, so things like fatigue, sensitivity to lights, to sounds, to smells, for example, and other symptoms that would suggest that are much more common in chronic pelvic pain. So you can see that some people will only have 1 or 2 of those, but some people will have sort of 4 or 5 of these different sensitivity symptoms. We can see that if we apply painful stimuli either to the pelvis or to other control areas like the hand or the foot, for example, that people with chronic pelvic pain will report much greater pain to the same intensity of stimulus. So again, suggesting a sort of wind up of the sensitivity of the nervous system, and that even neurotransmitters in the brain that are responsible for exciting these pathways are increased in association with chronic pelvic pain. Of course, it may be that what we do to treat our chronic pain patients also leads to the development of a new pain. And one of the things that, again, we're increasingly recognized is the fact that we see relatively high incidences of post-surgical pain after any kind of surgery. So about 10 to 20% of people will still have a new type of pain after their operation 3 months later. And over 2% of those will describe severe pain, and these figures pretty much persist beyond that time. So if you look at a year or 2 years, there's still very high rates of chronic post-surgical pain. And it doesn't seem to be related to the size of the incision, because the rates seem to be roughly the same between laparoscopy and laparotomy. It doesn't seem to be related to the processes, day surgery or inpatient processes seem to be the same. And it's thought that neuropathic features occur in about 30% of those, but we don't fully understand the other features that might lead to the development of chronic post-surgical pain. We're increasingly recognizing what the risk factors are, and unfortunately for our population, they are being female, being anxious, being in pain before your operation, and having inflammatory processes within the pelvis. So all of those things are likely to be features that apply to at least some of our patients with endometriosis before they go for surgery. And some of our data from a cross-sectional survey was looking at rates of neuropathic pain in women with endometriosis. And we saw a strong relationship between the number of surgeries that women had and the rates of neuropathic pain. So you can see on this graph, the top blue line is the rates of neuropathic pain. And if you'd only had 1 surgery, so the surgery you would have needed to have diagnosed the endometriosis that allowed you to enter the study, then we saw about 30% would have been classified as having neuropathic pain. By the time they'd had 5 or more surgeries, we were looking at about 50%. Now, of course, this was cross-sectional data, so it doesn't say for definite that surgery itself is causing the neuropathic pain. It may be that if you have neuropathic pain, you don't respond to standard treatments so well, and therefore you're having repeated surgeries because of that. But I think it does highlight that there's a relationship there that maybe we should stop and consider. Another common form of treatment that we use for all sorts of pain, both acute and chronic, is non-steroidals. And some new data came out in the last couple of years that's really made people stop and think a little bit in the pain community. And this was looking at a cohort of people who had acute back pain and looking at who of those went on to still be describing chronic back pain at 3 months. And you can see on here, the orange line throughout these graphs is those who remained in persistent pain. The green line is those who, whose pain dropped. And they looked at expression of genes in the blood of this population. And what they saw was that there was a significantly higher gene expression in those whose pain resolved than in those whose pain persisted. And when they looked at this in more detail, it was about the inflammatory response, but it was about the inflammatory response at the time of the acute pain rather than at the time that the pain had resolved. And so they then went on to explore this in various human populations and animal models to see whether damping down the inflammatory response was actually a predictor of going on to develop chronic pain. And that's certainly what this data looks like. So in their model, it was the use of non-steroidals and having more sites of pain at the time that you had acute pain that were significant predictors of going on to develop long-term pain. Now, I don't think this means that we should stop using NSAIDs in our young women with dysmenorrhea, for example, because they are actually a treatment that works. but I think it's something that we need to be keeping in mind and following up how the data comes out in the future. So, how can thinking about comorbid pain conditions inform our clinical practice? Well, I think it can really improve our history taking. We know that our women appreciate being taken seriously and being given the time to describe their symptoms. So actually, we can ask about all their pain symptoms, not just their pelvic pain. And I would really recommend that we think about asking, do you have pain throughout your body? Because I think this widespread pain as a marker of nociplastic processes is really important to understand. We know that headaches, particularly migraine, are commonly comorbid with endometriosis and with pelvic pain more generally, and therefore we should really take a detailed headache history and confirm whether migraine is actually present, because some people are given that label a little bit loosely, but also make sure that we know whether or not they have aura associated with that pain. We should ask about other diagnoses, because if you say, do you have chronic pain or pain elsewhere, but actually things like inflammatory bowel disease or rheumatoid arthritis are quiescent at the time, they may not think to describe that, but they will have still had those inflammatory processes that may predispose them to developing other things. And perhaps we should ask about a family history of pain, because that might help us understand some mechanisms. And I also think we should inquire about adverse child events in a very gentle way, allowing people the opportunity to talk about it, but then making sure that we don't then subsequently imply that any pain that they have is psychological. So there's a really great book for people who are interested in this called The Body Holds the Score, that talks about how our bodies hold a record of what our past experiences are. And I think those kind of descriptions can be really helpful to explaining to patients that it's not just that they didn't cope with their adverse events that means they now experience pain. I think thinking about comorbidities can really help us think about whether we should do further surgery in our patients with persistent pain. So we know that having endometriosis significantly increases your risk of having a further surgical procedure. But work from Susie Asani and Dan Claw in Michigan showed that every point increase on the questionnaire that measures centralized pain, so a measure of nociplastic processes, was associated with a 27% increase in the risk of still having persistent pain 6 months after a hysterectomy for chronic pelvic pain. So that should really make us stop and think. I also think we can rationalize our use of medication. We can remember not to use the combined pill for patients with migraine and aura. But we can also think of their treatments that might address more than one type of chronic pain. So, for example, hormones can be used, as we've heard already today, for endometriosis, but actually we know that some people have very hormonally driven headaches, so actually maybe our hormone therapy could be tailored to match the time when patients don't suffer with worse headaches. We know that joint pain is often cyclical as well, so we could ask those questions too. We've got increasing data that histamine-related processes are responsible for many of the visceral pain conditions. So maybe if we have patients who describe IBS-type symptoms, bladder pain-type symptoms, and asthma, for example, maybe we could try a trial of antihistamine regimes as are already in the guidance for the management of bladder pain syndrome. We could also just think about reducing the overall pain burden for our patients. So can you manage any of their other symptoms, or do you need to refer them on to another specialist, to a headache clinic, to a gastroenterologist, for example, to ensure that their comorbidities are properly managed? But we also know that our treatments can often worsen symptoms. We heard about joint pains with aromatase inhibitors, for example. So should we stop and think, if they have comorbid pains, is there a risk that we will actually make them worse with our treatments? We know that, as I said, hormones for headaches and joint pains, but also non-steroidals can massively worsen medication overuse, headache, and also inflammatory bowel disease. And I think, though not currently well evidenced in pelvic pain, I think for those who do look like they have nociplastic processes and have multiple pain conditions, particularly with widespread pain, we should maybe think about whether a centrally acting drug might be useful. And again, you might not feel comfortable prescribing that yourself, but you can talk to your pain clinics or the complex musculoskeletal clinicians who are very experienced at using drugs such as duloxetine, and in the US, a lot of success with milnacipran as well for conditions like fibromyalgia. I also think it's really important that we think about the education we give to our patients. We often hear stories of patients who come and have been told that all their pain was related to their endometriosis, and then they're massively disappointed when their symptoms don't improve after surgery, or they keep requesting further surgeries when they have another procedure. And we know in chronic pain more generally that pain education is a key part of the management of chronic pain and is one of the most evidence-based components of chronic pain management. And Amelie Marden from Australia published in Pain just at the end of last year her review of all the guidelines around the world on the management of chronic pelvic pain and how poorly referenced pain education was in any of them. So it's just not really something we provide, particularly within a gynecological setting. And I think explaining to patients what chronic pain is, the different processes that can be involved in the development of it, can be incredibly helpful. And I think if we think about comorbid pains, it's really important that we move away from thinking solely about a kind of peripheral pathology or organ-driven approach to thinking much more holistically about something that's going to tackle their full pain burden rather than just their pelvis. So we can consider lifestyle approaches, and that's sometimes something that patients find quite hard to hear, particularly when they've had a very long journey to getting a diagnosis and to getting taken seriously, to then start to talk about exercise, meditation, diet. That can feel really dismissing, but actually, if we talk about it in the context of pain education and in the context of benefiting multiple different types of pain, then I think that can feel easier to hear. But I also think we need to educate our patients on our rationale for why we're not using medication or why we're not recommending further surgical procedures. We're all very used to taking consent for a surgical procedure, for example, but really explaining in detail why we wouldn't recommend another operation and why we would suggest taking another approach. I think it's much easier for our patients to buy into that if they really understand the rationale. So, in conclusion, I think that comorbidities are really common in pelvic pain. We know that multiple mechanisms probably underlie this observation, and I think over the next few years we're beginning to see a lot of work unpicking all of these, and I think we'll have a far better understanding of which are the key mechanisms and what order things are happening in the next sort of 5 to 10 years. We know that chronic pain is affected with low quality of life, but comorbid pain conditions definitely worsen this. But we can be positive about it. Even if we haven't got any more treatments we can think of for their pelvic pain, then maybe we could treat their headaches or their joint pain and improve their pain burden overall. And then we might find that they become generally better, including their pelvic pain. I think we should think about comorbidities to rationalize our clinical approach, and that understanding comorbid pain might also help us develop novel therapeutic strategies. So thinking about what can we translate from rheumatoid arthritis that would make sense for those that have comorbid rheumatoid arthritis and endometriosis, for example. And that's just to acknowledge the team that I work with clinically and academically and the funders of our research. And I thought I'd just leave you with this painting that came from a public engagement art project that one of our PhD students, Danielle Perret, did a couple of years ago. And this is a painting that a woman made of her endometriosis-associated pain. And as you can see, that's not just within her pelvis.
Thanks, Katie. That was a really informative overview, and you kept really well to time, leaving a few minutes for questions. Does anyone have any questions? Maybe got time for 2 questions. Can I just start by asking you what your thoughts are about prescribing perioperatively and adjuvant neuromodulation, because you showed very nicely this increase in neuropathic pain. Is that something that you think is helpful or could be helpful?
I think that's a really good question. There have been a few studies looking at it in pelvic pain. There have been some other studies looking more broadly in other conditions, and I don't think we have a definitive answer at the moment. I think that if we think about chronic pain reflecting changes throughout the central nervous system, then that vulnerability is already there, that you would amplify the signals from the tissue damage, and therefore just a short period of perioperative neuropathic antihyperalgesic medication is probably not going to be helpful. I wonder whether for people who don't have any pain beforehand, it may stop the development of a a first chronic pain condition, if you see what I mean. But I don't think we have well enough phenotyped cohorts and big enough data sets to be able to answer that yet. But I don't, we know from GAP, for example, how many side effects people had from gabapentin if they were using it chronically. You really don't see that with acute high dosing at the time of an operation, and therefore maybe it's a relatively low risk approach to reducing a big problem.
Tanner, thank you.
Thanks so much. Very nice presentation. What do you think about the central sensitization? So sometimes we diagnose one case, deep endometriosis, and she's suffering for 10 years. When we apply the surgery, after the surgery, when we ask, how is your pain? The patient said that my pain is gone. But in some condition, we said that some patients have a central sensitization. Because of that, you do a lot of things, but the pain is going on. How can we explain these two conditions?
So where I talked about nociplastic pain, that's thinking about central sensitization type processes. It's just been, the language has been changed slightly by IASP, which is maybe not that helpful. I think we know that not everyone develops central sensitization. So not everybody with chronic pain has central sensitization. And there will be a subgroup for whom an active ongoing peripheral driver is the cause of their pain. But there'll also be a group where it wasn't at all the cause of the pain, and another group that will have both central sensitization and an ongoing peripheral driver, and therefore may only see a partial benefit from surgery. I also think we don't fully understand what the processes that happen around surgery are, because we know that even doing a diagnostic laparoscopy, if we talk to patients at 6 weeks and even 3 months, they will often describe their pain as being a lot better. And then as they start to return back to their normal functions, their pain level increases again. And I do wonder whether there's something a bit like the question Andrew asked about the general anesthetic and the other medicines that are given around the time of surgery that may damp down that central nervous system excitability for a temporary length of time. And I also think there's something about the process of laparoscopy when we tent the abdomen, and that probably then also stretches out the pelvic floor as well, and whether we see less of those musculoskeletal factors that we're increasingly aware of in chronic pain. And I think we definitely know that pelvic floor spasm, for example, contributes to dyspareunia and bowel and bladder symptoms. And so I wonder whether that gets kind of released temporarily after surgery, and it's only as they start to return back to their normal function that we really see those symptoms return. But I think we need much better data to really understand who it will be that will get benefit from surgery, given particularly with the deep disease, how high risk it can be.
Okay, Engin, final question.
Thank you, Dr. Vincent. Is primary dysmenorrhea is a precursor for the future development of the endometriosis? If your answer is yes, we are using in these teenage girls much more non-state anti-inflammatory drugs. Maybe this is another etiology for the future development of endometriosis. What do you think?
I think we have a real problem in the literature at the moment, because the term primary dysmenorrhea is used very widely, and actually a laparoscopy hasn't been performed, so we don't actually know whether all pathologies have been excluded. So I definitely have an interest in whether dysmenorrhea itself could be a predisposing factor to all sorts of chronic pain conditions, and we're running a study looking at that at the moment. I think in our teenage cohort, when we do MRIs, we see relatively high rates of adenomyosis as well. And so I also wonder whether what we used to term as primary dysmenorrhea may well be adenomyosis related. And I don't think we have good enough data to really understand what adolescent endometriosis looks like either. And again, I know that there are lots of groups, particularly in the US, looking at that at the moment. So hopefully, again, that will be something we have a better understanding of.
Okay, well, thank you very much again, Katie. Let's get on to the next speaker.
Again, wonderful speech from Katie, and you might have some research ideas popped into your head, and if you don't know how to put them in reality to an actual research, our next speaker is Yannis Mitsidelis. I have actually met him in the animal house the first time in 2017, and he's a postdoctoral research fellow at the Gibson Lab at University of Edinburgh, and he's going to talk about animal models in endometriosis.
Thank you, Pira. I would like to thank the organizers for inviting me here to give a presentation on animal models of endometriosis. And in the next 15 minutes or so, I hope I can summarize the kind of current studies that are using animal models and the different experimental approaches that are used. So we know that endometriosis is a complex condition. That complexity is reflected in the etiology, the mechanisms underlying pathophysiology, symptoms, and comorbidities. And it's quite challenging to develop animal models that encompass all these different aspects of the condition. So there is no one model fits all. However, in recent years, despite some limitations, there have been leaps in the generation and refinement of animal models in endometriosis, and the aim is to get to the point where we can mimic the human condition and have high reproducibility and high fidelity models for translational research. Now, for the interest of time, I will focus on mouse models of endometriosis for this talk. The main differences between humans and mice that are of relevance to endometriosis research are that, first of all, mice don't spontaneously menstruate, and also they have a closed tube-ovarian junction, so they don't get endometriosis spontaneously. However, they have several advantages that render them ideal models for, like I said, replicating some of the aspects of the human condition. So they have small size, short estrous cycle and gestation. There are inbred strains that allow for smaller sample sizes. And there are species that's well annotated molecularly, and they allow for easy genetic manipulation. Now, if you're interested in the topic, I suggest this review by Catherine Burns, which was published last year. It's a fantastic review, and they essentially look at all the different models of endometriosis that have been used in research, specifically rodents, and they group them in different tables. Now, I don't expect you to read this, but what should stand out with a glance is that different groups have used vastly different approaches. So that's one of the problems with the animal models in endometriosis research, that there is little reproducibility between studies. Now, I will touch upon some considerations when using animal models in research, and specifically mice. So first of all, it's the type of tissue we want to use for induction of lesions. We can have either tissue of human or mouse origin. And then there's the mouse strains, and it has been shown that there are mouse strain specific effects in lesion formation. We have transgenic mouse lines that allow us to investigate the mechanisms behind endometriosis. There's the effect of hormones. So mice undergo estrous cycles, so their hormone levels change throughout the cycle. So if we want to eliminate this as an extra variable, we can overreactomize the animals to deplete endogenous hormones and then add hormones exogenously. Another consideration is the tissue used for the induction of lesions in mouse models. So we can use, and that's the most common approach, is mincing essentially whole uterine horns of donor animals and grafting them into recipients. However, this has some disadvantages, that because we have myometrium there, the lesions that form are mainly cystic. So that's another consideration. And finally, the lesions induction method, whether we use a kind of less invasive approach where we inject the tissue intraperitoneally, or a more invasive approach where we do surgery. Now, Rowan will present some key studies that I think use different approaches that will give you kind of a flavor of all the different approaches that are used. So the first thing that I want to talk about is the kind of human or mouse tissue that we use for in a mouse model of endometriosis. So if we use human tissue, we need to use appropriate mouse strains so that we don't get rejection of the tissue, and this can be either skid or nude mice. And the tissues can develop either somewhere in the peritoneal cavity or under the renal capsule, which is highly vascularized and allows the formation of lesions. Now, this study by Dodds et al. From 2017 investigated the role of mouse strain as well as estrous cycle on lesion formation. As you can see, this was a study with a large sample size. They used a syngeneic approach, and they used two different strains, C57 black 6 mice, as well as bulb Cs. And they induced lesions during either proestrus, when estrogen levels were high in the animals, or estrus, when estrogen levels had dropped. And lesions were harvested 3 weeks later.
Now, what they found was that the majority of lesions formed on the connective tissue on the pancreas, the stomach, the peritoneal wall, as well as the white fat close to the uterus and the ovaries. They showed that there was a higher incidence of lesions forming the more amount of donor tissue was introduced, and this dose response is really important when designing experiments using mouse models. They showed that C57 black 6 mice develop more lesions during proestrus where estrogen levels are high compared to estrus, and this effect was not seen with bulb Cs. And they also showed that C57 black 6 mice develop more dense type lesions in contrast to bulb Cs, which have more cystic-like lesions. And what this tells us is that we should always read papers with caution and see, keep in mind always that there are strain-specific differences when translating results from publications. The next thing I want to talk about is about the role of estrogen in endometriosis, and we know that endometriosis is an estrogen-dependent disorder. And Kenneth Korak's group looked at the effects of estrogens using mouse models of endometriosis. So to address the role of estrogen signaling in lesion formation, they used wild-type and ER-alpha knockout mice in a syngeneic model of endometriosis. Intact cycling mice were used as donors, and uterine tissue was minced and injected IP into recipients that were ovariectomized and treated with estradiol. And lesions were harvested at different time points to look at the effect of lesion formation. Now, what they showed was that during the early time points post-lesion induction, so up to 72 hours, we get attachment of the tissue in locations of the peritoneal cavity. This tissue starts to get vascularized, and this happens regardless of ER-alpha genotype, highlighting the fact that during these early stages of lesion attachment, estrogen might not be that important. They also assessed concentrations of different cytokines and chemokines in the peritoneal fluid of these endometriosis mice. And what they showed was that there was a significant increase in the levels of some of these chemokines, within the first 72 hours post-induction, regardless of the presence of estrogen. And after 3 weeks, these levels went back to those of sham animals. And what the author suggested, together with other supporting data, was that those initial kind of inflammatory immune-mediated effects might outweigh estrogen effects early during lesion formation. However, if we looked at 3 weeks after lesion induction, we see that the effects of estrogen become important. And in the group where we have an ER-alpha knockout donor tissue injected into wild-type recipient, we see that in this case, estrogen will not cause an increase in the number of lesions in these animals or in the weight of those lesions. The experiments that I described until now used donor tissue, which was essentially a uterine horn minced and grafted into recipients. However, this approach does not replicate retrograde menstruation, which is the most prevalent theory for the etiology of endometriosis. Now, mice do not spontaneously menstruate. However, we can use a mouse model of artificial menstruation as donor tissue, and this model was developed in Philippa Saunders' group in Edinburgh. And what we do is we take donor animals, we ovariectomize them to deplete endogenous steroid hormones, then give them a treatment regime of different steroids that mimic the normal menstrual cycle of humans, and then in the end withdraw progesterone, and this causes tissue breakdown and a menstrual-like tissue, which can then be injected into recipients. The lesions form in the peritoneal cavity in different locations, such as the peritoneal wall or the fat. And the lesions morphologically resemble those of humans with a defined stroma and epithelial compartment. And studies using this model have shown that macrophages within the lesions are derived from the utopic endometrium of the donors, but also from circulating large peritoneal macrophages and monocytes of the recipients. Now, for the majority of publications in the literature using mouse models of endometriosis, you'll see that the lesions tend to form on specific locations, most of the times randomly and very rarely on the ovary. And this is reflected in the number of publications that have used mouse models of ovarian endometriosis. So a Japanese group, I think it was last year, developed a novel mouse model of endometriosis, where what they did was, for the recipients, they exposed the ovaries they injected PBS into the ovarian bursa so that they could detect where the bursa was essentially, and then excised it and attached uterine tissue from donors on the ovary. And what they found was that lesions form on the ovary, and sometimes they infiltrate neighboring peritoneal wall or the intestines. And when they looked at different features of endometriosis, they discovered that these lesions exhibit fibrosis, as shown here by staining with Mason's trichrome, and fibrosis increases in a time-dependent manner throughout the experiment. And they also showed increased iron deposition and a subfertility phenotype with these animals that the author suggested came from increased oxidative stress that caused follicular growth in the ovary. So this is a model that could be refined and further validated to be used furthermore. Now, one of the limitations that we have with mouse models of endometriosis is that it's really challenging to monitor attachment of lesions and also their kind of establishment and growth. However, there are some non-invasive methods that include luminescence-based approaches or MRI-based approaches where we can monitor lesions longitudinally. And if you want to look up more about this, you can read this paper from Erin Greaves' group, where they used mice as donors that ubiquitously express luciferase, and then they formed lesions in recipients that are wild types. And when you give luciferin to these mice, they emit bioluminescence. And this bioluminescence is proportional to the amount of lesions. And we can locate exactly where the lesions are, and then we can use treatments. For example, in their experiments, they treated endometriosis mice with a GnRH antagonist cetrorelix, which caused a reduction in the signal and also in the lesions in these animals. Now, one of the main symptoms of endometriosis is endometriosis-associated infertility. However, it's quite challenging to generate mouse models to investigate that. And again, this is reflected by the number of publications where mouse models of endometriosis are used to assess fertility. This is one of the most recent papers where they used, again, a syngeneic mouse model of endometriosis, where the uterine horn of donors was cut in pieces and sutured on the mesentery of recipients. And the endometriosis mice were mated with male animals, and pregnancy was monitored throughout. And some of the main findings include that the only difference they saw was that there was a reduction in pregnancy rates in endometriosis animals compared to sham controls. However, other aspects of pregnancy, such as litter size, pup weights, and resorption rates, were not changed. And similar effects were replicated in other studies, for example, in this paper from Hugh Taylor's group, where they looked at the effect of lesion burden or the number of lesions per animal and how this impacted pregnancy, and they showed a similar effect in pregnancy rates. So animals with endometriosis exhibit reduced pregnancy rates. Now, the other main symptom of endometriosis is pain, and we heard from Katie how complex pain is to understand. So it's quite tricky as well to model in mouse models. Now, we know that there is no correlation between disease severity and pain symptoms in humans, so this makes it even more challenging to model this in mice. A few years ago, we were involved in an IMI Pain Care European Union consortium, and what we did was we conducted a literature review to look at mouse models in endometriosis and whether these reflected pain symptoms of patients as well as they had kind of translatable effects. And what we showed was that in a very small minority of papers, they actually look at pain, and even less so for non-evoked pain. So there is an unmet need for endometriosis animal models to replicate and assess both evoked and non-evoked pain. Now, again, I will give a publication as an example. In this paper, they used a syngeneic mouse model of endometriosis, and they looked at several evoked and non-evoked pain and behavioral changes. Importantly, they showed nerve fiber presence within the lesions, which shows us that there might be a possibility of these animals having some kind of hypersensitivity. So as their evoked pain measurement, they looked at vaginal distension. So this is a test that has been predominantly used in rat models of endometriosis. and it involves putting a balloon in either the vagina or in the case of colorectal distension, in the colon of the animals, and then applying different pressures and seeing responses, or as they call visceromotor responses to vaginal distension. So essentially, we're recording the electrical activity produced by abdominal muscle contractions in response to non-noxious and noxious distensions. And what they showed in this study was that animals with endometriosis show a significant increase in this visceromotor response, and this was seen both with non-noxious and noxious distensions, highlighting the fact that these animals exhibited vaginal hypersensitivity. As a non-evoked pain test, they used spontaneous voiding patterns to look at the possibility of an altered bladder function. So they recorded total uterine spots as well as the distribution of these spots for these animals. And once again, they showed that there was a disrupted voiding pattern following the development of endometriosis. Now, for the last part of the talk, I want to focus on this paper, which was published last year in Science Translational Medicine, looking at a connection between endometriosis, a bacterial infection, and fibroblast activation and differentiation. And I think this is an excellent example of a study that's using an animal model to back-translate human findings and also expand our understanding of the mechanisms that could be involved in the pathogenesis of the disease. Now, what the authors showed was that infiltration of a bacterial species, specifically Fusobacterium nucleatum, was significantly more frequent in patients with endometriosis compared to controls. And doing genetic profiling, they showed that there was a significant increase in a protein called transgellin in fibroblasts from patients with endometriosis compared to controls. And transgellin is a protein that's binding to actin cytoskeleton and is upregulated in activated myofibroblasts and is involved in fibrosis. They also showed that there was an increase in the infiltration of CD163 positive macrophages and in the expression of TGF-beta1 in the stroma of Fusobacterium positive patients. So with the animal model they used, they replicated these findings using donor bulb C mice, which were infected with the bacteria intravaginally, and lesions were induced in recipient animals by IP inoculation of endometrial tissue. So these in vivo experiments demonstrated that both the Fusobacterium infection and the amount of injected tissue, both of these are important parameters for the induction of endometriosis lesions. They also showed that endometrial stroma of bacterial-infected donor uteri and the endometriotic lesions exhibit an increased infiltration of macrophages, increased expression of TGF-beta1, as well as an increased number of transgellin-positive myofibroblasts. And then when they tried to test these using different bacterial species, they did not replicate these findings, meaning that this is specific for this bacterial species. They also showed that when you use antibiotics to which Fusobacterium nucleatum is sensitive, and you administer this orally for 5 days to mice with lesions, you get a reversal of the effect. So fibrosis is reduced, infiltration of macrophages stops, and you get a reduction in the weights of endometriotic lesions after 21 days after the initiation of the treatment, highlighting the fact that antibiotic treatment may facilitate treatment of endometriosis. It's one of the first studies to suggest a link between infection and endometriosis, so it's of great importance. So to conclude, there is no one model of endometriosis, and there are limitations with different models, so it's important when designing studies using most models of endometriosis to take these limitations into account. Fertility and pain, which are the two pivotal symptoms of endometriosis, are underrepresented in vivo preclinical endometriosis research. So there's more work that needs to be done, but for that to happen, we need first of all to understand what's happening in humans. There's incomplete recording of data and negative results, unfortunately, when it comes to animal models of endometriosis, so we need to do better at that and report all the findings. And finally, most models of endometriosis, despite their limitations, represent a powerful resource for translational research in endometriosis, but it is important to know which aspects of the disease we can model and not try and overinterpret data. Thank you.
Thank you, Yanni, for wonderful speech. And I know it could be it could sometimes be frustrating to develop a model, so it's good to have experienced people like you to ask rounds. We have time for one question, maybe.
Hi, doctor. Thank you very much for your presentation. I just want to ask you something about in vivo systemic circulation. We are confirming by using models in animals. But what do you think that if we have a chance to do it in vitro culture with the endometrium area locally and classify it in vitro? What do you think about that? Thank you.
Yes, that is a possibility, and there are models that have used in vitro modeling of lesions. The only problem is that you get a static system most of the times, and endometriosis has an immune component that is really important for the lesion persistence. And there are some in vitro models, but I'd say that it is very difficult to model endometriosis in vitro. I think that, my opinion is that we need to get more information from humans to start developing better models with better reproducibility. And because at the moment, like I say, different groups do kind of slightly different things. So in the end, it's very tricky to interpret results.
Thank you, Yanni.
Thank you.
Okay, so our next speaker is Fitnat Selzuki. It's a huge pleasure to ask Fitnat to come to the stage. Fitnat's an incredible person because she manages not only to practice clinically in obstetrics and gynecology in Istanbul, but is also studying towards a PhD in Oxford. And Fitnat's going to talk to us about mechanisms of infertility and endometriosis.
Thank you. First, before I start, I would like to thank the chairs for giving me the opportunity to speak to such an incredible audience. And I'm also a little stressed because I know among you are a lot of infertility specialists. And today I'm going to try to give an overview on the mechanisms of infertility in endometriosis. Clinically, an association between endometriosis and infertility is well established in literature. It has been also known since the ages of Hippocrates, because in his scripts he said that women with menstrual pain are less likely to conceive or not conceive at all. As you all very well know, endometriosis is seen in 10% of women of reproductive ages. However, when endometriosis comes into play, when infertility comes into play, these numbers change, so they increase. So among infertile women, 25 to 50% are endometriosis patients, and 30 to 50% of women with endometriosis are infertile. When you look at the fecundity rates of normal couples, it's reported to be 15 to 20%, but this rate is very low in women with untreated endometriosis, which goes down to 2 to 10%. It is also known that endometriosis affects the probability of becoming pregnant. So in 55% of women who 55% pregnancy rate is 55% in women with unexplained infertility, but this rate is lower when the infertility is caused by endometriosis. As you all are very well aware, endometriosis is a multifactorial systemic disease. And there are many factors playing in the pathophysiology, as this morning, Krina Zondervan gave us the overview of the pathophysiological mechanisms. It is important to understand these mechanisms, of course, with what we know today, which might change in the future, to be able to interpret why endometriosis causes infertility. However, as I said, the exact mechanisms are still unknown, and there is room for more research in this field. It is postulated that the chronic inflammatory peritoneal environment, the progesterone dysregulation, and the proestrogenic environment, oxidative stress, and anatomical factors such as fibrosis, adhesions, and anatomical distortions, and endometriosis-related pain cause infertility. These multifactorial pathophysiological effects play on different steps of the process that leads to infertility. These are ovarian reserve, gamete and embryo. Endometriosis have an effect on ovulation, embryo transport, endometrial receptivity, and the uterus itself. So any dysfunctions in any of these steps can lead to infertility. First, I will start with pain, because in order to achieve conception, feasibility of sexual intercourse is very important. As you all know, and as Katie talked into detail about endometriosis and pain, endometriosis is associated with dyspareunia, which can lead to avoidance of sexual activity. Additionally, chronic and non-menstrual pelvic pain, which is also associated with endometriosis, is also known to reduce desire, frequency, and sexual intercourse, arousal, and orgasm, which all lead to avoidance of sexual intercourse. In a study conducted by Kate Wall et al., which involved 300 women with surgically confirmed endometriosis, the women were asked to fill out a endometriosis health profile EHP 30 questionnaire along with a more extant baseline questionnaire. The study, one of the aspects they looked in the study and evaluated was the superficial and deep dyspareunia and how these women perceived that this affected their fertility. And especially women with superficial dyspareunia reported that they are afraid that they might be infertile significantly more than the controls, which shows that, or at least the authors interpreted that, these women, because of this pain, are more likely to avoid sexual intercourse. Following pain, mechanical factors are also important in causing infertility. As you all know that endometriosis and inflammation causes pelvic adhesions, anatomical distortion, and these are mostly because of inflammation, fibrosis, and the surgeries which these patients go through. These anatomical problems and the mechanical factors impair ovulation, tubal pickup of ovum and transport of ovum, and block the transfer of sperm through the fallopian tube, leading to compromised fertilization and compromised transport and implantation of the embryo. When we take a look at the ovaries, ovarian reserve is one of the main prognostic factors regarding fertility. It is, as you already know, defined by the supply of non-growing, unrecruited primordial follicles. The pathophysiological effect of endometriosis in ovarian reserve is unknown, but evidence shows that the presence of endometrioma has an adverse effect on the ovarian function. Endometriotic cystic fluid contains high concentrations of free iron, which mediates the production of reactive oxygen species. These oxygen species are diffusible in the cellular compartments that can go to the adjacent healthy tissue and affect the healthy ovarian cortex surrounding the endometrioma. Proteolytic enzymes, inflammatory, and adhesion molecules are also present in the endometriotic cysts. These toxic endometriotic cystic content is known to induce oxidative stress, damage healthy tissue, which causes loss of corticostroma because of fibrosis, impair vascularization, reduce follicular maturation, which all can lead to atresia of the ovary. Reduced follicular number and activity compared to other benign cysts has also been reported in histopathological specimens that have been retrieved from patients with endometrioma. Despite concerns of specificity in clinical practice, we still use serum anti-Müllerian hormone levels and antral follicle count as the most while evaluating the ovarian function. As demonstrated here by Kasapoğlu et al., they show that anti-Müllerian hormone levels decrease significantly in patients with endometriomas. This has been shown in other studies in literature as well, and the studies yield similar results, regardless of whether the endometrioma is unilateral or bilateral, which indicates that this is a systematic disease. Focal inflammation in the ovarian cortex of the ovaries with endometriomas caused by the cystic content leads to enhanced follicular recruitment, and as a result of the loss of the cortex-specific stroma to atresia. These leads to excessive primordial follicular activation and burnout of the follicular reservoir. Primordial follicular activation is an irreversible process. Therefore, it leads to follicular depletion. It has been shown that it is known that PI3K, PTEN, ACT, and FOXO3 pathways are primordial follicle activation regulators. Takeuchi et al. In their study showed that the number of primordial follicles was diminished in endometrioma patients, whereas the primary, secondary, antral, and growing follicle numbers were increased in these patients. And these all were mediated by these 4 pathways. Another study by Dionisio et al., where they evaluated the expression of pro-apoptotic markers in the context of endometriosis, they demonstrated higher levels of expression of pro-apoptotic markers such as total phosphorylated P53, caspase 8, and NF-kappa B. In the cortical tissue surrounding the endometrioma, these were all compared to controls, and this they interpreted indicates that the ovarian cortex is sensitized for apoptosis and inflammation. There is an ongoing debate on whether surgery has a negative effect or impact on residual ovarian function. Several histological studies in the literature confirm that cystectomy is generally associated with inadvertent removal of healthy ovarian tissue and the primordial follicles that are adjacent to the cysts. Unlike other benign cysts, endometrioma is not surrounded by a well-defined capsule, and technical difficulties may arise due to the absence of this clear cleavage where you see in benign cysts. Therefore, a surgical expertise is very important in endometrioma excision. Surgical damage may also cause depletion of ovarian function due to local inflammation and vascular compromise, which leads to bleeding, tearing, and the overuse of electrocoagulation, which are, again, can be solved by the surgical expertise. The SAFE study, however, showed that 50% of women following endometrioma excision surgery had spontaneous pregnancies within 6 to 12 months after surgery, which shows that still surgery is not detrimental to ovarian function where it's done by experts who know what they're doing actually. So as recommended by Professors Becker and Usta today, surgery can be used in the management of endometriomas depending on what you're treating the patient for. Clinical data concerning spontaneous ovulation rate in women with endometriosis is poor and controversial. prolactin levels are usually high, preventing luteinizing hormone pulsatility and therefore the ovulation. Luteinized unruptured follicle syndrome is another potential cause of ovulation failure. In this condition, the dominant follicle undergoes luteinization but fails to rupture. Additionally, altered patterns of estrogen and progesterone secretion, demonstrated by Dr. Gibson today, can lead to abnormal luteal phase and compromise ovulation in these women. Endometriosis also affects fallopian tube and therefore embryo transport. Inflammatory environment impairs tubal function and decreases tubal motility. Disordered myometrial contractions can impair gamete transport and embryo implantation. The endometriotic pro-inflammatory peritoneal fluid microenvironment may also affect sperm function by inducing sperm DNA fragmentation, disrupting sperm membrane permeability or integrity, reducing sperm mobility, impairing sperm oocyte interaction, and causing abnormal sperm or chromosome reaction. There are conflicting opinions in literature regarding embryo quality and endometriosis. A recent meta-analysis, including 22 studies, analyzed embryo quality, cleavage rate, and embryo formation rate, and they concluded that endometriosis does not affect embryo morphology. Additionally, when disease stages are compared, advanced disease is associated with poor implantation and poor clinical pregnancy rates. However, whether this is due to only because of adverse effects on embryo quality and morphology is not clear. Also, other meta-analyses in literature compared IVF outcomes, and these are still, although very controversial, show that endometriosis also affects IVF outcomes poorly. We all know that implantation rates are diminished in women with endometriosis. This could be due to reduced endometrial receptivity or decidualization capacity. The utopic endometrium of women with endometriosis is affected, which probably is the underlying reason for implantation failure. However, there are still many unknowns in the underlying mechanism of how endometriosis affects utopic endometrium. There are several pathways that are in play, and Wnt7a upregulation, expression of Wnt7a upregulation of expression of Wnt7a and 17 other genes in the Wingless pathway is one of them. It's known that Wnt7a plays an important role in estrogen-mediated uterine growth and implantation in mice. Increased expression of ectopic Wnt7a disrupts the normal epithelial stromal polarity required for the normal fertility and implantation. This aberrant activation or dysregulation of the Wnt pathway disrupts endometrial development during the implantation window. Another gene that is known to play a role is the Homebox A10 gene and its aberrant expression. This gene is involved in the embryogenesis of uterus and subsequently in endometrial regeneration in each menstrual cycle. Expression of gene is necessary for endometrial receptivity. It has been shown that mice with targeted disruption of this gene show complete loss of endometrial receptivity. Also, women with lower expression of HOXA10 have lower implantation rates. In women, there is a cyclical endometrial expression of this gene, which peaks during the window of implantation. As you can see in the first line, the expression is higher and then it's reduced. However, this cyclical expression is diminished or non-existent in women with endometriosis, which causes problems with decidualization and endometrial receptivity. Integrins are cell adhesion molecules expressed in the endometrium during the receptive window and involved in implantation. Beta-3 integrin subunit is a direct downstream target of both HOX810 gene and ESR1 gene that Doug mentioned this morning, and its aberrant expression have been described in the endometrium of women with endometriosis. Additionally, it has been shown that ART is less effective in patients with lower expression levels of B3 in ectopic endometrium. Transcriptional factors involved in progesterone signaling are also reduced in endometriosis patients. IGF-BP1, GATA2, FOXO1, ARID1A, NOTCH1, and WNT4 are among these transcriptional factors. As an example, it has been shown that when GATA2 expression is disrupted, there is a dysregulation of the decidualization. And their diminished expression or dysregulation causes diminished presence of decidualization markers. Aromatase has also been studied extensively in endometriosis, which was also one of Doug Gibson's topics. It has been shown that abnormal levels of aromatase are present in both ectopic and utopic endometrium, where it normally not present, resulting in increased production of estradiol, which also, as we know from the pathophysiology, causes a not suitable environment for implantation. Progesterone induces endometrial decidualization during the luteal phase, so it plays an essential role in normal pregnancy. Promoter region of the progesterone receptor isoform B gene is hypermethylated in endometriosis, which causes the dysregulation and leads to the progesterone resistance. All of these lead to implantation failure and also unopposed estrogen state, which makes the microenvironment of the endometrium not suitable for implantation. Lastly, when infertility is evaluated in the concept of adenomyosis, several pathogenic hypotheses have been postulated, such as junctional zone thickness and perturbed uterine peristalsis that can alter the uterotubal transport. Additionally, the utopic endometrial microenvironment in adenomyosis patients differs from the endometrium of unaffected women. However, data on adenomyosis research has several limitations in the existing literature when we compare adenomyosis to controls or even to endometriosis. One of the most important problems with the literature and the studies is that endometriosis and adenomyosis coexist, and most of the studies do not exclude endometriosis from their cohort. So at the end of the day, we do not know if the infertility is caused solely by adenomyosis or if it's a joint mixture and effect of endometriosis and adenomyosis together. There is also no consensus regarding diagnostic features of adenomyosis using imaging in terms of research. Therefore, this makes observational studies and the interpretation of these observational studies challenging. And on a final note, we know that adenomyosis frequently coexists with other gynecological conditions, which are potential confounders to cause infertility, such as uterine leiomyomas. I would like to thank you for listening to me and for your attention.
Thanks very much. That was a great overview, Fitnat. We're running slightly behind time, but you kept so well to time. I think we can maybe have two questions from the floor. Does anyone have a question?
I also want to say I left ovarian quality out because I know that Professor Semra Kahraman is going to talk about it later on, so that's why I didn't go into that topic.
Great. Can I just ask you, you mentioned about prolactin being raised in endometriosis, and I think that's been shown in more later stage disease. What are your thoughts about dopamine agonists for treating infertility in patients with endometriosis?
Yes, it is in the later stages. However, of course, my knowledge is very limited in this field. But as far as I know, the studies they do not differ on the perspective that they do not make much of a difference between the early and later stage. Even if the study is making that difference, the data is very limited. That's why I'm not able to, or I wasn't able to find that much of an information from what I've done for this presentation. But maybe if someone knows in the audience, as I said, there are many infertility experts, they might enlighten me as well.
Engin.
The first question for hyperprolactinemia in the endometriosis patients, we have this data, but unfortunately, they tried the treatment for hyperprolactinemia, but there is no answer for the fertility and other problems. It's interesting, but we know the 20-30% of patients has hyperprolactinemia in these patients. And the comment, another one, we know from in vitro and animal models, a little bit decreases implantation rate in endometriosis patient, but in a clinical manner, this is the vice versa. And we know from the IVF data and the donor cycles data, there is no difference between the healthy patients and endometriosis patients about implantation rate, the same rate, both of them.
Great. Well, if there are no more questions, just want to thank Fitnat again. I think we're running a little bit behind, so maybe should we have a break for 10 minutes? Come back in 10 minutes? Okay, thank you.
Hey, hey. Hey, hey. Deneme 1, 2, 3, 4, 5, 6. Pazartesi, Salı, Çarşamba, Perşembe, Cuma. Hey, hey. 1, 2, 3, 4, 5, 6. Tamam.
Ses deneme 1, 2, 3 ses prova.
Tamam.
Şu muydu hocam?
Evet. Yok, onu büyük yapıyoruz. Demin konuştuk bunu.
Bunu yapsın.
Yapmıştı arkadaş aslında ama.
Kapatmadı ya hocam.
Hmm. Bunu büyük ekran yapacağım.
Tabii hocam. Teşekkür ederim.
Tamam. Şöyle değiştireceğim. Değil mi? Oradan da şöyle yapacağım. Bu alttaki şey değil mi?
Evet hocam. Biz sonraki botumuz zoom olduğu için hocam.
Bunu büyüğe alabiliriz. Tamam. Şöyle yapabiliyoruz değil mi?
Öyle yapabilirsiniz ya da buradaki yön tuşlarıyla da yapabilirsiniz.
Yok, şey için. Şurayla gitmeyi düşünüyorum da hani bir şey gösterirken şu zor mu oluyor bunu kullanmak? Onu kullanmak istemiyorum. Çünkü orada terse dönüyorum falan.
Merhabalar,
Merhabalar,
nasılsınız? Hoş
teşekkür
geldiniz.
ederim. Hoş bulduk. Siz nasılsınız?
Ben de gayet iyiyim, sağ olun.
Güzel bir toplantı hazırlamışsınız.
Sizler de güzel.
Öğleden sonrasına katılabildik ama
Sizler de
güzel
güzel.
konular, güzel gerçekten. Peki, teşekkür ederim.
Videonuz falan var mıydı?
Hayır.
Tamam.
Şöyle bir bakabilir miyim?
Tabii hocam.
Tekrar başa bırakayım.
Bu
Kaçın.
görünecek mi buradan? Şu şey görmeyebilir miyim?
Yok hocam.
Evet, bravo. Harika. Tamam. Süper.
Size video ses var mı hocam?
Yok, hayır. Benim sesim var. Harika. Başa dönüp şey yapabiliriz. Sağ olun.
Tamam, oradan mesela kontrolü.
Hazır şöyle git işte. Ne bileyim. Gel şuna buraya git.
Şu gözükmemiş lazım. Bu masaüstü gözükmemiş lazım. Anladın mı? Direkt uzakta bırakıyoruz. Anladın mı?
Gel yapalım şunu, hazır olsun. Kadın gelecek ve sonra niye
Hoca
öpecek?
dedi olmaz. Hoca dedi ya.
Kendisi için söyledi o ya. Kendi saatine gelmesini istemedi.
Ben ayarlayacağım orayı.
Gel yap şunu, öyle git ya.
Oğlum ben oradan ayarlıyorum zaten.
Gel buradan yap buradan. Şu saatini yok et, öyle git.
Şimdi oluyor.
Biliyorum. Yok et, öyle git şimdi.
Tamam, ayarladım, böyle kalsın.
Unutma ama.
afternoon. We will be moving to a very interesting subject, which is infertility, endometriosis, and adenomyosis. And the first part of our session will be devoted to some lectures. After each lecture, we will take 2 questions, but we will have a very interesting debate on deep endometriosis and related infertility at the end of the session. The first, Professor Semra Kahraman. No need to say anything about her. She is one of the leaders in infertility in Turkey. So she will be talking on endometriosis and adenomyosis, the effect on oocyte quality.
Thank you, Mr. Chairman. It's a great pleasure to be here, and I would like to extend my thanks to the organizing committee, and in particular, Dr. Engin Oral, for inviting me today to speak on the endometriosis and adenomyosis, the effect on oocyte quality. Well, as you all know, endometriosis is an estrogen-dependent chronic inflammatory disease which affects women in their reproductive period, causing infertility and pelvic pain. And as a fertility specialist, actually, approximately 30% of the infertile patients that I see have endometriosis. So although it is encountered so commonly, the mechanisms by which endometriosis causes infertility remains poorly understood. If we look at the, I can't change the slide. No, it's not moving. Is there anybody to help me that it's not working? Okay. Şimdi başladı. Teşekkür ediyorum. So if we look at the endometriosis-related infertility, well, we can see that endometriosis can distort pelvic anatomy and may cause infertility. It can damage fallopian tubes, thus creating pelvic inflammatory environment, and increased cytokines impairs tubal function and decreases tubal motility. And it can lead to disturbed folliculogenesis and ovulation and decreased ovarian reserve. But what about the altered oocyte quality, which is my subject today? Does the presence of endometriosis adversely affect oocyte quality? This is one of the questions, and actually there has been much debate as to whether IVF outcomes in women with endometriosis are affected to altered oocyte quality. And there is conflicting evidence in the literature about this question. When we look at the literature, this review here by Sanya Latif and Ertan Sarıdoğan just summarizes the relationship between the endometriosis, oocyte, and embryo quality. And here you can see the summary of the findings from studies examining the impact of endometriosis on oocyte and embryo quality. So according to these studies, although there is evidence that endometriosis adversely affects oocyte quality, however, when we look at these studies in details, the number of the patients examined in these studies is very limited. 28 women, 12 women, 72 women, and furthermore, some of these studies are in vitro maturation studies where the oocyte morphology cannot be accurately described. Well, although there is some studies like you see in here, that suggesting endometriosis decreases oocyte quality. However, most studies have just suggested that endometriosis does not alter oocyte quality and does not alter embryo quality. So as you see here, for example, one study from Robin et al. Just shows that oocyte quality is unaffected. They found that the oocyte morphology is normal. However, the lower number of top quality embryos and lower cumulative clinical pregnancy rate are both attributed to lower oocyte yield. And some other studies found equivalent fertilization rates, and the others found no effect on embryo quality. So as you see, there are studies suggesting that there is evidence that endometriosis alters oocyte quality. However, on the other hand, the other studies does not show any effect on oocyte and embryo quality. And this study, this review by Korakan et al., just listed a number of hypotheses just to show that how endometriosis affects oocyte quality. And one of the hypotheses is that altered steroidogenesis and folliculogenesis may affect oocyte quality. Higher oxidative stress, particularly in the ovarian cortex, may cause decreased oocyte quality. Reactive oxygen species has been suggested to reduce oocyte quality by just promoting the chromosomal instability and also meiotic abnormalities. And some other hypotheses just show that altered cell cycle progression may cause lower oocyte quality. Also, inflammation and angiogenesis in the follicular environment, and this exposes oocytes to a hostile environment, inflammatory environment. So these are all hypotheses. So in order to evaluate the oocyte quality in endometriosis cases, and in order to answer the question, does the presence of ovarian endometrioma may negatively affect oocyte quality, we need to describe the oocyte quality. But how is oocyte quality measured for a study? Which parameters should be considered to describe oocyte morphology? So, as you know, oocyte quality is not only reflected by oocyte morphology, but also the ability of an oocyte to complete the maturation and fertilization are the main characteristics to show the oocyte quality. So, how is abnormal oocyte morphology described? In the study that we performed in our center, we just evaluated the heterogeneity of cytoplasm because cytoplasmic granulation, either central or homogeneous granulation, may reflect the oocyte abnormality, but also indirectly reflect the maturity of the oocyte. Presence of vacuole or multiple vacuoles, presence of smooth endoplasmic reticulum, abnormal first polar body morphology, these are all abnormal oocyte morphology description, expanded perivitellin space size, zona pellucida defects, thick, thin, multi-layered or oval zona pellucida, and oocyte shape abnormalities. So these abnormalities may be seen either in isolation, I mean, one oocyte individually may have one of these oocyte morphological abnormality, or in combination, 2, 3, or more oocyte morphological defect can be seen in the individual oocyte. So, well, I would like to show some examples that we routinely record in our database program for the oocyte morphological description. Cytoplasmic granulation, as you see here, this is a big cytoplasmic granulation, presence of smooth endoplasmic reticulum, multiple vacuoles, zona pellucida defects, expanded perivitellin space, and abnormal first polar body morphology. And furthermore, refractile body, ovoid-shaped oocyte, fragmented oocyte, and multiple oocyte abnormalities, as you see in here, the flat polar body, very narrow perivitellin space, and multi-layered zona, or as you see in here, the homogeneous oocyte central granulation and refractile body. So again, does the presence of endometriosis adversely affect oocyte quality? We looked for an answer for this question, and we did a study at Istanbul Memorial Hospital. And in this study, the cases were included between the years of 2011, 2023. The data were collected from our clinic-specific AURA database. And actually for this presentation, we evaluated more than 29,000 cycles, 4,600 cycles, women with endometriosis, with more than 27,000 oocytes, and more than 24,000 cycles, women without endometriosis, with approximately 180,000 oocytes, which were morphologically evaluated, either for isolated abnormality or multiple abnormality. And a total of more than 200,000 oocytes were evaluated morphologically. So for this large data, we did multiple statistical analysis, Shapiro-Wilk test, Kolmogorov-Smirnov test for the suitabilities of the variables for normal distribution. Mann-Whitney-U test was used to compare the groups, and Pearson chi-square test was used for the comparison of categorical variables between groups. And when the sample sizes were large and the p-value was statistically significant, we use another statistical method, which is effect size. And for the effect size, for the non-parametric tests, we use Cliff's delta effect size. And for the categorical data, we use PHI effect size. And you see the interpretation in here. It could be negligible, small, medium, large, or for the PHI effect, could be negligible, weak, moderate, relatively strong, strong, very strong. So as you see in here, the characteristics of patients with or without endometriosis is clearly seen in here. There was no significant difference between the female age and the body mass index in patients with endometriosis and non-endometriosis. However, the number of previous unsuccessful cycles, duration of infertility, and the AMH levels were all found significantly different than the non-endometriosis group. So a higher number of previous cycles, a longer duration of infertility, and lower AMH levels were found in endometriosis cases, which is actually expected results because most of these patients had previous surgeries, either unilateral or bilateral, or many of them had big, large endometriomas in the ovaries, either single or multiple endometriomas. When we look at the cycle characteristics of these patients with or without endometriosis, actually the total gonadotropin dosage that used for endometriosis patients and duration of ovarian stimulation, the level of estradiol on the trigger day, the number of aspirated metaphase 2 and fertilized oocytes were all found statistically significantly different in endometriosis group. A higher dosage of total gonadotropin were used. The duration of ovarian stimulation were longer. We had a lower estradiol level on the day of trigger, and the number of the aspirated metaphase 2 and fertilized oocytes were all found significantly lower than the non-endometriosis group. And when we further look at the cycle characteristics of cases with or without endometriosis, we looked at the rate of oocyte maturation, oocyte fertilization, blastulation, and the rate of top and good quality usable blastocyst. Actually, these statistics were interesting because in the endometriosis group, the rate of maturation, fertilization, blastulation, and usable blastocysts were found significantly higher according to the p-value than the non-endometriosis patients, suggesting that once the oocytes are retrieved, then the no difference or significantly higher difference in endometriosis cases with all these parameters. However, when we look at the effect size for this comparison, we saw that looking at the effect size, there was no significant or there was no large difference in between these endometriosis and non-endometriosis group. So although p-value showed that these values were significantly different than the other group, but effect size, looking at effect size, all interpretation just showed that this difference is negligible. So the main subject, the oocyte morphological abnormalities evaluated in isolation in patients with or without endometriosis, here show that although there is a significant difference in cytoplasmic granulation, large perivitellin space, zona pellucida abnormalities, and polar body defects. However, again, when we look at the effect size, then this difference was small and there was no, this difference was negligible in both groups. So in terms of isolated oocyte morphological defect, there was no difference between the endometriosis and non-endometriosis group. When we look at the multiple oocyte morphological abnormalities in patients with or without endometriosis, again, for the oocytes, individual oocytes, which have 2 or more oocyte morphological defects, we did not see any significant difference in terms of oocyte morphological abnormality in both groups according to the effect size. So what about the situation in adenomyosis cases? Well, oocyte morphological characteristics between adenomyosis and non-adenomyosis cycles were further evaluated. And again, in the morphological abnormality, in none of the morphological abnormality between the cases with adenomyosis and without adenomyosis, there was no statistically significant difference in terms of these severe morphological abnormality. And these cases are severe adenomyosis cases. When, again, multiple oocyte morphological abnormalities in patients with or without adenomyosis were evaluated, we did not find any significant effect according to the p-value and according to the effect size. What about the pregnancy outcomes of the patients with or without endometriosis? Does endometriosis alters not only the oocyte quality, but also the embryo quality? For these results, well, we use the frozen taught single blastocyst transfer cycles. In endometriosis cases, although the biochemical pregnancy, clinical pregnancy loss, and total pregnancy loss were found significantly different in both groups. However, again, the effect size showed no difference in terms of these parameters. Furthermore, the live birth rate were nearly exactly the same in both groups with endometriosis and non-endometriosis, indicating that embryo development is not affected by the presence of endometriosis. So when we look at the same results, pregnancy results in patients with or without adenomyosis, the situation was different than the isolated endometriosis cases. Well, all these parameters, including biochemical pregnancy rate, clinical pregnancy loss rate, total pregnancy loss rate, and live birth rate, were all significantly different than the patients without endometriosis. All live birth rate was significantly lower. The pregnancy loss was significantly higher in this group. So in the adenomyosis cases, the results were completely different than the other cases with endometriosis. So as conclusion, in endometriosis cases, a significantly higher number of previous cycles, longer duration of infertility, and lower AMH levels were found. In endometriosis cases, there were some significant differences. Higher total gonadotropin dosage were used in these patients. Longer duration of ovarian stimulation was found. Lower estradiol level on trigger day. Lower number of aspirated metaphase 2 and fertilized oocytes were found in endometriosis cases compared to the non-endometriosis cases. No significant differences were found between the endometriosis and non-endometriosis groups when oocyte morphological abnormalities, either in isolation or in combination, when evaluated using effect size. No statistically significant differences were found in oocyte morphological abnormalities, either in isolation or in combination between cases with and without adenomyosis. Once an oocyte was fertilized, embryo development was not affected by the presence of endometriosis. No significant difference was found in live birth rates. And for the adenomyosis, the presence of adenomyosis did not significantly affect the oocyte quality. However, ongoing pregnancy and live birth rates were found significantly lower than patients without adenomyosis. Furthermore, a significantly higher total pregnancy loss rate was found than those cases without adenomyosis. The presence of adenomyosis increases the risk of pregnancy losses and decreases live birth rate rather than oocyte quality and the number of retrieved oocytes. So if we come back to the original question, which was my topic title today, does the presence of endometriosis adversely affect oocyte quality? Endometriosis does not adversely affect oocyte quality. Thank you very much for your attention.
Thank you very much, Professor Karaman. I think congratulations. No, we will take two questions now. So it's one of the biggest data in the world. So it is, congratulations. So
it's just because of recording all the
but this is
all the morphological abnormalities over the years.
Okay. Ümit sen miydin?
Yes, thank you. I'm very much impressed as usual. Big data. I was just wondering, we know that there is coexistence of adenomyosis and endometriosis. I was just wondering whether if you excluded those coexistent cases, or did you look at just purely adenomyosis, or did you look at adenomyosis include all cases?
Well, as you know, it is very difficult to just exclude the adenomyosis cases because in most of the cases, endometriosis, ovarian endometrioma, pelvic adhesions, tubal factors, and ovarian and uterine adenomyosis are together. So these are the data that from the endometriosis group, we just evaluated the severe adenomyosis cases. So we did not really pick up the isolated endometriosis, uterine adenomyosis cases, as it is not possible every time, as you know, with the ultrasonography or with the other techniques. So these are from the endometriosis cases.
There is no question, but I think that it is a really wonderful data, but we are just missing something, because when we are just saying that endometriosis is not a one disease. So I think we have to go deeper a little bit more because this morning we talk about the endometriosis, minimal, mild, ovarian. So I think we have to, or you have to look for it because, you know, especially this severe and ovarian endometriosis can affect. Maybe it is going to be a little bit different in
But
the
I would like
quality. Maybe you
Yeah.
could do it.
I would like to explain that in order to diagnose endometriosis, severe endometriosis, you need the ultrasound findings, you need some MRI findings. Some of these patients had already laparoscopy, or some of them had multiple surgery. So these are not actually the minimal endometriosis cases, because how can you describe for the minimal endometriosis by ultrasounds? You can have the history only, but you can't rely on the history alone. So, of course, these are severe endometriosis cases
Yes.
that I presented
This
in here.
is okay. Thank you very much. We understood that adenomyosis cannot
Does not affect endometriosis, and adenomyosis does not affect the oocyte quality. This is what we found
Yes,
in
it
our
shows
data.
like this. Okay, we thought before it affects, later on no effect, now no effect. It's very good because we are just producing the embryos, so we could have the ability to detect, select the embryo in the laboratory to when we are just doing one blastocyst transfer.
So this is the reason that I
Yeah.
showed the blastulation rate, top and good quality
Yeah.
embryos also.
Thank you very much because I look for everything. It's a very fresh data. Thank you very much, Semra.
Thank you. Thank you.
See you.
And now, next speaker is Professor Dr. Engin Oral. Professor Oral is the founding president of the Turkish Endometriosis and Adenomyosis Society. And now, Professor Oral is talking about EMH and endometriosis management. And the stage is yours. Thank you.
Good afternoon, dear chairmen. And you just listened to one of the most experienced and scientific authors about ART in Turkey, in Europe, even in world, Professor Semra Kahraman. I will discuss the AMH and endometriosis management. Just as one of my case, she is 33 years old, just married 1 year. She has also pain. And when I come to our clinic, on the right side, 10 centimeter endometrioma, left side, no pathology. There is no other visible pathology. AMH level 0.6 in a good laboratory. And we discussed the patient and the IVF or surgery. I favor the IVF, but they choose the surgery instead of the IVF. And I did right cystectomy this patient, and the pathology was endometriosis. And 3 months later, she has spontaneous pregnancy. I did cesarean section. And after 2 years, there was no any news. 2 years later, she came to my office and she said, I am pregnant. I look at the sonography, 12 weeks of pregnancy, spontaneous. And 1 year later, I did another cesarean section. And the AMH levels, it was 0.6 before the first pregnancy. What is the role of AMH in endometriosis? I believe in these 5 manners is important. Follow up the patient with conservative, if you choose the conservative approach, or decide of fertility preservation, and decide of after the surgery to management of the patients, management of IVF cycle before or after IVF cycle, or maybe to discuss the patient prediction of the menopause. Should we prescribe AMH to every patient present with endometriosis? I believe the answer is yes, for academic issues, for legal issues, or for the most important patient management. Let's speak about a little bit physiology about the ovarian reserve. What does it mean? As you know, reproductive potential and oocyte function both qualitative and quantitative in this patient. And we believe that endometriomas can affect ovarian reserve in two ways. The first one, impairing circulation because of the compression of the cyst and the inflammatory settings. What we have ovarian reserve tests in our practice, we have 4 tests: antral follicle count, FSH, we don't use anymore too much, AMH, maybe if you tried the IVF cycle, you can see the number of the oocyte. This one is the expensive one if you compare with the other ones. As I say, you can use three of them for the ovarian reserve, FSH for 10, the limit, and the AMH 0.2 to 0.7, antral follicle count 3 to 10. But as you see, FSH is a limited reliability, and the other ones, good one. And 5 years later, they say only you can use the AMH and antral follicle count. This is the simple, sensitive, specific measure for ovarian reserve. But before the AMH issue, we have to remember it is not so easy to take and to evaluate it, because as you see, from the follicular AMH to do measured blood AMH in the clinical way, and a lot of variant affect the AMH levels. That's very, very important, and to take the AMH in the right time, right way, and right laboratory. What is the normal one? This is the question for us, not for endometriosis, for our fertility issue. And it looks like peak AMH is coming from the year 2011. There is no new data, unfortunately, and the peak AMH levels around 24 years, as you see. What is the normal, abnormal one? It discussed for the different age, but without age comparison, I believe below 1 nanogram per ml, that means the decreased fertility. How about the nomogram for the antral follicle count? Unfortunately, we have no data. This data is coming from our friends from the Hacettepe from Turkey. They look at the population, general population, fertile population. In the early 20s, around 14, early 30s, around 10. After the 40, around 7. The number of the patients are not so high. By just give some clues about what is the normal for antral follicle count. Let's discuss the endometriosis patient, as Dr. Kahraman said, in huge data from the ART department. This is data from START data, and more than 40,000. This is the last data they just published 2 weeks ago. And they say the reason for the ART is, you see, endometriosis is just 6%. How about the diminished ovarian reserve? 28%. In the 13 years ago, this was the same. Endometriosis rate 4%, diminished ovarian reserve, but it was around 8 to 9%. That means this is our dilemma for the fertility patients. How about the result? This is again the START data. If you say just endometriosis patient, more than 10,000 ART cycles, looks like the same with the other ones. I mean the male factor and tubal factor. But if you classify patient not endometriosis, diminished ovarian reserve, as you see, all of the live birth rate are decreased until the age 40. The problem, how can we evaluate in the management of the patients? This is just published from we have a lot of British author in here. This is the British Society practice recommendation for endometriosis IVF patients. As you see, there is no ovarian reserve in algorithm. Ne oldu?
Yasin çağırır mısın?
It was the most exciting one. Maybe British sabotage?
Çıkarıp taktın mı?
We need more time. Okay, man.
Thinks it happened to the president. Anyway, so think about that. What is your, idea if we should measure the IMH for the endometriosis patients? Is there anybody say that we want to do it? I'm just asking to you, to the audience. Who is going to make the ovarian reserve testing for endometriosis cases? For everybody, okay? Nobody. Who is doing it? Nobody. So you are not sleeping at all, right? No? Okay. What do you think? Bülent. Because the idea was just the AMH is not suggested, huh? So we are just okay. Anyway.
Thank you.
So,
And if, according to British Society Policy and Practice, they didn't mention about the ovarian reserve for the management of the IVF patient in endometriosis. The big question is in 2 points. And ovarian endometrioma itself on the serum AMH level or surgery, which one is harmful? I believe the surgery is harmful with itself. The question is, we choose the conservative approach, especially in our practice. What is the recommended management, young woman with an intact endometrioma, design future fertility? And if we say the no surgery, maybe she has the risk for the decreased ovarian reserve. And how about the superficial endometriosis? Just one slide. Without endometrioma, there is no any effect for the ovarian reserve, either AMH or anti-follicular count is similar, superficial or not. There are a lot of studies about discuss the without surgery to effect of the endometrioma. According to this Korea study, they say there is no difference between the group endometrioma versus control group, except the stage 4 patients, a little bit decreased ovarian reserve. Another one, again from the Europe, and they say unilateral ovarian endometrioma has no significant effect, but she has the bilateral endometrioma, maybe decreased ovarian reserve. Another study from Turkey, our friend from the Bursa, they called the error study. They look at the decline of ovarian reserve in a 6 months, and they say reduction in AMH in this patients group if you compare with the healthier one. Another study from the USA, they look at what is the effect of the endometriosis on AMH level, and they say endometriosis patient has low level AMH with the healthy ones. Another study from, again, Korea, they look at the normal patients, benign ovarian cyst and endometrioma, there was no difference between them. Another meta-analysis, it has to be updated. Dr. Muzi was here. Oh, okay. He is our honor. He is here. According to his meta-analysis, he says, and this 17 publication, around 1,000 patients, he says in the AMH is reduced in patients ovarian endometrioma with the, again, benign ovarian cyst and the patients with healthy ovaries. This is my clinical, not large, like Dr. Karaman, this is just a small group of patients and the more than 200 patients, some of them with endometriosis, some of them not endometriosis. I look at the rate of more than 5 centimeter endometrial prevalence is around 35%. How about the bilaterality? Around 20%. And these patients has the same laboratory AMH measurement. This is very, very important, same laboratory. And there is no difference in this infertile patients with or without endometriosis. And we look at the different age group. As you see, all the age group, the AMH levels were similar with or without endometriosis. For this lecture, we look at the, again, 150 patients, non-endometriosis, I mean, again, infertile patients to our clinic, and half of them with the endometriosis patients, and the mean age was 34 in the non-endometriosis patient. Endometriosis patients a little bit younger, 2 years younger, but if you look at the AMH levels, 2.2, nearly 2. There was no difference between them. This is the small data, but again, the other data, this data looks up that there is no difference between the infertile patients with or without endometriosis. The question is very, very important. She has low ovarian reserve. How about the oocyte quality? How about the pregnancy rate? There is no difference between them according to Rome data in Italy. Just one slide. What is the AMH level in adenomyosis patient? We have this publication, just 8 abstract, no publication, and it says probably reduced adenomyosis patient reduced AMH level, but in my practice, I believe this is vice versa. In adenomyosis patients has higher AMH level than the healthy controls. How about the antral follicle count? Is it reliable or not? As you know, we are using this one in our fertility practice, and it's important. What is the effect of the endometriosis on antral follicle count? According to this meta-analysis from China, antral follicle count is decreased. Endometriosis without endometrioma or the other side, and they say it's decreased antral follicle count. This is good study from the Tulane group. They look at the age and ovarian reserve in an endometriosis patient because it's overlap between them. It's getting older, AMH level is decreasing, antral follicle is decreasing. They say before age 35, and they looks like similar, a little bit difference, but after 35, there is big difference. I mean, the favor of the healthy patients. Another question, again, maybe endometriosis patients has a lower number of the oocyte, but the important one, they produce lower output rate. That's very important in our clinical practice when we're dealing with endometriosis patient. Another publication, 2 publications from France, they say, except the old mention, maybe high size ovarian endometrioma has the more AMH level, both of them from the Chaperon group and Horace Roman group, and the Nelson and Jonas say maybe we have to keep in mind in our practice maybe large endometrioma, but we don't know what does it mean. Large means more than 5 centimeter, 8 centimeter, 10 centimeter, maybe high serum AMH levels. How about the medical treatment? Is it useful or not? And we are using, as you know, in our practice, Dianogest. In this study, they say from academia, it is useful to use Dianogest for the ovarian reserve. Another is coming from the Japan. They look at the GnRH versus Dianogest. They say Dianogest is better than the GnRH for the reserve of the ovarian in this patient. Another study from China just published a few months ago. They say we couldn't see any difference with or without Dianogest for the ovarian reserve. And this one is one of the most important publications from Yonis. And they look at the endometrioma surgery. What is the effect of the AMH and antral follicle count? They say surgery is harmful for these patients. That's why Michel Nizol from Belgium, they say in conclusion, AMH should be included in the preoperative evaluation for each patient in women with endometriosis. Another publication 2 years ago, they look at again endometrioma surgery, the effect of the ovarian reserve. They say endometrioma surgery has the short, medium, and long term. That means 1 year has a detrimental effect for AMH levels. How about the cystectomy versus ablation? It looks like similar damage for the ovarian reserve. Another topic we have to discuss in a few slides, could we do sclerotherapy, either transvaginal or laparoscopic? If you look at the detail in the publication, in these publications, there is no difference for the ovarian reserve. And another one, and they say maybe it could be better with the sclerotherapy. Another one, again, from 2 years ago, they say there is no, and this is strategic review, there is no conclusive evidence sclerotherapy is better than the other modalities, I mean cystectomy, other ones. And this one is from, again, from Horace Roman. This time, laparoscopic approach for the sclerotherapy. Especially, they say, with the larger endometrioma, if you do the sclerotherapy by laparoscopy, the decrease of the AMH levels is higher than the other ones. It's important. This study is coming from the, again, Korea, but these are all of them radiologists, and they say, for the, especially for the recurrent endometrioma, you could use the sclerotherapy for the vaginal route. Maybe in the future, unfortunately, we don't have, maybe one of the microRNA, a biomarker for the AMH for the in the future, not now. This is from Barishata, one of the publications. I modified this one. Endometrioma itself, antral follicle count has limited value. AMH studies inconsistently, but it's the warning, maybe decrease the AMH levels, but the sizes may be important, but the surgery, either antral follicle and AMH level is decreased in surgery. My take-home message, and endometriomas affect the ovarian reserve either by itself or by surgery. And which test you have to use? Definitely you have to use AMH instead of the antral follicle. And before the surgery, and not early time, 1 year surgery to evaluate the AMH level or antral follicle level. And the World Endometriosis Society, in last year it was in Edinburgh, and next year it will be in Sydney, Australia, for the 16th Congress, and to discuss every issue about the endometriosis and adenomyosis. Thank you.
Thank you, Professor Oral, for excellent presentation. And now we can take 2 questions from home.
Anybody? No? So I want to ask Dr. Topbaş Selçuklu is here? No? No. I'm just saying that if she is here, because by now we got the information from the two previous speakers. So we have to know why some of the endometriosis ladies, women, are infertile. Because some of them are infertile. If the quality of the oocyte and everything is just okay with the IMH, and you see that there is nothing, so we have to try to describe what's going on, because starting at the very early morning, by now that we are just talking, including Topbaş, saying that lots of theories just may affect the infertility, or just some of them can be fertile, some of them cannot be fertile. Anyway, at the end of this session, we will have some time to talk about that. So we are just saying that the AMH is not very important, huh?
It's very important.
Very important. Okay, so we could include it in our practice. So sometimes, we cannot get really very big information if it's too high. So we cannot say that we will end up with the good number of the oocyte or the embryos.
AMH doesn't say about the future fertility, only
Yes.
say that our ovarian response. That's important.
Yeah. Yeah, anyway, so I thought something like that, because Professor Karaman said that, there is no too much increase the amount of FSH that we are using. Sometimes this is very important. Okay, so we are just getting the information at the end to discuss. Thank you very much.
Thank you.
So we are just moving to another side, because this is, Emre Papucu is a newcomer to our scientific world. So I think this is endometriosis and fertility preservation.
Yes.
Why we should do the fertility preservation if everything is just okay?
No, they are infertile.
Okay, please.
Dear Chair and colleagues, I would like to thank the organizing committee and society for giving the opportunity for being together with all the experts all around the globe in Istanbul. And also I want to express my warm regards to all attendees and Professor Engin Oral and Ümit İnceboz and all staff that have been prepared the organization. As we all know that endometriosis leads a chronic systematic inflammatory process, tubal ovulatory changes, and it's present up to 50% of infertile couples. The question is, should we offer fertility preservation in women with endometriosis? The question is yes, actually. I'm giving the answer of my lecture just in the beginning because it's a very, very important knowledge that these kind of women may experience severe infertility facts. So, other question is, should we offer fertility preservation for all women with endometriosis? And this answer is no. Why? I'm going to explain it. My flow is just like this: the evaluation of cryobiology, why to preserve, how to preserve, guidelines, and controversies. Evaluation of this technology have been developed starting from 1930s by discovering the more effective cryoprotectants. And the cryopreservation of the female gamete for fertility preservation occurred during the second half of the 2000s. There was a huge gap between the first reports and the clinical practice facts. So, concomitantly with the upsurge and escalation of efficient oocyte vitrification. So what is vitrification? It is a pseudo second order phase transition. In other words, converting a material into a glassy amorphous solid. To vitrify something, some biologic material, you have to use cryoprotectants. And among all, a lot of studies have been published so far. The one of the 15% ethylene glycol, 15% dimethyl sulfoxide, and the sucrose seems to be the best one in terms of efficiency. Accordingly, when we compare vitrification with that of slow freezing for both oocytes and embryos, vitrification survival rates are significantly higher when we compare it with the slow freezing. And as you all can see, the percentages of the fertility preservation procedures with respect to the total vitrification procedures, this EFP is the elective fertility preservation trend, is going much higher than the oncologic fertility preservation procedures. And let's look at the trend in the literature. Starting from 2000s, there is a tremendous increase in the published papers. So, as first baby born from this technique in 1999, in which conditions we offer fertility preservation, such as low ovarian reserve, uterine malformations, ovarian diseases, other benign gynecologic conditions, but the endometriosis and adenomyosis stands in the front. You know why? To preserve fertility. I'm going to explain Professor Gürgan's question in the upcoming slides. Because there is a fact that there is endometriosis-related diminished ovarian reserve by itself, not surgery. That might be oxidative stress, iron, mechanical stretching, or less ovulation, or less aspirated oocytes during assisted reproduction. Another aspect is the poor oocyte and embryo quality. It's somehow controversial, but in my opinion, the most important fact is the iatrogenic damage. In other words, the surgery. Because as we all know that the vast majority of the available evidence shows the classical laparoscopic stripping of ovarian endometriomas damages the ovarian reserve. Serum AMH decreases, ovarian function tests were compromised, ovarian response to hyperstimulation was halved, women operated, especially for bilateral endometriomas, entered menopause earlier, low but consistent risk of post-surgical ovarian failure. Is there any safe surgery? That might be. However, laparotomic approach, suturing, hemostatic agents, experience, it's not changing the fact that the surgery reduces serum AMH levels to 30 to 45%, and the risk of premature ovarian failure is nearly 3% in overall population. So how do we preserve the fertility? We have three major forms of fertility preservation. The first one is the oocyte, the second one is the embryo, and the third one is the ovarian tissue. I'm not going to tell more about the ovarian tissue cryopreservation because it's been published about a case series with prepubertal girls, published by Done and the colleagues with relatively successful results. But I will mainly focus on the oocyte cryopreservation in the endometriosis patient population. This is the first case report published in the FNS. This 25-year-old woman, of which they aspirated 25 oocytes. But till now, we don't know the prognosis of this case because she didn't return to the IVF center yet. This is the first landmark study that published by Cobo and colleagues from Spain. This study included 485 patients out of 1,000 women who returned to the same IVF center to thaw their vitrified oocytes, being fertilized, and to get embryo transfers. The patient's mean age was 35, and the great majority of the patients were the advanced stage. The return rate was 46%. The first important finding of this study is the overall high survival rates. It's 85%. And overall cumulative live birth rate is 45%. But when we look at the different age group, in other words, if we look at the younger women, the survival rates, collected mature oocytes, and the cumulative live birth rates are significantly higher when we compare it with that of women older than 35. What about the effect of the surgery? The women who did not have prior surgery revealed significantly more oocytes and revealed significantly high cumulative live birth rate per patient, according to this large observational study. According to numerous studies, they examined the fertility preservation endometriosis revealed nearly between 3 to 7 mature oocytes that were harvested and vitrified by using both agonist and the antagonist protocols. These are all the recent studies. And this is another recent observational study. This is another landmark study. This is 146 women, endometriosis patients, who underwent ovarian stimulation for fertility preservation. It's 56% had undergone more than one stimulation, more than 70% had at least one endometrioma lesion, and 36 had previous endometriosis surgery. When compared with women who had surgery, unoperated women revealed significantly more oocytes, more mature oocytes, and less canceled cycles when compared to the women with prior surgeries. And this study also reported an adjusted multiple linear regression analysis that the factors significantly reduced the number of harvested oocytes. The first one is the history of the surgery, and the second one is the woman's age. This is another study, a recent study about the fertility preservation. The research question was, what is the outcome of fertility preservation in women with endometrioma, especially those with larger than 4 centimeters? This was a retrospective cohort study that published in the RBM Online 2021. The mean age is 31, and 56% of women had at least one surgery. And as for the results, ages were similar, comparable. The serum AMH and the M2 oocytes were significantly higher with women without any prior surgeries. And this study also reported an adjusted linear analysis. It's interestingly, the prior surgery and the age were the two major confounding factors affected the numbers of oocytes. And when we put together these two recent studies, underlines the importance of the prior surgery and the age. So, note up to now, it seems better to implement fertility preservation just prior to surgery, especially for younger women, because the oocyte quality and the quantity are much more advantageous when compared to the older women. And the cyst diameter seems not to influence the efficacy of fertility preservation if this preservation carried out before surgery. So the important question would be, therefore, how many oocytes do we need to achieve a successful live birth in endometriosis women? This is another very important study from the same team from Spain. This is a retrospective study, including that 485 patients cohort with endometriosis who had their oocytes vitrified and went on attempt to pregnancy. And when we divide into two subgroups of younger and the older ladies, if you collect the three oocytes, the cumulative live birth rate is around 4 or 5% for both groups. But if you collect 5 oocytes, the cumulative live birth rate is 11. If you collect 3 more oocytes, 5 to 8, the cumulative live birth rate increased to 28%. If you collect 2 more, it's going to be 40%. A tremendous increase in each 2-3 oocytes. But this increase is not shown to be valid in older women. As for the conclusion of this study, the probability of live birth increases as the number of oocytes used increases in patients with endometriosis, but better outcomes were observed within the young population. It's consistent with the recent data from Dracopoulos that in every ovarian reserve group, if you collect more oocytes, it translates into more mature oocytes, and it translates into more cumulative live birth rates without making any plateau if you take frozen embryo transfer cycles into the game. So there have been some editorials, including Ertan Sarıdoğan as an author, that we need to adopt the fertility preservation as a routine practice. It was a letter, so I just searched the literature, and I found a survey that it's a fertility preservation counseling for women with endometriosis survey. It was an online survey. The nearly 60 participants from 13 countries just responded. 85% of centers offered fertility preservation for endometriosis, 32% all patients to endometriosis, nearly 30% only patients with large endometriosis. So, which criteria they choose to offer fertility preservation? The first one in place is age. The second one is the diminished ovarian reserve. And the third one is the unilateral or bilateral endometriomas. So if fertility preservation is suggested to patients with endometriosis, which is the method of choice? The majority of attendants replied that freezing of unfertilized oocytes. This is also the available international guidelines and recommendations on endometriosis, infertility, and fertility preservation. As we all know, this is ACOG, ASRM, ESHRE, but you know we have some recommendations on fertility preservation, yes, but no single guideline underlined any topic about the endometriosis. This is the only thing I find in the ESHRE's guideline about the endometriosis. Its relevance to future fertility is unclear, they concluded. So far so good, we concluded. However, do we have some concerns? Patients rarely receive reproductive counseling at early to mild stages. Mostly in advanced stage they got it. Oocyte retrieval performed in women with advanced endometriosis can be challenging. And spontaneous prognosis, what is the spontaneous pregnancy chance, conception chance of the couples with relatively normal ovarian reserve tests, normal semen parameters, and normal hysterosalpingogram? This is another recent letter. One of the authors, the Christian Becker, I think he is within us in this meeting. Maybe he's going to make his comment about this letter. They reported that there are no controlled RCTs. Cumulative live birth rates of operated versus non-operated seems similar. Young good prognosis women, we don't know the long-term prognosis, cost and psychological burden. If we count that how many million of women are suffering from endometriosis and how many of that will going to take the counseling or will going to afford the fertility preservation programs. I would like to recommend this article for our young colleagues for the fertility preservation in women with endometriosis. This is published in Human Reproduction by Somugliana and colleagues. This is a really nice paper, practical paper. In this narrative review, they put the assumptions used for the evaluation of the potential relevance of egg vitrification. The assumptions for endometriosis based on the level of evidence that endometriosis affects fertility. Endometriosis is a recurrent disease. Ovarian surgery damages ovarian reserve, and endometriomas damages ovarian reserve. These are consistent facts. And I think table 2 is very important, that the validity of fertility preservation in different clinical scenarios. They concluded that bilateral endometrioma cases and previous monolateral excision of endometriomas with contralateral recurrence. These are the two severe clinical scenarios that fertility preservation should be offered at least constant. For women with endometriosis and diminished ovarian reserve, bilateral endometriomas, and recurrent uni or bilateral endometriomas after surgery seems at least require fertility preservations. What we do, as in our clinic, besides, we have some medical legal restrictions in our country as well, because not every woman can vitrify their oocytes just depending based on their wish, because we have some restrictions. You should just document your serum AMH level just below 1 nanogram ml. It's a really big problem in our legislation system. If there is documented diminished ovarian reserve in terms of serum AMH, you can vitrify your oocytes. Unilateral or bilateral ovarian endometriomas with low serum AMH, we offer oocyte vitrification. For married women, unilateral or bilateral endometriomas, regardless of size and regardless of surgery history, just ask for the desire of the future pregnancy. If it's yes, then manage according to the ovarian reserve tests, hysterosalpingogram, and sperm analysis and symptoms. If it's going to be no, that, in my opinion, the critical point is here. If the woman is older than 35 and or low ovarian reserve tests, we should offer embryo vitrification. If the age is less than 35 with normal ovarian reserve tests, embryo vitrification can be discussed, at least in the assumptions and in the scenarios. In summary, patient's perception and wishes, the effect of endometriosis on reproductive outcome, the potential impact of surgery, additional individual risk factors such as BMI, smoking, etc. Baseline tests and the opportunity to cryopreserve oocytes should be taken into consideration. And this is my last slide. Don't forget, endometriosis is a progressive disease. Age is irreversible, and perfect is the good of enemy. So vitrification of a couple of oocytes can change the game. Better to preserve fertility in women who are at risk and elective fertility preservation instead of immediate surgery. Thank you for your attention.
Thank you very much, Professor Papuchu. Any question? So we understood that, you know, when we are just approaching young ladies, even if they don't want a baby or they are married, do not want a baby, or they are just single, we could offer them. The question is just when we are just deciding them to treat them with IVF after an expectant management, whatever, but it affects the age and the ovarian reserve. Two questions. The first one, when we are just offering them IVF, so I think do we have to go to fresh embryo transfers because we have the data, just embryo quality is just good and whatever. The another very important thing is just if we make the operation with the damage, I think there are some few data showing that when we get the oocyte and fertilize it and to get the embryo, it is different than the other causes of problems related to age. Even if we have lower oocytes, we could get better pregnancy comparison to the age-related problems. What do you think?
Let's answer the first question. There's an ongoing debate that whether we choose the fresh or the frozen embryo transfer in endometriosis patients. Up to now, no single guideline is recommending the frozen embryo transfer. However, if the data are going to be accumulated by more and more data based upon our clinical findings and the case reports and the observational studies in the literature, it's going to be changed. they probably will offer us to choose frozen, deferred embryo transfer instead of fresh, because, you know, endometriosis is a chronic inflammatory diseases, the estradiol-dependent diseases. So in a very high supraphysiologic estradiol levels, it's really irrational to put an embryo, blastocyst, into the endometrium. So we also published in the recent ASRM that 150 patients, something like that, the frozen embryo transfers implantation rates are pretty high than fresh embryo transfers. The second question is, yes, you are correct. It's a fact. If you choose surgery rather than in vitro fertilization, it might be a really good solution, especially for young women with relatively normal or high ovarian reserve markers. However, you cannot control the feature of that woman, because you might be a good surgeon. You may preserve the ovarian reserve by your technique, but it's not, you know, 100% guarantee that that woman will go into other centers or other doctors or might have other empirical treatments. So in my opinion, if you offer the fertility preservation by cost, and if it's okay for your legislation, it's reasonable to vitrify at least 8 to 10 oocytes, because in young age women, they strictly insisting on to put 10 to 15 oocytes into the tank to sustain cumulative live birth rates rather than surgeries.
One, I think Christopher Becker is here. Hi.
Yeah, Christopher.
So I think by now, Christopher, we only discussed the endometriosis and the oocyte quality and the embryo quality. I think it's the time sometimes to add something, which is the endometrial receptivity. There are some data showing that there would be some problems affecting the endometrium in the endometriosis cases. Sometimes we are missing some important point because we are just talking all the time about the oocyte embryos and maybe the genetic analysis of this, you know, embryos, blastocyst formation, even if sometimes we could get less implantation rate and increase abortion rates. Sorry, what's your idea? Because I know that in Asia, we are just moving towards sometimes the implantation, so we have to take care.
Yeah, thank you. I think it's complex. I think probably the main reason why some patients, and it looks like also from the data we've seen earlier, not everybody has an implantation problem who has endometriosis. I think some of the patients who may have that may also have adenomyosis. I think adenomyosis is probably the bigger problem when it comes to implantation than endometriosis per se. So that's my personal opinion. But the other thing is, I agree with what you said and how you presented it, that you have to have I think we should offer or explain to the patient, this is an option. You have endometriosis and it may have an effect on fertility in 30 to 50% or so. And it might affect in the future your ability to conceive. We have to be very careful that we don't say, because you have endometriosis, you're not having children, because that's a lot of patients are worried about this. You know, these are young patients often, and we don't have to necessarily push them into the corner. You need to preserve your fertility, but if we give them all the information, they can make their own choice and go from there. And it is a financial situation. If you're 20, 25, you probably don't have the money to pay for this. So that's
Dr. Becker, one small question. I'm sorry. What is your routine clinical algorithm for the must-have fertility preservation?
We don't have a clear algorithm. What we usually do, I mean, it's a bit wishy-washy, but what we usually do, if you have a slightly older patient with a low AMH, and I think I agree, like Michel Nizol was saying, you should have an AMH as a routine test in patients with an endometrioma for certain. So someone with a lower AMH, especially if she's slightly older, the younger one, well, you could argue also in the younger one, then you should probably suggest it a bit stronger than in someone who has a very good ovarian reserve.
Thank you.
Thank you very much, Christian. And Bülent, Orman. Sorry for saying the Christopher. Sorry. So, see, because I don't know, is it Christopher? Christian is better, right? Thank you. Bülent.
Regarding endometrial receptivity, I believe there was a relatively recent Chinese study that looked at frozen embryo transfers, all that were euploid by pre-implantation genetic diagnosis. So I believe that correction for euploidy is especially important if you are going to make any suggestion about any disease that affects endometrial receptivity. And that study actually did not show any significant difference between endometriosis and non-endometriosis controls. So endometrial receptivity may be affected, but I completely agree that you cannot dissect the effect of adenomyosis from endometriosis. So if a patient doesn't have adenomyosis, I don't think the endometrial receptivity is affected at all. And even in patients with adenomyosis, it makes a difference whether it's internal or external, whether you have diffuse or focal adenomyosis. So, I mean, that's my take on endometrial receptivity and endometriosis. My question to you is regarding your algorithm about freezing embryos or oocytes. And you said, I'm a believer in doing fertility preservation in patients with endometriosis, and all patients should be given this option or at least discussed, even patients who are younger, so and probably not married. So you said you would freeze only embryos after age 35, and you would consider egg freezing at younger ages. How did you end up with this algorithm, and depending on what data?
And the beginning of that algorithm, that was written married. Did you check that? For the married woman, we cryopreserve embryos rather than oocytes because it's not legal to freeze oocytes in the married couples.
How about married woman?
The married woman is that we should make the embryo cryopreservation. But
how are you sure that they're going to remain married in a year or two?
I don't know. It's my guideline. I think we should ask to the Christian Becker or someone else. But for unmarried women, yes, you're right. You can freeze oocytes. You have to freeze the oocytes, even in 40 years, even in 45 years. But if you are living and practicing in Turkey, besides the exceptional reasons such as azoospermia, legislations do not let you to freeze the oocytes because we face that issue. But I agree with you. If the woman is older than 35 years and not married, yes.
I don't think marriage makes any difference because the legislation doesn't forbid you to freeze oocytes if the woman is married.
I think it's the legislation restricts us.
No,
in my opinion.
No, it doesn't.
We have to double check that.
Yeah.
Can I? There is no legislation about this issue. This is on the gray zone. That's why we don't know there is the permission from the government marriage or not.
Yeah.
It depends on the clinical condition. It depends on your algorithm. But the problem is, I would like to ask the Oxford group, what is the rate of if you compare with the in this general population, not endometriosis patient, after the throwing the oocyte or embryos, the success rate, is there a difference between them, or embryos more than success than the oocytes one?
I wouldn't know, so I don't know what the local success rates are for this particularly in Oxford.
You don't know. But I believe embryos is more successful than the oocytes still.
Probably. I mean, we've seen the data. It's slightly better. What was it, 99% versus 95% or so. So that's right. coming back to how it is in the UK, so the HFEA clearly says that eggs belong to the woman, embryos belong to the couple, and if they separate or he says, no, they're not going back, then they're not going back. So I always counsel my patients, even if they're in a relationship or if they're in a relationship or if they're married, exactly about that, even if the male partner sits right next to it, because it is stressful and not every relationship survives this.
Waiting for us, Dr. Orman. So we will start the debate, right? So I think there will be a very important debate. Now we are starting. It is just the first deep endometriosis related infertility surgery as first approach. After that, Dr. Urman is going to say that IVF is the first approach, and then we will discuss which is the best. Okay, so Horace Roman. Buradan çıkmayacağım.
Vereceğim hocam. Siz oraya açabilirsiniz, oraya da aktaracağım.
Hi, Dr. Roman.
Yes.
We are in the, the meeting room, saying hi. So I think you finished the surgery, huh?
No, I have just no. I have just. I'm waiting for a place in the operative room because I have an emergency. A patient managed last week came with abdominal liquid collection, and I have to drain it before having a fistula.
It's no problem. So we are here, very big crowded here, waiting for you. I think you are a young, handsome surgeon. So we have the other people are waiting for you. So you will be talking about the deep endometriosis related infertility. Surgery is the first approach. And also there's a Professor Bülent Urman. He is a really good surgeon, but he's just talking about the IVF as the first approach. So there are two excellent surgeons. We will wait for you. What do you say?
Perfect. So can I share the screen?
Yes, you are here.
Perfect. So do you have everything in full screen? Do you see my screen, my slide?
Yes, we see you.
So everything is okay. May I start?
Yes.
Perfect. So it is my great pleasure to be with you online. I would have been more happy to be with you in person because I like very much Turkey and my Turkish colleagues, but unfortunately the job and the family do not allow us to travel as much as we would like. So it is my pleasure to share with you some thoughts about the management, surgical management of deep endometriosis related infertility. And you know that endometriosis is a chronic debilitating disease which leads to inflammation and sometimes is associated to infertility by several means, because it may attack the fertility mechanisms at every level. As most of women are young and with pregnancy intention, so we have to face with this dilemma how we manage her pregnancy intention or her infertility. You have to know that when you look at the literature about women with pregnancy intention, it is very difficult in different series to understand if all the women included in the series are actually infertile or presumed to be infertile and presenting with severe endometriosis. But anyway, you have to choose between surgery first or ART first. And to propose the surgery, you need some arguments. And amongst the best arguments, it should be that the surgery should increase the rate of natural conception. The surgery should not decrease the ART pregnancies, or even to increase the rate of ART conception. And also, surgery should improve the quality of life. And I will show you that the surgery may fill all these requirements. If you look at the ESHRE guidelines, the conclusions concerning women with deep endometriosis and pregnancy intention, the conclusions say that today we do not have evidence to state that the surgery improves the results, the pregnancy outcomes in patients who have IVF. But the guidelines open the door to the surgery if the woman suffers from pain, from unbearable pain. The problem is that women with deep endometriosis may present a very severe disease, but not be actually very painful. However, there are many other symptoms, or in my opinion, more severe than the pain itself. For example, a woman may have deep dyspareunia, cannot be able to accept sexual intercourse. However, they are not painful if they do not have sexual intercourse. And I am always amazed when I see that some colleagues do not take into account this impossibility to have sexual intercourse, because they consider that it is not painful if, of course, the sexual intercourse are not employed. However, patients who do not have sexual intercourse are infertile, for sure. Also, some patients are not painful, but they do not longer they cannot avoid empty the bladder, because deep endometriosis of the parametrium injured the splanchnic nerves of the bladder. For me, the bladder dysfunction is a major, very severe symptoms and requires surgery, even though the pain is absent. Other women with bowel endometriosis may not be painful, but may present a bloating, a continuous bloating, a permanent bloating, and they may also have occlusion. So the bloating for me is indication for surgery, even though the patient do not have too much pain except in the bloating. Again, do not wait for women with stenosis of the ureter to be painful before to perform the surgery, because you may have a silent kidney without any pain, without any lumbar hyperalgia. So this woman also require a surgery without being painful. Last but not least, some pain may exist, but may be not attributed to the endometriosis. However, when you have a compression of pudendal nerve or sciatic nerve, patients may be painful into the leg, into the buttock, or in the perineum, and this is actually a pain related to endometriosis. So I think that the surgery may be proposed in patients intending to get pregnant with this kind of symptoms and not only pain. But now the patient may ask you, okay, you have surgery or you have IVF. What is the pregnancy rate I can expect if I have surgery or if I have IVF? And if the patient presents with colorectal endometriosis, we can answer that if the patient has first-line surgery, the first-line IVF, the pregnancy rate may grow up to 70%. And we have two studies, two French studies, one from Cochin Hospital, one from Tenon Hospital, where patients with colorectal endometriosis had not surgery but first-line IVF. And if this patient have up to 4 IVF cycles, they may expect to have a pregnancy rate around 70% and a live birth rate around 64%. And you can see that most of pregnancies results from the first and the second IVF, while the third IVF is responsible for 10% of pregnancy and not more. So 60%, 60 to 70%. Now you can say, okay, if I perform the surgery, you may expect until 80% of postoperative pregnancy. And these results are taken from my randomized trial, ENDO, where patients with severe rectal endometriosis were randomized between colorectal resection or conservative surgery, and then digestive outcomes were compared. But this woman had also a secondary endpoint, the pregnancy rate. So all the patients had the surgery, and then we look at the pregnancy rate after the surgery, and they observed that among patients who intended to get pregnant after my surgery, 81% were pregnant with a majority of spontaneous conceptions. And if I focus only on patients with proved infertility before the surgery, meaning that had attempted to get pregnant for longer than 12 months, I observed that this woman were pregnant after my surgery in 74% of cases, and more than half of conceptions were natural. So for me, this study demonstrates that the surgery of colorectal endometriosis, even though very severe, does not lead to infertility, but increases the rate of natural conception. Furthermore, in this study, I compared women who were directed after the surgery to the IVF and women in which the natural conception was allowed. And I observed that when the patients tried to get naturally pregnant, they reached to be pregnant earlier than with IVF. Because the IVF, there were several appointments, and then the first IVF is not 100% of results. So if the patient wants to go fast to the surgery, it is faster with natural conception in my Syria. Now the patient will say, okay, you presented me results from Paris and results from Bordeaux, but the patients are not comparable. Do you have any study where patients managed in the same center or compared between IVF and surgery? And we have some studies. And one of them is this one, involving several centers in France, among which my center. We use the propensity score, and we observe that at any cycle, patients with surgery prior the IVF have better results than women with endometriosis and IVF. So this study suggests that higher pregnancy and live birth rates may be observed and the surgery precedes the IVF. Surgery well done, of course. Now we have 2 randomized trials. Right now, 2 randomized trials are ongoing where the patients with colorectal endometriosis are randomized between surgery and IVF. And our center participates to both of them. But the enrollment is not easy because patients who come to me have an idea that they need surgery, and the randomization is not easy to do. But we'll take the time we need to have a definitive answer if performing the surgery before IVF actually increases the pregnancy rate. Now, my colleague will try to support the first-line IVF will say, as many of our colleagues, that okay, surgery works, but there is a high risk of complications related to the surgery, of course. And now I'll try to see if this complications rate may balance completely the advantages related to the surgery. So is the postoperative complications rate so scary? In my opinion, no, because looking at our Syria in Bordeaux, we observed that after the colorectal endometriosis surgery, we have 3% of bowel fistula, 2% of abscess. But conversely, we have this result should be put in balance with high postoperative pregnancy rate with a majority of natural conception and the long-term improvement in pain in quality of life, because now I'm preparing the 10-year follow-up of this randomized trial. And I can tell you before the publication that the good result of the surgery observed 2 years after the surgery is constant over years until 10 years after the surgery. So the surgery is efficient in terms of pain quality of life and pregnancy, while the rate of recurrences is very, very, very low. But even though the patient has a complication, it does not mean that she's completely and definitively infertile, because we also published another study where we look at the pregnancy rate in patients with postoperative complications. And we observed that this patient who had a second surgery due to complications had a pregnancy rate 42% after 5 years postoperatively, with a majority of natural conception. So in my opinion, the risk of complication cannot balance out the advantages related to the surgery. Now we can also see the question on the other side. Let's say that we choose the IVF, first-line IVF, but is it actually and always safe to postpone the surgery for years? And in my opinion, no, it depends on the disease. Because my colleagues from Aarhus assessed what's happened when women with colorectal endometriosis had first-line surgery, and they observed that if you carry out 1, 2, or several IVF cycles in women with colorectal endometriosis, you may expect to have an impairment, such an impairment of symptoms that you have to stop the stimulation. You may have an occlusion and subocclusion. So you may, by performing the IVF, you may induce a subocclusion or an occlusion and have a surgery in emergency, which is, of course, less favorable than the planned surgery. So take care with the IVF in patients with severe colorectal endometriosis. And then we published a study where we had a look at what's happened with the nodule in patients who had not surgery, and we observed that in 40% of women who have not continuous amenorrhea because they try to get pregnant naturally or by IVF, in 40% of cases there is an obvious growth of the nodule during 3 years. Now, the take-home message is not let's perform the surgery in all the patients with severe endometriosis and pregnancy wish. Not at all. My message is that we have to listen to patients, we have to assess our deep endometriosis, and we have to propose a custom-made management. We have to rule out this fake news, which is that the surgery impairs the fertility. You're too young to have the surgery. If you have the surgery, you will be infertile. This is a fake news. So we have to propose something which is suitable in each patient. That's why my advice is if you go with the IVF, first-line IVF, please, it would be welcome to start by completely assessing your deep endometriosis, then to discuss your case with a surgeon, but a surgeon with experience in deep endometriosis, because you ask advice for a surgeon who never carry out the surgery, he will answer you, oh, it's too complicated, do not do the surgery. So discuss with surgeons experienced in endometriosis, and then give a pill continuously until the surgery. So do not leave the patient without pill between two IVF. It is difficult to give a definitive advice, and it is very difficult to state who is right and who is wrong, because we are several specialists around this patient, and we are look at the patient from a different angle. Me, I'm a surgeon. I have a high rate of prevalence of severe cases. I carried out a subocclusive endometriosis every week. So, of course, in which each woman with deep endometriosis, for me, it is potential evolution, very, very severe disease. The ART specialist will keep in mind the good results of successful IVF in this woman. We say, okay, but we have very good results and we have never had occlusion or something like this. Of course, but he's looked only at the fertility outcome, while the gynecologist or GP has a high prevalence of minor or moderate cases, in my opinion, cannot take the right dimension of the problem of patients with very severe deep endometriosis. So it's not astonishing that we do not agree always on the management of this patient. That's why we have to build pluridisciplinary meetings, but with experienced surgeons and experienced ART specialists. So thank you very much for your kind invitation.
Thank you very much, Dr. Roman. After 900 cases of colorectal surgery, it is easy to talk like this. Let's say, listen the same story from.
We will take questions after the debate. And now, the next speaker is Professor Urman. The stage is yours. Thank you.
Thank you very much. Hi, Horace. So I will try to make the opposite statement, saying that IVF is probably a better option than doing surgery, especially in patients with deep endometriosis-related infertility. So, so I will defend the IVF side, and hopefully at the end I will conclude that current evidence does not support the performance of surgery to enhance fertility in patients with deep endometriosis, unless surgery is indicated for other reasons, which I will touch upon at the end of my talk. So we have to understand the fact that endometriosis actually affects the performance of ART at different levels. So initially you have folliculogenesis, fertilization, embryo development, and afterwards endometrial receptivity, placentation, which may affect the course of the pregnancy, fetal development, and finally the born baby. So if you look at deep endometriosis, there are studies, especially preclinical studies, suggesting that deep endometriosis might have an effect on all of these. So, and there are also confounding factors when we take into account the results of the surgical intervention, especially for endometriosis. So what are these confounding factors? Some of which we have discussed in the previous session, such as adenomyosis, which is very difficult to dissect off endometriosis, especially given the fact that they quite commonly coexist. And the other one is the presence of endometriomas, which again coexist with both severe endometriosis cases and also with adenomyosis. So you have confounding factors that will affect at several levels. For example, if you are going to do a deep endometriosis surgery, it's probably relatively rare that you will ever find a patient with a solitary nodule and nothing else that is wrong in the pelvis. So most often you will have retrocervical endometriosis. Most often you will have adenomyosis at the posterior aspect of the uterus, and most often you may have endometriomas that will change the course of surgery that you are going to perform. So when you are initiating surgery for deep endometriosis, let's say a patient has endometriomas on both sides, what are you supposed to do? Leave the endometriomas behind and not touch those endometriomas, see what happens afterwards, which will increase the recurrence risk of the disease because you're not taking the whole pathology out. So if you look at the outcomes of IVF and ICSI in endometriosis compared to non-endometriosis controls, this is a relatively new study that lumped together almost all of the studies in the literature and shows that there is a slight decrease in the clinical pregnancy rate in patients with endometriosis. And also, together with a slight decrease in pregnancy rates, there is a slight increase in miscarriage rates and also a slight decrease in live birth rates in patients with endometriosis undergoing ART. This is the study actually I mentioned about in the previous discussion session. What the authors actually did was in frozen embryo transfer cycles, they replaced euploid embryos in patients with endometriosis and compared the implantation rate of these embryos with control patients. And there appeared to be a slight decrease in total aneuploidy, but if you looked at chromosome abnormality rates in these blastocysts, there was no difference in patients with endometriosis and in patients with controls. So endometriosis does not appear to affect at least the euploidy of the blastocyst. And if you take into account that this is one of the most important factors that determine implantation in women with infertility, you would concur that without correcting for aneuploidy, any study that you do on endometrial receptivity and also the success rates will actually be less than optimal. So there's limited evidence also showing that surgical excision of endometriosis actually improves implantation. We saw that Dr. Roman showed a few studies, and there are a couple studies underway that have been registered to clinical trials, but I do concur that patient recruitment would be very, very slow and difficult, especially for surgical studies. So until the results of those studies are published, it's very difficult to say which should be the first line. And adenomyosis is really very important. If you look at pregnancy rates, miscarriage rates, and live birth rates in patients with adenomyosis, you will see that this is actually the pathology that affects the outcome much more than endometriosis itself. And we know that deep infiltrating endometriosis is associated with adenomyosis in over 70% of the cases. So you don't just have deep lesions and have a completely normal pelvis otherwise. This is an Italian multi-center study that looked at pregnancy outcomes in women with endometriosis who conceived naturally versus controls. And if you look at the outcomes, there was no difference in live birth, miscarriage, or ectopic pregnancy rates in these patients. These are patients who conceived naturally. And again, the same study showed that there was a slight increase in preterm delivery rates and admission to the NICU, but all of the other parameters that they examined were the same. But if you take cases with adenomyosis, and if you take especially cases with diffuse adenomyosis, then it's a completely different story. For example, as you can see over here, the risk of placenta previa, cesarean delivery, preterm delivery are all significantly increased when the patient has a severe form of adenomyosis compared to patients who are free of the disease or compared to patients who have slighter versions of the disease. And the increase in placenta previa has also been shown in other studies, as you can see over here. The problem is we do not know whether surgical excision of endometriosis actually rectifies this, whether it decreases the chance of these abnormal mishaps during pregnancy. And also placenta accreta is increased in these patients. And this is a large study that put everything together, showing that placenta previa is increased. But it's not only in patients with endometriosis. It's also in patients with endometriosis who conceive after ART. It's also in patients who conceive spontaneously. Endometriosis patients also have an increased risk of placenta previa. So the question is whether if we do surgery, do we improve spontaneous pregnancy rates? This is a very old study that was published almost 20 years ago. It's a study that looked at the cumulative 24-month probability of becoming pregnant in patients who were treated for deep infiltrating endometriosis by laparotomy versus controls. So this study actually showed that treatment of these patients at the end of a 24-month follow-up period did not make any difference when expected management was taken as a control group. So the other problem is if you look at studies that were performed, especially surgical studies, we are not exactly sure whether all patients subjected to surgery were initially infertile or not. You see, if you take into account patients who got pregnant spontaneous or with IVF, irrespective of the fertility status, then you will see approximately a 40% chance of getting pregnant in patients with advanced stage endometriosis after surgery. However, if you consider only fertile patients to start with, then the chances of getting pregnant is approximately 25%. This is another recent study that was done in patients with endometriosis. These are patients with rectovaginal endometriosis, with or without an endometrioma, and the authors looked at the cumulative pregnancy rate at 12 months after surgery or expected management, and surgical group actually fared better than the expectant group. So in summary, in patients with endometriosis, it appears that surgery may improve pregnancy rates, but this has to be proven in randomized studies, which we are lacking currently. So does deep infiltrating endometriosis surgery improve pregnancy rates with ART? This is the other question. So until the results of these two randomized trials that have been registered but not completed are out, we are left behind with unfortunately poor quality studies in the literature that have severe biases. And these are the studies that report that previous surgical treatment of severe endometriosis actually improves pregnancy rates when done before ART. But as you can see over here, these studies, all of them, have many biases, especially related to selection and allocation. And two of the studies over here also included patients with endometriomas in the non-surgery group. So this may have decreased the chances of getting pregnant in those patients. That is also, unfortunately, an end result of retrospective studies. So we know that both pathologies, endometriosis, deep infiltrating lesions, adenomyosis, and endometriosis coexist. So how should we deal with them if we do surgery? We don't exactly know. We know that excision of endometriomas is associated with decrease in ovarian reserve, and this is true irrespective of who does the surgery. Even very experienced surgeons, when perform surgery, will eventually have the patient with a diminished reserve at the end. And if you prefer to leave the endometriosis lesions behind, let's say you do the surgery for a deep infiltrating lesion, but with the fear of decreasing the reserve, you don't touch the endometrioma. And this is sometimes what the patients actually ask of us, especially when we do surgery, let's say for a hydrosalpinx to improve implantation rates with IVF. Then the patients ask you, what are you going to do to my endometrioma? Are you going to take it out? Because if you're going to take it out, then I may end up with a decreased reserve. So this is a very difficult dilemma, because if you take, let's say, the deep infiltrating lesion or the colorectal lesion, but leave the endometrioma behind, then you are beating the philosophy behind the surgery by leaving the pathology behind and giving the patient a very high risk of recurrence in the future. And we know that adenomyosis is associated with endometriosis in over 70% of the cases. So what are you going to do with adenomyosis when you do surgery? That's the other important question. Are you going to remove it? Are you going to leave it behind? And how is this going to affect your future pregnancy rates? One subset of patients that I would agree with Horace, especially for doing surgery, are the patients who failed IVF. But I also believe that one should at least give a chance to doing a euploid embryo transfer and making sure that failed IVF is not due to embryonic aneuploidy before embarking upon surgery. And another option is to freeze embryos, send them off for PGT, see the end result, and then do the surgery in these patients, because the surgery may include also ovarian endometriomas and may hamper the ovarian reserve. So the other feared complication of not doing surgery and directing the patient to IVF would be increased risk of complications during pregnancy. So this is the other one. So if you look at Horace's argument that he published in 2015, he's saying that delaying surgery may lead to bowel occlusion during pregnancy, higher rates of radical colorectal procedures, increased postoperative morbidity, prolonged painful complaints. And he also states that to provide a definitive answer, you would need a very large sample size if you do a randomized control trial. So these are the patients, these are actually the case reports that we have in the literature in patients with endometriosis and what happened to them when they are not operated and subjected to IVF. Unfortunately, there are only a few case reports, and we can't generalize and we can't give a certain percentage of the complications that may arise in these patients. But this study shows that if you look at the pain symptoms, they do not change when you do IVF. So the patient does not have an increased pain score after performing IVF. It's interesting that if you look at the complications of delivery in the endometriosis and in control groups, that if we exclude some of these over here, which are statistically significant, I would like to draw your attention to what happens during cesarean delivery. In patients with deep endometriosis, few patients over here, as you see, 2 patients necessitate necessitated a hysterectomy. One patient had a hemiperitoneum. One patient had a bladder resection, bowel resection, and the other one, and 2 patients had bladder injury. So these are actually serious complications. But we do not know whether doing surgery prior to IVF is going to decrease them. We know that endometriosis in its deep form is going to increase these complications, but we do not have any results as to what happens after surgery. There also are a few cases of spontaneous hemiperitoneum, as you can see over here, bleeding from decidulized vessels on the uterus, and it may be severe at times, but one study looked at the incidence of this, published in FNS in 2000 study from the Italian group, and they showed that they had only 1 case of spontaneous hemiperitoneum in pregnancy, corresponding to a percentage of 0.3. Does IVF increase the severity, or does it make deep infiltrating endometriosis more severe? This is another question that remains to be answered, and we do not have any data on this. But we do have data on the outcomes of colorectal surgery, which even in very experienced hands is not without any complications, as you can see over here. So rectovaginal fistula, anastomotic leakages, anastomotic stenosis, voiding dysfunction are all real complications of doing surgery. And imagine if somebody who's not experienced, who does the surgery, I'm sure that these complications will be even more increased. So this is my last slide. I would suggest doing surgery only in patients with intensive pain, interfering with their daily activities and precluding intercourse, patients with uteral constriction or obstruction, patients with impending bowel obstruction, failed IVF cycles, but I do believe that these should be corrected for aneuploidy prior to giving consideration for surgery, and concomitant pathologies that may decrease the success of ART. For example, if the patient has a hydrosalpinx or inaccessible ovaries, but even in patients with hydrosalpinx, maybe it's a better option to cryopreserve eggs or embryos prior to embarking upon surgery. Thank you very much.
Next. Thank you very much. I think at the end of the discussion, Dr. Roman is following us.
Yes.
I think you come closer for the special agreement. At least we have to select the patient to choose the treatment option. I don't know. Do you want to say something, Dr. Roman?
Yes. So I refer patients to IVF specifically when they have no symptoms, no pain, but also not symptoms which I presented previously. Particularly if the ovarian reserve is not very high and not very low. So an ovarian reserve, an AMH between 0.7 to 2. If, of course, the husband has some sperm abnormality, so I can say that I refer to the IVF maybe 15% of patients with deep endometriosis, the pregnancy intention. Now, when I perform surgery, of course, I make the difference between the surgery of deep endometriosis and the surgery of ovarian endometriosis. So I can be more radical on the deep endometriosis, and I also know only drain the ovarian endometriosis if the IVF is most likely after the surgery, because in this case I try to keep the ovarian reserve as high as I can. And of course, I also refer to IVF patients who have between 38 to 40-year-old, because in this patient, if I perform the surgery and I have a complication such as fistula, I may lose 6 to 12 months before to refer them in IVF, and this time may be very important in the probability of the pregnancy. So I have a lot of arguments which I put together in order to balance between surgery first and IVF, but in most of cases I propose surgery.
Thank you very much. Do you have any contradiction to the last slide of the Professor Urman? He put everything together, huh? Anyway, so we are waiting. If you want to say something, I don't know, you have seen the last slide. And what do you say?
I did not see any slide. I heard Professor Brun, but I did not see any site on the conference room or on his slide.
Thank you.
Because your camera was switched off.
What do you say, Dr. Urman, at the end?
Well, I mean, I do lots of endometriosis surgery, and I actually enjoy doing surgery. So, and I also do lots of endometriosis, deep endometriosis surgery, including colorectal involvement. But if we are taking into consideration, which the initial question is, IVF or surgery for deep endometriosis related infertility. If we are just taking this question into account, I mean, just for increasing the success rate of the patient, I mean, just increasing the reproductive outcome in terms of pregnancy rates, I believe IVF is a better option. And some of the slides that Horace actually showed, and I know in one of the studies where they looked at, first they did surgery and then 1 cycle of IVF, 70% of the patients got pregnant. In the other group, 1 cycle of IVF, 35% of the patients got pregnant. But if you look at the end of 4 IVF cycles, so the chances of getting pregnant in both groups were the same. So the question then arises, is it better to do surgery, IVF, IVF, or IVF, IVF, IVF? I think doing 1 more extra cycle of IVF will eventually yield the same result as doing 1 surgery and 1 IVF. So I think it's going to be the same. I mean, the number of interventions is going to be the same.
Yeah,
the
main issue is just, you know, not everybody is an excellent surgeon for the colorectal surgery. It is another, and it's the cost and whatever they have to think. I think I agree with you that, you know, we have to select, you are just saying, depend upon what is the instrument in your hand, because IVF treatment is just very common. Lots of centers can provide it to the patients. The last question is just, if all the time we are just talking the failure of the IVF, but if we could get 2 good, you know, aneuploidic, you know, euploidic embryo transfers, the pregnancy rate is very high for those patients we saw today. Do you have any idea that if the pregnancy happens, it may make a positive effect to the endometriosis cases, the deep endometriosis?
I am not sure about any data as to what happens to deep endometriosis after pregnancy and lactation. But given the fact that both of these are a high progesterogenic environment, I would presume that the lesions maybe would regress, especially in patients with endometrioma. What we see if we do a cesarean section, that the endometriomas are nearly invisible when you do a cesarean section. And sometimes you have to really look for it to find it. And knowing that the patient had initially an endometrioma of 4 or 5 centimeters, they practically disappear at the end of the pregnancy. And then the following lactation period is also a suppressive period because of the high prolactin levels and because of the hypoestrogenic environment. I don't know what Dr. Roman thinks about this. I mean, is there a study looking at what happens to deep infiltrating lesions after pregnancy?
Could you repeat the question because I did not understand? So, deep endometriosis?
The pregnancy, pregnancy, yes, may have a positive effect to the deep endometriosis.
Yeah, of course. I think that the 9 months amenorrhea has the same effect on endometriosis as 1 year of generate analogs of 1 year of pill, but it is a very transitory effect. So, of course, I think the team of Chaperon showed that after the pregnancy, the nodules may be reduced by, I don't know, 5%, 10%, not more. But this is very a temporary evolution. They do not disappear and they do not dramatically decrease. But I forget to mention that I have another very specific group of patients where I propose the surgery. They are the patients with very low AMH. Patients with AMH lower than 0.5, where we know that the IVF does not provide good results. I think that the surgery followed by natural conception may provide good results, and we demonstrated it. We reported this in a paper in Fertility and Sterility, where in women with severe endometriosis and surgery and baseline AMH lower than 1, the pregnancy rate was 68%, with 2/3 of pregnancies natural. So in this patient, the surgery may try to put the spot on the natural conception, as well as the IVF is less efficient.
Thank you very much, Urman. Is okay?
No further comments.
Thank you very much. Okay, thank you very much, Dr. Roman. Hope to see you in Turkey live next time.
Enjoy your weekend and your congress.
Thank you. Bye-bye.
Thank you. Goodbye.
And now, last speaker of the last session is Professor Dr. Ümit İnceboz. And Professor İnceboz is talking about adenomyosis and fertility.
Mr. Chairman, dear colleagues, thanks for being here at this time. It's quite late, I know that, but I'll try to be focused as much as I can. I'll try to finish earlier, hopefully. As you see, we have been dealing with million-dollar questions from the beginning of this meeting, and this is the last bit of million-dollar question. It's adenomyosis and fertility. What is the current status? Although adenomyosis has been implied by Karl von Rokitansky by 1860 and has been defined as it is now in 1972 by Charles Bird, it has long believed that it's the disease of multiparous women at older ages. There have been some case reports, but in 1995, there was a report linking adenomyosis and infertility. They were strongly suggested that there should be a link. What they have done was they have looked at 26 patients, and they have checked with MRI. They found that an adenomyosis was quite high in that infertile population. It's approximately 54%. Interestingly, what they have also found that endometriosis was also there, but apart when they exclude the effect of endometriosis, they have found a strong link between adenomyosis and infertility.
So in my talk, I'm going to cover the possible effects of adenomyosis on fertility, and also does adenomyosis actually affect infertility, and how can we manage fertility in women with adenomyosis? And the first one is possible effects of adenomyosis on fertility. Actually, Fitnat has covered some of those. Let me go through once again. We can divide into two parts. The first one is abnormal uterotubal transport. It can be due to anatomical distortion similar to fibroids. This may also block tubal ostia, as well as may affect some sperm transportation. And also there may be disturbed uterine peristaltis. It has been shown that invaginated endometrium also cause abnormal hyperplasia of the myometrium. This may cause hyperperistaltis of the uterus, and this in fact, in turn, may affect sperm transportation. And destruction of normal myometrial architecture has been also shown. Single myometrial cells in adenomyosis are different from normal myometrial cells. So this is another factor may affect peristaltis of the uterus. And the second possible effect, second group of possible effects of adenomyosis is altered endometrial function and receptivity, in other words, implantation. Obviously, it has been shown that aberrant endometrial metabolism, it has been shown that menstrual blood estrogen level is higher in patients with adenomyosis. Also, cytochrome P450 expression is increased. So there is abnormal estrogen metabolism there. And also inflammatory response, it has been shown that macrophages are increased. So it may affect inflammation. And also altered estrogen and progesterone receptors, it has been shown that estrogen receptor alpha expression is higher in adenomyosis. It's actually different from endometriosis, as said early in the morning by Doug. And also progesterone receptors alpha and progesterone receptors A and B they are decreased as well in all layers. So there should be ENP receptor problem as well. The other one is altered uterine oxidative stress. It has also been shown that reactive oxygen species are increased in women with adenomyosis. And also impaired implantation can be explained by reduced, or it can be shown by reduced implantation markers. Basically, leukemia inhibiting factor is one of them. And lower HOXA10 is important substance, obviously, for implantation and also embryo development. So as seen here, there are many theoretical mechanisms that can affect fertility in women with adenomyosis. Does it really affect fertility in reality? I mean, does adenomyosis actually affect fertility? Well, it is really difficult to study because, as told many times today, adenomyosis is highly correlated with other fertility affecting factors like endometriosis and myoma. And also there is not universally accepted criteria and classification for diagnosing adenomyosis. Since the correlation with high correlation with endometriosis, myoma, and adenomyosis, that's what we named in one of our articles with Professor Engin Oral, we named it Dragon's Triangle. So let's come back to the question, does adenomyosis actually affect fertility? Until now, there is no human study exploring the effect of adenomyosis on natural conception. We don't know it. But there is one study on baboons. What they have done was they have looked at 37 baboon with adenomyosis and 38 without adenomyosis. And they have searched in necropsy material, and they found that presence of adenomyosis is really a strong problem for primary infertility, lifelong primary infertility, even in the absence of coexistence of endometriosis. So this is the animal model showing natural conception is decreased with adenomyosis. How about the human? Well, the best model would be ART cycles, and this is the study from Pados and co-workers published in 2023. They have looked at 5 meta-analyses, 2 retrospective cohorts. The first one of those is from Vercellini 2014, and it has been shown that patients with adenomyosis has decreased clinical pregnancy in IVF treatment cycles. This is the other one from Nurgianakis, published in 2021 in RBM Online. What they have done was they have separated the IVF treatments according to the drugs that are being used. So they have looked at long regimens, ultra-long suppression, and also checked short generate and also antagonist protocol. What they found was short and antagonist protocol gave the lowest rate of pregnancy. So according to this last meta-analysis from Pados, it is for sure that there is lower implantation, lower clinical pregnancy, and higher miscarriage rate in women with adenomyosis, according to ART cycles. Well, one question would be, is the negative effect of ART on ART cycles due to adenomyosis or adenomyosis plus endometriosis? It's not absolutely solved yet, but there are some studies that I'd like to show you. This one is from Vercellini's meta-analysis in 2014. In their meta-analysis, they have performed meta-regression of 2 studies that they have included. And they found that if there is adenomyosis there, then the clinical pregnancy rate is quite low. So adenomyosis does affect fertility in ART cycles. This is from Sanita Sharma group. They have done very clever study. They have looked at tubal factor as a control group. They have also included endometriosis only, endometriosis plus adenomyosis, and also adenomyosis only. And what they found was, if there is adenomyosis in the field, then the pregnancy rate is lower. So, sorry. What they have done was, when they include adenomyosis, group B, C, those were with adenomyosis, the clinical pregnancy rate is lower. And also live birth rate is lower and miscarriage rate is higher. So when there is adenomyosis, pregnancy rate, also miscarriage rate has been influenced. Also, what they have done was they have looked at ultrasonographic criteria. When there are more criteria, then the prognosis is worse, pregnancy outcome is worse. So they have compared only three criteria, and on the other side, all 5 criteria. When there are 5 criteria, then the pregnancy outcome is worse.
it means that if there is higher severity of adenomyosis, then the outcome is worse. Obviously, obstetric outcomes matter in adenomyosis. Our aim is not having the woman reach to pregnancy, but also obstetric outcomes are quite important as well. This has been shown by previous speaker. From Vercellini group, they have published a paper looking at association of endometriosis and adenomyosis with pregnancy and infertility. Regarding to adenomyosis, pregnancy rate after ART and live birth rate after ART decreased, miscarriage rate is increased. When we look at the obstetric outcomes, it is interesting. Perhaps it's difficult to see. I would probably summarize here. Significant increase in preeclampsia with adenomyosis, more than doubled preterm delivery with adenomyosis, threefold increase in postpartum hemorrhage, which is logical indeed, in adenomyosis, and significant increase in small for gestational age babies. So there is some problem with implantation, or there is a bit of problem there. How about placenta previa? It has been shown that if there is adenomyosis plus endometriosis, then placenta previa risk is higher. But we know that endometriosis go along with high risk of placenta previa. But in terms of adenomyosis, if there is endometriosis as well, plus to adenomyosis, the placenta previa risk is increased. Severity and location. In terms of severity, I have mentioned if the there are more sonographic signs, then the risk of infertility is higher. How about the location of the adenomyosis? It's very difficult to study, really. But there is one study from Chaperon Group, 2020. They have looked at with MRI, and what they found was with focal adenomyosis of outer myometrium was identified as an independent associated factor of infertility, primary infertility. So it is important to see focal adenomyosis of outer myometrium. Another question would be regarding to infertility and adenomyosis. Does adenomyosis cause low ovarian reserve? For this question, I didn't bring the data that Professor Oral showed. In that data, in 2022 data, they have just briefly looked at the abstracts, and the results was correlated just saying this with surgery or with HIFU as a treatment option do not cause any harm to ovarian reserve. But when we look at the ovarian reserve, it is difficult to look at because we know that those ladies are a little bit high ages, at older ages. According to the studies, approximately 20% of women with adenomyosis are younger than 40. And also, concurrence of endometrioma may affect ovarian reserve. However, there is one study, but this is not the this is not their aim, but they have found women with adenomyosis had higher mean antral follicle count. That was the side finding of their study, but interesting. As Professor Oral said, we also see that higher antral follicle count among those ladies. How can we manage infertility in women with adenomyosis? It is important to note that there is no medical treatment option that can relieve symptoms of adenomyosis while still allowing them to conceive. Instead, the treatment options only do suppression of the lesion. So we don't have any magical treatment that we give to a woman and then expect her to be pregnant. One question would be, how about ovulation induction and intrauterine insemination? Would it be an optional treatment for adenomyosis? There is no data on this, but there is one poster presentation presented in 2005 in ASRM from Arıcı group. They have noticed that very low pregnancy rates, 5%. So it is logical to omit this when we consider the mechanisms of implantation in women with adenomyosis. It will do no benefit. We don't have any benefit from this kind of treatment. In terms of treatment modalities in adenomyosis, obviously there are many treatment options. However, when we include infertility, the options are less and less. Perhaps you may see the asterisks around. These are the only treatment options, proven treatment options for infertility plus adenomyosis, namely levonorgestrel IUS, GnRH analogs, not antagonists at the moment, so we don't have enough data, perhaps in future. Complete resection or cytoreductive surgery, in other words, surgical treatment and HIFU. Perhaps radiofrequency ablation might be included in those treatment options. Let's go through quickly. One option is levonorgestrel IUS. It has been shown that if we do an IVF cycle and divide the ladies into two, one of those get IUS, levonorgestrel IUS, for 3 months before frozen-thawed embryo transfer, and the other ladies go directly fresh embryo transfer cycle. That's what they have done in this study, and they have found that higher implantation, clinical pregnancy, and ongoing pregnancy rates with levonorgestrel IUS before transferring the embryos. How about GnRH analog? It is an unresolved problem, really, but most studies showing that if we use suppressive therapy after having the embryos, then the outcome is better. This is one of those examples. They have looked at fresh transfer cycle without treatment, fresh transfer cycle with GnRH pre-treatment. Before the cycle, they have treated with GnRH. And what they have also done, they have do the IVF and freeze them all, and then give GnRH analog, and then do the transfer. Which is better? The last one was the better one. So if we freeze all embryo and then give GnRH suppression, then the outcome is better. How about conservative surgery? We have to select the patients. Before my talk, as you know, there's a debate regarding to deep infiltrative endometriosis. It is similar to that, so we have to select the patients properly. It has been shown that it is suitable for younger women, and it has been shown that if the age is over 40, then the pregnancy rate with surgery is around 4%. So there is no point to do surgery after older ages. And if there is uterine cavity distortion, or if there is recurrent implantation failure, then we can consider surgery for those ladies with adenomyosis and infertility. We have to bear in mind that in diffuse form, it is difficult to do the proper surgery, even in very experienced surgeon hands. In diffuse adenomyosis, the risk of rupture is around 4 to 6%, approximately 6%. So we have to definitely mention this to the woman when we offer surgery. And also, sufficient contraception should be discussed. We don't know how many months later we can let her have the pregnancy. but it is approximately 6 to 12 months after surgery. It is generally thought that it is 6 to 12 months. This is the paper from Osada, 2018, and he has gathered all the knowledges, all the papers, and looked at the surgical management. And what he found was pregnancy rate differs quite a lot, and miscarriage rate is around 1/3, and uterine rupture is 3.6, and treatment failure approximately 10%. So we have to bear these numbers in mind. Obviously, as Osada's paper pointed out, there are factors affecting uterine rupture, method, degree of extirpation, extent and size of uterine muscle defect, method of reconstruction, postoperative wound infection or hematoma formation, the period of contraception after the operation, and obviously the skill of the surgeon. These may affect the uterine rupture. Let's come to the other treatment option. It's HIFU, high intensity focus ultrasound. And focused ultrasonographic beams reach to the target, and when the temperature is above 65, there is a coagulative necrosis of the lesion, and that may affect the therapy. I mean, this is the method of therapy. According to the literature, it is very limited, but it is promising. It shows it is very good in infertile patients, but we need more data. And radiofrequency ablation is the same. We don't have much, but according to short studies with limited number, it is promising as well. Perhaps in future we'll hear more about it. So in conclusion, according to the literature, adenomyosis, and also according to the real world that at least I always face, adenomyosis negatively affects fertility in terms of decreasing clinical pregnancy rate, increasing the miscarriage rate, and also poor obstetric outcome might be expected, like preeclampsia, preterm delivery, small for gestational age, and postpartum hemorrhage. Generic analog suppression is advised after IVF for 3 to 6 months. Most studies is saying that 3 months, but it's arguable. Levonorgestrel IUS can be offered for those ladies before embryo transfer. Surgery for focal lesion can be offered if the age is less than 40 and if the lesion is focal. And HIFU or perhaps radiofrequency ablation seems promising options. Thank you very much.
Thank you very much, Professor İnci. Excellent.
Oh, thank you.
I tell you that, you know, it's really very good. Any questions? I think you are one of the organizers, so we are waiting. Do you want to ask anything to the people? You know, if they don't ask anything,
no,
you could select
perhaps,
one person.
Perhaps they may add something, or instead of asking questions, perhaps. No, I think it's too late.
but it's not too late. But it's adenomyosis is a new perspective for all of us. That lots of accumulating data is coming. You review very well. I think we have to be a little bit quicker, not to wait too much. The diagnosis is very important. When we diagnose it, when they want a baby and the pregnancy, we have to, you know, be a little bit earlier to offer them.
Yes. One message would be if you find endometrioma on a lady, definitely you should check for adenomyosis as well. So that's one of the message perhaps we can take home.
Yeah.
I think there's one question. Sorry.
Yeah, where is it? Okay.
Thank you very much for your nice lecture. I would like to learn your experience about the subcutaneous progesterone-releasing implants, not for infertility, but for the other symptoms of adenomyotic or endometriotic patients who cannot have intrauterine device, levonorgestrone-releasing intrauterine device implanted.
Right.
Is it clear?
The question was the progesterone implants and their effects on endometriosis and adenomyosis. Not included the fertility patients.
Sure.
Although my topic was on adenomyosis and infertility, I briefly answered this question as we don't have enough data yet. It is promising as well as I know. Perhaps any of the audience might add something. Yes.
Thanks.
Thank you so much for this question. Normally, we use that one for pelvic pain in some diffuse adenomyosis cases. Normally, in that cases, many colleagues apply the intrauterine device, but expulsion rate is a little bit higher, you know, especially if the patient have a diffuse adenomyosis and the cavity is getting smaller. In that conditions, when you insert the device, some patient haven't had good tolerance to insertion. We have some...
Sorry, I think it's implanted.
Implant of the progesterone. That's it.
Some women cannot use intrauterine device, levonorgestrone-releasing intrauterine device. In that cases, we may use subcutaneous implant.
I explained that one.
Sure.
Sorry.
Yeah.
Thank you.
So we use it in some severe cases. It is working well, but very low cases. I cannot say that we use that one over 30 cases, but it is working very good. Promising.
I just want to add something. With the implanted progesterone, there sometimes be very horrible bleedings that is sometimes very difficult to manage. They suppresses endometrium quite severely, and recovery of the endometrium may take some time, really. But it's not the obviously literature, but it's just my experience, perhaps. Hocam,
I think we have to finish it. Is it? Okay.
One more question or a comment, actually. So I think there was a recent publication by Jacques Launay where they used linzagolix, GnRH antagonists in adenomyosis patients, only a few ones, where they were able to shrink the volume of the uterus quite significantly. Now, whether that translates into better live birth rates remains to be seen, but maybe there's something in the future.
Yeah, it would be quite interesting to see the results, really. Thank you very much for your contribution.
Okay, so yeah. But anyway, I don't want to talk too much. Okay, it's finishing because I think the involvement of the junctional zone is just very important for deciding or about the pregnancy rates too. Because this is the good, let's say, the MR or the the diagnosis, and to see what's going on, it affects too much to the pregnancy rate and the abortion rates.
Yes.
Yeah. Okay. Thank you very much for staying here for a long period of time. See you tomorrow. Bye-bye.
Can I announce something? Please, before leaving, can we have a photo all together? Because some of us will not be here tomorrow. So it would be very nice to have a picture reminding today. Please do come over here.